Todd, do you think we should go ahead and begin?
Yep, I think just maybe give me 30 more seconds. I think set this
Okay.
up, and you're now co-host, Ashvin, just for FYI. So both
Sounds
of you.
good.
And then give me maybe a few more seconds here.
As people are joining in, see a comment. Is there a tweetable content policy? Yeah, we'll go over all the rules. I'll have Leo go over the rules before we start and have to interact with us.
Yep. Yeah, and actually the chat's going to be disabled now. Sorry about that, everybody who wants to use that. But yep.
Okay, Ashvin, why don't you go ahead and get started? I think we're straight.
Yeah, thanks for everyone to participate and joining us in today's symposium. Before we start, Leo, do you want to go over what participants how they can interact with us? And
yeah, sure. I'll just go over a couple quick notes. The first note is on the question and answering. That's the main interaction is basically going to be asking questions to the speakers. And for that, you should see a little Q&A down at the bottom of the screen there. And you're going to ask questions in that, and then people can vote on those. So there should be a little upvote, thumbs up button. So everybody can just basically look at those while the talk is going on and ask your questions through that. And then once the talk is over, I'll ask, depending on how much time is left, 1 or 2 or 3 questions to the speaker from the list of the highest voted of those.
Okay, and then we'll repeat that instructions throughout the meeting for people who are joining in later too.
Yeah. Yep. And for the speakers, just in the interest of time, we have a pretty packed day today. So my plan is I'm going to just interject when there's about 2 minutes left before we have to turn it over to the next speaker. And I'm just going to let you know, hey, there's about 2 minutes left. So, if you want to have any question time, you want to wrap up pretty soon after that. Yep. Great. Thanks.
So yeah, my name is Afshin Beheshti, and I'm from NASA Ames Research Center in KBR. And my co-host for this moderator, which he set everything up here, is Dr. Todd Triagen from Rice University. And we thank Rice University for allowing this platform to happen. So I'm going to just give you guys a brief introduction of what we're going to talk about, what COVID is, COVID International Research Team, and what to expect today. So as you can see here from the agenda, we have as Leo had said, we have a nice packed agenda of speakers that are going to be talking about COVID-19 related research. And also, as you can see, we got some very nice talks coming up from very good researchers here. And then we got a panel session at the end to with the hosts here to actually discuss topics. We had universal topics. We had themes of today's meeting, and the audience can interact that way with us also. So as I said, my name is Afshin Beheshti. I'm at NASA Ames at KBR. I'm just going to give you a brief update or brief preview of how COVIRT, the COVID International Research Team, started, and where we're going, and then what to expect from us, and how you can participate with us also. So there's an initiative at NASA called Gene Lab, which is here. People can go. So basically, Gene Lab is set up to be where any type of omics bioinformatics, you can deposit it there. It's open source, open science, and anyone could go in there and actually mine any type of omics that are related to space biology. So basically, if a PI or if a researcher had did some space biology research and had omics, it's there. So people can actually mine that data, come up with new hypotheses, and so on. So briefly, within NASA Gene Lab, we have groups called analysis working groups. And I lead the multi-omics group, which you could go to this site and find it. And within our group, we have a lot of talented, we have about 60 members currently, and we have a lot of talented bioinformaticians, computational biologists, and so on. And a meeting when COVID-19 started, unfortunately, taking off, I asked several members if they were working on COVID-19, and Todd was one of them, and Chris Mason was another one. So we decided to form a subgroup and start together, hopefully tackling COVID-19, see if we could come up with new ideas, new theories, new hypotheses, and help the overall COVID-19 research for the scientific community. So our group started on March 25th. Todd and I are the co-leads. Goal of COVID is to be open science. So we think that basically the only way we could actually solve this and come up with a vaccine as a whole, as a society, is if we tackle COVID-19 as an open science initiative. So we share data, we publish, if we're going to publish something, we can put it on bioarchive if we want to for our publications and so on. So, and the only way I think, we can actually do this individually, it's going to be difficult, but as a whole, this is how we're going to move forward. Right now we have 90 members and we're still growing, and we got people from all around the world helping us out, and we're all working at a pretty fast pace and efficiently together. And we're also, open for new members to join, and so if people want to help us out and think they can contribute, contact Todd or I, which you'll have our contact information throughout this meeting. And, we could decide if you think you could contribute to the overall work and the group pushing forward would be great. So these are some of the members I'm going to go over. So as you can see, we got a nice representative from many people from different institutions. Deanne from CHOP, she's done a lot to help us out with, coming up with nice little platforms where we could share the omics data together and analyze things. Chris Mason, but she's going to give a talk after for our first speaker. He's doing this massive RNA-seq data project, which he'll talk about, and the data we're going to be analyzing as a group. So thanks to him. Todd's lab, Krista was initially involved, is one of the founders also of this group that, she's been instrumental and so on. So we got all these members here, Matt, and then you can see a lot of people here like Matt Freeman, who's a chronovirologist with our group, and many other people who are involved. Here's the rest of the group. It takes 2 pages. So, and of course, some of these people are going to give talks, and people who want to actually approach us, Todd and I later, we could actually, discuss people interactions. So as I said, how it started was Todd, Michael, and Krista had this nice resource set up, and it's publicly available. Anyone here, go to this link. And they were trying to find publicly available data. There's not much out there for omics. So they were able to actually get this data and use it as a resource, and this is where we started. And so this is some of the data people are going to present is based on this public data that they were able to find from Wuhan infected patients, and also like controls that were from other places. As Chris will talk about, he has some resources, which I don't know if he's talking his talk, but there's something coming that he's shared with us. It was exciting, which is tracking time, doubling rates, and mortality rates in different counties. He can talk about it more. He's contributing some data to the Trevor Bedford's Next Strain platform. Kyle Henderson is in our group, and he's going to be releasing this soon, which is, it seems like a very useful platform for actually tracking how COVID-19 is occurring throughout the world. And he's actually has some nice clinical correlations, which people can actually use to say, if what certain counties or countries or cities have more effectiveness to curb COVID-19 infections and not. So it's a really useful platform. So keep an eye out when they actually officially release that one too. So goals of our COVIRT, our COVID International Research Team, is this: basically, enhance public data sets for everyone to use eventually, once we get all the stuff, and determine potential optimal targets and drug targets, and then, test these newly developed therapeutics, and then eventually apply for human trials. So basically, today's presentations, you'll see people from our group presenting various aspects of this and their own work that they've been doing and theories. And we're on social media too, so that's our presence. So I'll switch this over to Todd now, because he's gonna have some introductions, and then we'll switch over to our first speaker, Chris.
All right, thanks everyone. Sorry for that minor delay. Let me share my screen here. So first of all, I just want to thank everyone for attending. Thank Afshin and everyone who made this happen. I'm not going to talk about myself much, but I just want to highlight I'm an assistant professor at Rice University in the computer science department here in Houston, Texas. I work on computational biology, metagenomics, and microbial genomics problems. And just very excited and super enthusiastic to have everyone here today. Okay, and just high level, and I haven't. We've been trying hard to keep track of this. There's been a lot of enthusiasm to join the meeting, which has been awesome. We've been keeping this updated, but what you have on the screen right now is a map of the location of all the registered attendees as of maybe 30, 40 minutes ago. Maybe not fully up to date, but pretty close. And it's great to see we have representation from 13, 14, I think up to 15 countries across the world. primarily, the large majority of participants are coming from the US, but nice international participation, good participation from several countries in Europe and et cetera. So I was very excited to share this. We'll provide this information after the conference with updated statistics, but this was really exciting to see this nice international participation. And then overall, just a breakdown in terms of participation by kind of sector, if you will, mostly coming from academia today, maybe about 60%, but we have 9% of the participants are coming from government, and then around 30% others, most of which are industry, and then some were just kind of hard to kind of decipher, but overall it looked like mostly industry and then some other participants, which is just, again, amazing. This is really nice to see the spread of participation across government, industry, and academia. So one of the highlights, I'd say, and then listing of some of the institutions that people provided when they were registering. So I know it's hard to read, but just to give you kind of a sense. And with that, I just want to get out of the way of the symposium. Really great lineup. Very thankful to everyone who's speaking today, which were listed on the screen, especially to the co-lead of COV IRT, Ashvin. But I'm just really excited about the speakers we have lined up today and the panelists. Just all around excellent, and thanks to all of them for agreeing to spend time with us here today, and et cetera. So just to end here, I just want to end on some highlights. As of just 10 minutes ago, we actually hit 800 attendees for the event registered, which is awesome, and spanning I think 18 countries and nearly I think over 100 institutions. And then before I hand it over to Leo to get to the scientific content, I just wanted to start with some acknowledgements here. And of course, the co-lead, Ashvin, who's just been amazing, helping all of this get off the ground, and we've been just been a pleasure to work with him in all of this. And then I just want to thank the Rice Computer Science Department and George R. Brown School of Engineering, been very supportive of this event and helping me get things organized and supporting me in this endeavor to bring community together on this very important issue. Also want to say thanks to Rice Ken Kennedy Institute and the Rice Bioengineering, also provided support for this. And then call out a few names. Elisa Yusi, Deanne Taylor, and Sonia Vilpol for symposium organization. They've just been really helpful, but not just them, more, but I wanted specifically how to highlight their help. And then of course, all the COV IRT members who have been working hard, contributing, actively participating over the last several weeks, since March. And of course, all the 21 speakers and panelists. Again, this wouldn't happen if it weren't for you. And so thank you for your time. And then of course, thank you to everyone who registered to attend. It really helps kind of create additional momentum, enthusiasm, and see so much engagement and interest in this event. So thank you to everyone. And with that, I'll hand it off to our moderator for today's event, Dr. Leo Elworth, who's a postdoctoral scientist in my group at Rice University. And yeah, I'll just hand it over to Leo and let us get started. So Leo, take it away.
All right. Thank you very much, Todd. And let's go ahead and get started with our first speaker. Dr. Mason, if you want to go ahead and share your screen and unmute yourself. The only thing I'll say before we start real quick is for question asking, again, there's a Q&A box in the bottom, and you can vote for the questions you want to see the most to have asked, and then I'll ask those at the end. So our first speaker, Dr. Christopher Mason, is an associate professor of genomics at Weill Cornell Medicine and the director of the World Quant Initiative for Quantitative Prediction, as well as an affiliate of the Memorial Sloan Kettering Cancer Center, Rockefeller University, and Yale Law School. Welcome, Dr. Mason.
Great. Thanks so much for having me. And I'm going to move some things around my screen here. Can you hear me okay?
Yeah.
Okay, great. Put this down here. Yep. So,
okay, looks good.
And I have another talk I'm giving at 11:30, so I'm going to be brief. And then if I don't get all the questions, I'll be happy to follow up by email later today or chat with people about these data, which are also being shared amongst the COV IRT consortium. So I'll jump in and just dive right into the data. So I'll do a quick background and then jump into some sequencing we're doing on a variety of sample types, as well as a new assay for rapid diagnostics, and then quickly end with the website that Ashvin was referring to. So first, I want to thank basically everyone from Path and Micro who made this possible, plus everyone in the Mason Lab COVID team, which is rapidly expanded. And of course, New York Genome Center, Biosha, and also New York Genome Center, a lot of collaborators really helped make this possible. So I want to thank them first. So I won't belabor this point because I think everyone is very familiar with this website and seeing the increased number of cases. It's one of the biggest health threats we've ever faced as a species. And it's not just our species. Of course, there's even tigers that might be having some of the coronavirus. So it's an interesting time. I think it's at this moment that everyone probably looked at their cats if they have one with a quizzical stare and maybe turned their head like this cat was. And so it's an unusual time. But this has given us also an unusual time to look at what happens when the sequencing, it can reveal changes in the host as well as the viral dynamics during infection, and seeing where it is, and then how we can also think about ways to track it faster. So in particular, we've been thinking about the problem a little bit like this, is that ideally you'd have really comprehensive as well as fast methods to characterize these samples. Since it's a once in a century pandemic, we should have sort of a once in a century unified and I think global response, and also thinking about how do we characterize these samples at great depth so we can learn as much as possible about this virus to hopefully avoid the next pandemic. So I'll start with this set of data here and come back up to LAMP, and we're doing RT-PCR on all these samples, and also doing some testing of twist capture panels and other amplicon methods like the Arctic primers, which I won't talk about today, but we're testing basically all these methods as a means to better understand the virus as well as the host response. So in particular, some of the first data that we've generated, which is in its way being shared with the whole consortium, is basically a mixture of COVID positive and negative patients with a total RNA-seq, meaning we do a ribodepletion for the host and look at all species of RNA that are there and do paired N150 reads. We've also done some environmental sampling, some pilot data from our Metasub consortium, which I'll show you, which are basically seeing is there presence of the virus in public surfaces. And also we've done some controls, so synthetic RNAs, negative controls, and also virus-6 cells that are infected. And you can see here the percentage of, I'll come back to this plot a little bit more detail, but you can see the positive samples in red have a lot more of the virus present, sometimes as much as in the 60% of the total RNA from a sample is positive. But we do see sometimes these are RT-PCR negative and RT-PCR positive. These are control samples, positive and negative, and these are the subway samples you can see are very low, basically in the noise. So we don't think there's any, at least in our samples yet, that we found the virus there, as a quick tease for the end of the talk. But there are some cases where RT-PCR thinks it's negative, but we're seeing pretty high abundance of and coverage of the virus. So but it seems to be relatively rare. There was one paper that estimated 30% of RT-PCR was false negatives. We don't think it's that high. It might be as much as 3 or 4%, but I don't think it's as high as what that paper predicted from what we can see so far. And so what we've seen is if you take all the samples and you map to archaea, bacteria, fungi, other chordates, humans here in green, and then the virus here in this kind of tan, you can see for the RT-PCR positive samples for P and negative for N, and these are environmental samples, and those are positives. You can see some of these big spikes. So basically, these are the ones that are probably some RT-PCR false negatives, but they're pretty rare. And here's sometimes where we have really high abundance of the virus in some of these clinical samples. So this is interesting to see that from this metatranscriptomic profile, we can see mostly human bacteria and viral RNA, but we do see an ability to see lots of other species, which is if I had a lot more time to talk, I'd go into detail on the sort of microbiome we're detecting. That's a longer discussion, but we can actually, as you can see here in blue, see lots of the bacteria present in these samples. The other thing that's kind of really cool about the total RNA-seq is it is a method to actually get pretty full coverage of the genome. You can see in some cases like here, you don't always get the full length of the virus, but generally we do for about 90% of the samples, we get complete coverage of the viral genome. So the advantage of total RNA-seq and why we like it is you get actually a good host response, which I'll show you shortly, but then you also get the viral genotyping data to figure out the strain and where it's coming from. So we've seen so far that the RT-PCR essentially is pretty highly correlated with the total RNA-seq data. You can see here it's an R-squared of minus 0.78, so pretty correlated. Here's the ones that are beyond the 40 CT cycle, the CT threshold for RT-PCR, but we think these are actually positive from the NGS data. Again, relatively rare from what we've seen, but sometimes we do see it. And these are kind of in the high CT or low viral titer range, and so these are kind of a bit in the noise range from what we've seen. So again, getting back to the sequence data, what we can do is actually sequence them and line up all the variants we're detecting across these samples and actually compare it to the Wuhan strain and see these variants. This is what is in next strain, which I'm sure most, actually Afshin just mentioned this, but we can quickly characterize our variants relative to the public databases and see we're spiking and seeing consistent presence of some of the same common variation that's been previously observed, which is not unexpected in this case. And we can see here, so far in red, most of the samples in New York are enriched for Europe and some from Asia. You can see down here in the purple area, this is next strain, and in blue here is where the Cornell samples are in New York Presbyterian. So we can see this sort of red swath here looks like there's a lot of enrichment from European samples, and some from Asia is where we predict where these are likely come from. Keeping in mind that every phylogeny is a hypothesis and not necessarily a gospel, but it is sort of what we see so far. Then this actually validates what's been posted just about a week ago from the Mount Sinai group, is that they also saw most of it coming likely from European origins, which is good to see. So the other thing we looked at in the data is, there was evidence in the early outbreak in Wuhan that the L strain was more abundant at 70% of the samples. We see the same thing here, except our enrichment looks to be about 90%. So we think that the majority of the samples that were in the New York City outbreak were from this L strain, which currently has a pretty debated functional significance, but at least as a genotypic difference in sort of phylogenetic and geospatial difference, we can see it here in the New York City data sets. And all these figures are actually for a paper that's hopefully almost done by tonight if we make good enough progress. So now, as I was alluding to, it's not just targeted amplicons. We're also, you can see what else is there. This is from Trevor Bedford's site, which does this really cool Seattle flu study. And you can see all sorts of other viruses that can pop up. And so we wanted to see, do we find them in our data set? Here you can see all the differentially expressed host genes. This is for RT-PCR positive, sort of high, medium, and low abundance. And then these are negative samples on the left. And you can see up here other RNA viruses that are popping up sometimes at really high abundance. And so this includes things that are not surprising from the winter flu perspective, like other coronaviruses, influenza, some rhinoviruses, even some enteroviruses we found in some of these patients. And so that's another interesting thing to see. We can trace and then discover some of these other viruses as well. So on the host side, we can actually see thousands of differentially expressed genes. Most of them, there's this core set of upregulated genes here in orange for basically high, medium, and low viral count versus just positive versus negative, this sort of consistently upregulated set of genes in particular. Some consistently downregulated, but not as many. You can see the sort of overlapping heat map. And what these are sort of pathways that wouldn't surprise anyone in terms of sort of interferon response, differential signaling in response to virus, but also some ribosome profiling, ribosome variations as well. So this is what we're seeing is so far the pathways that seem to be up and down regulated for their enrichment score based on GSEA. And again, for people who have looked at virus samples, these aren't too necessarily surprising so far, but it's good to see them validated in the cells that we get from the NP swabs. So what we're doing now is thinking about why did some get sick and some not. Some people took arms against the sea of troubles. If any of you like Hamlet, some people didn't have to as much. And so this is kind of a question for the Host Genetics Initiative. It's also a large-scale international network of people collaborating and sharing data to look at the host response. There's also a COVID-19 effort from Rockefeller looking at human genetic variation. So these are ongoing questions. And so if you think about going forward, I think we don't just sequence the RNA, which I've just shown you a lot of data of things you can predict from the RNA, but ideally we look at genetic variation also linked to hospital record data going forward. So we can actually get a much better sense of what's likely driving risk and also response to some of the viral infections. Okay, so a quick bit of environmental profiling. What we've done so far is leverage a consortium called Metasub, which we started 5 years ago, mostly focusing on environmental surveillance, and then using this to also track RNA variations around the world. So in particular, we're collecting about 10,000 samples that should end at the end of May around the world, including 12 hospitals, and looking in public surfaces, especially to see what's the presence of the virus in these different areas. As I kind of alluded to, there's not much evidence of it so far yet in New York City, but we do see other RNA passengers like influenza again, and also some other phage viruses, rubella virus, also some tobacco mosaic virus. So we do see other things kind of hanging around in the subway, but so far no evidence of SARS-CoV-2, which is so far good news. But this project's ongoing, so stay tuned for some of this analysis. Okay, so last two things I want to go through is a LAMP assay we've developed, which is something called loop-mediated isothermal amplification. First published in 2000 and been commercialized by New England Biolabs in 2014. But basically, you make nested primers that target a specific piece of DNA and actually have a strand displacing polymerase that quickly makes copies of your target DNA with these set of 6 primers that make loops. And so we've been working with primers that were published first by Nathan Tanner, and then also been optimizing these. But you basically set up this target for this nucleocapsid gene. And when you do it, this is basically how it looks. If you take synthetic RNAs, if you do 1 million copies and do log 10 dilutions down to 0, at 0 minutes, everything's pink. It's kind of like a, something you do for a pool chemistry reaction, then it starts to become yellow. And then eventually you can see between 10 and 100 viral copies where we think the sensitivity is for the assay. And we've repeated this on different kinds of synthetic RNAs, plus on the clinical positive samples where we can see it. And so far, we can see it's pretty correlated. Again, this is the same plot as before, but now with LAMP data added on top. And so we can see pretty good correlation in terms of the fluorometric readout of LAMP versus these other methods, which is pretty promising. And also has pretty good sensitivity and specificity. You can see up here with an aggregate about 94%, as low as 78% for the really low viral copy number samples. And so what we're working on now are these two things to say, can we set up pop-up clinics and pop-up labs around New York City and basically get everything together to put up a little LAMP testing site, maybe do it in Walgreens or City MDs. This is an ongoing project with the city council and the mayor's office to start to set this up. And the other thing we'll close on, since I'm short on time here, but I'll try and keep us on time, is this. So the other thing we've just launched is clinicalhorizon.com, which is a site I think a lot of us are trying to think about ways to visualize the data, and you can basically do a detailed analysis of different cities. So you can see here's the virus landing on the membrane. You can pick basically any city in the country and play with the data in terms of where the confirmed cases and deaths. You can kind of subset the data, select it. You can play with this sort of feature here if you want to see essentially in a real-time dynamic by city, state, or country. Where do we see high rates of doubling? How is it populating around different places around the world? And then, for example, if I want to know in the United States and China and select these data and populate these locations, for example, what does it look like in New York City? And when do we think that maybe New York City might start to look like, for example, most of China, in terms of you can change it from logs basic, for example. You could play with these data and they're real time and dynamic to see what are the recovered, confirmed, and active cases around the world. And so again, this is taken mostly from the Hopkins site, as well as some data from New York Times and CDC. And so it's a variety of data sources, but it's a quick way to play with the data in a variety of ways. You can even, if you want to have like a play through the history of how things have moved around the country and around the world, you can also play with that data here. So encourage everyone to play with the site. We just launched it. There's a bunch of other parameters here you can change and filters that are up here. And you can also look at location trajectories for one site or a global overview, and also US data. So please take a look, please give us feedback, and that's basically it in a nutshell. And wait, before I finish, I also want to thank industry, academic, and government partners, and Councilman Ben Kalos, and thanks to my daughter, who's been very tolerant, and my work spouse wife. Everyone has a work spouse, and I also have a home spouse as well as a work spouse. So I think I've got a double spouse, which is actually kind of quite nice. So that's it in a nutshell. And here's the obligatory screenshots in case the site crashed during a demo. That's it in a nutshell. Happy to share the slides, get them up online. Thank you so much for your time and attention, and I'll talk to everyone hopefully by email.
Thank you very much for that. All right, let's get a couple questions in here. First question, let's just start with a quick one here. Can you talk about the cost of the sequencing? How much does it cost? So the question, for instance, to get like 10x coverage for the whole genome, or just in general, the cost of the sequencing?
Yeah, great question. So it's about $500 per sample to get what you need there. So if you can sequence deeper to get more, but that's about that for the cost.
Okay, great. Next question, is there a link between clinical outcomes and co-infection with, for example, influenza?
Great question. So far the answer is no, but we've just started looking at that. And we don't have enough cases, but we'll know a lot more of that. So far it doesn't seem to be. If anything, it seems like the people that have SARS-CoV-2 do not have some of the other viruses in general. It's pretty rare. There's a JAMA paper that indicated it might be about a third or a quarter of the time. We're seeing more like 5 or 10% of the time. So it's less common so far from what we've seen.
Next question, can you determine the potential infectability from detection of a viral genome in the environment? Does the presence of the viral RNA indicate it might infect someone basically from the environment? And I might just follow that up, since there's not a whole lot of questions with, maybe if you want to just talk a little bit more about the environmental sample collection and maybe like the benefits of that.
Yeah, I think so. So far from testing in Vero 6 cells, it seems like a lot of them are not infectious. They're not fully encapsulated virions, but they are just traces of the RNA that's been degraded and left behind. So that's what we're testing now with Vero 6 cells on additional samples. And since I have to go talk at 11:30, I might run quickly to the next talk, but happy to stay in touch. I'll be back on the conference as soon as the other one's done.
Okay. All right.
Yeah.
Thank you very much, Chris.
Great.
Thank you.
Thanks, everyone.
For that. All right, Matt, if you want to go ahead and start getting set up there and share your screen. Our next speaker today is going to be Dr. Matt Freeman. Dr. Matt Freeman is an associate professor at the University of Maryland School of Medicine. He is a virologist specializing in coronavirus pathogenesis and therapeutic development. His lab uses both cell culture and mouse models to study SARS-1, MERS, and now SARS-2. His current focus is on developing therapeutics to combat SARS-2. Please welcome Dr. Matt Freeman.
There we go. Can you see the slides now?
Yeah, great, thanks.
Great. Hi, everybody. So I'm Matt Freeman. I'm at University of Maryland School of Medicine, and I am a coronavirologist. So I've been in the lab. We've been doing this for 10 years now that I've been at Maryland, and then previously at University of North Carolina, where I was a postdoc. And so I'll tell you a little bit background about COVID-19, as you've all seen it on the news, and a little data about our drug screening efforts that we're currently working on right now. So just to start with acknowledgements, really the lab has been pushing this really hard over the last several months. All the people in the lab are at the bottom here that have been really doing all of the work as a really team effort on all the screening as we get through drugs, antibodies, vaccines, and then animal models in the near future. We've also had a really large amount of collaborators that have we've developed over the last 3 months, amazingly enough. So we're pushing really hard on all of these therapeutic angles, just the drugs I'll tell you about today. So just a brief intro. I know if you're interested in this topic, you probably already know this, but coronaviruses are generally RNA or are single-stranded positive sense RNA viruses. We have 4 coronaviruses that give us a common cold every year called 229E, OC43, NL63, and HKU1. And then the other coronaviruses that are more highly pathogenic are SARS, MERS, and now SARS-2. These highly pathogenic ones are BSL-3 viruses, so to handle them, you have to have a special room and a negative pressure room for BSL-3 work. If we look at phylogeny, this is an older picture now, a couple years ago, of alpha, beta, and gamma coronaviruses, kind of all splayed on this this dendrogram. And what you find is that with the woman sneezing here are where the seasonal coronaviruses are that give us a cold. And SARS is up here on the upper right, and MERS is here on the upper left, showing that they are more pathogenic coronaviruses. And while SARS and MERS are related in the sense they're both beta coronaviruses, they're not sequence related at all, compared to the other coronaviruses. And SARS-CoV-2, our new virus, really fits right into this SARS coronavirus 1 clade, where we see it's really a bat SARS-like coronavirus that fits right in here with the other bat coronaviruses that we know of from the past bat sampling. One of the big questions lately has been about all these other seasonal coronaviruses. What do they look like in the wild? This is a paper from several years ago, but it shows the data that we know of and happens every year, which is basically we get seasonal infections during the winter between September and basically late March for all of these coronaviruses. And so some years are worse than others. Some with these viruses, some depending on the site, the location in the country where these are taken. These were out of Colorado. We've done sampling here at University of Maryland in the dorms at University of Maryland College Park. And it really depends. We get the same type of curves. It depends on the outbreak and what the seasonal coronavirus is at the time. Depends on what you are actually seeing in the wild. But we get them all the time. Even though, and everyone in this that's listening to this is seroconverted for all of these viruses, but we still get infected, which is a big question that we're trying to figure out how that happens right now. A quick slide on the origin of this new virus. The first report was December 30th and 31st on ProMed, where there was an outbreak of atypical pneumonia. And then on the 31st of December, the market where this initial outbreak in Wuhan was identified was closed. And the report initially was an atypical pneumonia. The virus was identified about a week later as a novel coronavirus, but not the sequence of the virus wasn't released until January 11th, where it was very clear that it was a bat SARS-like virus right in the phylogeny of what we think it probably looks like. Out of the data that's come out so far, if we map the mortality versus age of SARS, MERS, and COVID-19, what we see is that it really tracks, COVID-19 tracks with what we know from before, where the elderly are most susceptible for lethal disease. If we mapped severity of disease on top of this, what you'd find is that it really tracks back much lower in the age groups. So up until even 20 or 30-year-olds with COVID-19, you see more or less what you'd see for SARS here in red. As this outbreak has emerged over the last several months, what we find is that as the cases in China have increased, we now have outbreaks around the country, as you know, around the world, with doubling time in the exponential phase really every 2 to 3 days. This map was taken from about a week ago up through April 8th, where we really see potentially a peaking in the United States, but that's still yet to be determined if we really are, how broad this initial peak is going to be, and if we're going to see a downturn in the next several weeks. There's been a really nice data set at Hopkins put out recently looking at the county-based readouts of cases, and what you can see, you really drill down to where the hotspots are in the country. Clearly, New York and the New York tri-state area are incredibly high hotspots. Baltimore, Maryland, where I am here, Baltimore, DC area have another outbreak that's ongoing now, and you can see all of the red hotspots around the country. And this is really the next phase of this virus, where it goes from the really large metro centers into these smaller communities and smaller cities around the country. And it's yet to be determined how long these will really last in these peaks. So what we're focusing on in the lab is really looking at therapeutics. And we've been doing this for several years now once MERS emerged. And what we find, what's clear is that there are no approved therapeutics for any of the coronaviruses, both SARS, MERS, or SARS-2. And so there's lots of things to try, including repurposing FDA-approved drugs, which we'll talk about, identifying a lot of monoclonal or polyclonal antibodies for getting right into the clinic, using convalescent sera from those recovered, which is being trialed now, lots of vaccines, including many that are being tested already in the clinic now in phase 1 trials, potentially non-antivirals, but immunomodulators that affect the disease severity of the virus as it comes. And then as well as looking at novel inhibitors. These general antivirals won't be things that are going to be tested against this virus in the clinic right now, but hopefully they'll be here for the next virus and the next virus that comes out, both coronavirus family members and other viruses as they emerge. So what we've been doing over the last several years is looking at repurposing FDA-approved drugs. And the reason for this is because there's something called the Valley of Death in the drug discovery field, where while you can do a lot of drug discovery on biochemical processes and looking at in preclinical studies in cells and in mice to look for inhibition of a virus, things that get through clinical trials are much smaller, upwards of basically less than 1%, 0.1% of drugs that even get through preclinical studies get through to the FDA. This process also takes very many years, upwards of 10 years to get through to the clinic. So the better way to do this is to really focus on the already FDA-approved drugs and then repurpose them against your virus of interest. And so in 2014, we published a paper with groups at the IRF, at NIH, at Fort Detrick, USAMRIID, and a company called Zalicus. And we screened FDA-approved drugs that were effective against SARS and MERS. And from that study, we identified about 28 drugs that worked against both viruses. And so this is the list. You can read the paper if you want to see the exact drugs. But what we did for this, our current study now, is we've now screened all of the FDA-approved drugs from that list, as well as more. But we started with our priority 20 drugs that were ready to go and really ready to work into the clinic. This was a straight viro assay where we infected cells or pretreated cells with each of the drugs at multiple concentrations and a concentration curve, different MOIs of the virus, and then looked for a cell proliferation assay to identify IC50s and CC50s and selective index. All this work in these first compounds was done in 19 days, a really remarkable push for the lab to get this all working out. This is up on bioRxiv now, and it'll be part of a larger paper coming up very soon. But just to highlight some of the data that came from this paper, what we find is that chloroquine and hydroxychloroquine are quite effective at inhibiting SARS-CoV-2 in vitro, at least, as well as other drugs that we've been focusing on in the lab in the past. Imatinib, which targets the ABL kinase inhibitors, tamoxifen, which affects probably receptor dynamics and receptor trafficking of the virus and ACE2 receptor, as well as amidaquine, which probably affects endosomal processing as well. And so this is just some of the studies that are in this first story. And we've had now dozens more drugs to the list that are effective, which is good. So we have a lot of FDA-approved drugs that really can be forwarded to both animal studies and clinical trials. When we look past just the cell proliferation assays and really look for inhibition of virus to get IC50s and in a TCI50 assays, looking really at just virus infection dynamics, what we find is that hydroxychloroquine is quite effective. I know it's the favorite of the administration here in the United States and potentially around the world. It does look quite good in vitro. I'm not going to show you. We haven't tested it against SARS coronavirus 2 in animal studies. We have done it for SARS-1. And it doesn't drop into the virus titer in the mouse studies, but it does affect the clinical symptoms in the mice. And that's going to be on the new paper. In the past, we've also looked at the mechanism of action of a lot of these drugs. And we're doing this now for SARS-CoV-2. This is the data for SARS-1, where we use a lentivirus pseudotype assay where we can look for how these drugs affect entry of the virus into cells. And what we find is that a lot of the drugs, including chlorpromazine and chloroquine and hydroxychloroquine, really block entry of the virus. So this assay reports the amount of virus that gets through the endosome into the cytoplasm. So it blocks this process. So does imatinib, which is an Abl kinase inhibitor. But gemcitabine, another drug that's known to be a transcriptional inhibitor, doesn't block this process. And so we can now use mechanism of action studies and time of addition studies to identify where in the life cycle of the virus all of these drugs work, which is what we're doing now. So we're in the process of doing many more drugs so far. We've already really screened about 500 different drugs at multiple concentrations to identify IC50s, and we're now working up to screen all FDA-approved drugs in singles and in combinations to really look for efficacy in vitro so we can forward them to animal studies. We've worked with a lot of companies lately looking at both drugs that are in trial and other academic labs looking at things that they just want to test to see against the virus. So that's ongoing. And what we really want to do, and we've done in the lab in the past, is looked at novel host-directed antivirals. So most of these drugs from the FDA are not really targeting the virus other than the protease inhibitors. They're really probably targeting host factors. And so we're identifying those host factors now, and we have other compounds in the lab that we know target host factors that affect multiple viruses. And we have a handful of them that target SARS, MERS, SARS-1, SARS-2, MERS, flu, Ebola, and Marburg in studies now. And so we're looking forward to getting that out to the community very soon. And then the next step is to really get these drugs into animal studies so we can look for efficacy and protection of pathogenesis. So I don't have time to go into the animal work, but we are working currently on multiple animal models. Wild-type mice are not infectable by the wild-type SARS-CoV-2, but there's a lot of other systems that we can do in mice, both an adenovirus model as well as overexpression models, which we are working on in the lab. And then with collaborators looking at ferrets and hamsters and non-human primates for a lot of the drugs that are coming through our screens to get them tested and to see how they work in vivo. We're also now forwarding to additional platforms in the lab, looking at lung-on-a-chip systems from Emulate, doing single-cell sequencing with 10x Genomics, and then really fast-forwarding the process to look at a high-throughput screening core where we can put inside the BSL-3 and screen not just drugs for SARS-CoV-2, but for a lot of other BSL-3 viruses. And we hope to really fast-forward this so we're not stuck where we are now in the future, where we can really study these viruses, have drugs ready to go for the next outbreak. So hopefully I gave you a little flavor of what we're doing in the lab, where we're repurposing drugs and doing all of the in vivo studies in the BSL-3 for both in vitro and in vivo models in mice. And then really studying these at the mechanism of action level, looking at transcriptome and proteome and metabolome of how these work, both in cell culture as well as the lung-on-a-chip system. So we can really study them at a more physiological level in our controlled conditions. The last thing is that we are hiring. We have interested in postdocs and especially people with BSL-3 experience. Please contact me if you are interested. I'd love to talk to you and see if you can fit into our group. We're going to be doing this for a long time. I'm a coronavirologist by trade, and so we're not going to be leaving this field anytime in the near future. So these are all the people that did all the work, led by Stuart Weston, a postdoc in the lab, Rob Haupt and Jimmy Lowe, grad students, and then Crystal Matthews and Holly Hammond are technicians that are running all of the BSL-3 and BL2 lab, keeping us up and running. So I will leave it there and then take any questions. Thank you.
Great. Thank you very much, Matt. Let's start with this one. A lot of people are asking about moving from in vitro to in vivo. Like how easy is it? And is it like a direct one-to-one translation? Can you talk about that a little bit?
Sure. So it's not a one-to-one correlation with the IC50s that we see in vitro still need to be obviously tested in vivo. All of the drugs have different dosing requirements, whether it's oral bioavailability, they're IP dosed. And so it's going to be an experiment to see which of these drugs really works in vivo. And so we're working on that now. There's a lot of data from FDA studies and other trials where we know how to dose them, and that's what we're working on now. And really getting them at physiologically relevant concentrations. So in the lung-on-a-chip system, we can get to Cmax, which is really the amount of drug that you'd see in a human in the serum. And so we can keep those concentrations constant and really kind of level off the playing field so we know which are going to really work in people. And so we're doing as much as we can in vitro before we get to the trial stage.
Great. Quick question, one of the top voted actually. Why is CoV-2 BSL-3 instead of BSL-4? And maybe you might just talk about the general process for how they decide these things.
Sure. I thought you were going to go the other way and making it BL2.
Yeah.
So a BSL-4 pathogen is like Ebola and anthrax, where it can be heavily weaponized. It's highly transmissible, and has a lot of other kind of worrisome kind of defense-oriented angles to it. This virus is similar to SARS. It's a BSL-2 virus. It's not a select agent, so there's another level above where SARS is. So SARS, I didn't put a picture of the PAPR, but we wear a full Tyvek suit, which is basically a FedEx envelope with arms and legs, a HEPA-filtered air PAPR that pushes air over our face, but it's not a sealed suit, which is the big difference between BL3 and BL4. And then BL2 are all the other kind of seasonal flu viruses and seasonal coronaviruses that we can work on just in a biosafety cabinet. So this is considered a BSL-3 virus. If it was, there's other characteristics that make it a BL4, which is a lot of other kind of defense-oriented things. But since it's widespread in the community, it doesn't make sense to kind of put it in that kind of BSL-4 lab system.
Mm-hmm. Great. How about cocktails versus single drug use? Any thoughts there?
Yeah, so I think that what we're going toward in the future is doing combination therapy with in vitro. So to test to see which things synergize with each other. The goal would be to identify drugs that work on entry or exit of the virus or transcription or translation, and then really combine those together that work on different aspects of the virus. And it would make sense to now target multiple parts of the pathway. And that's what the mechanism of action studies will tell us, is where in the life cycle of the virus they really work, and then combining those together. And so that's definitely something we're doing in the near future.
Great, and we'll do the last one from actually one of our speakers, Dr. Robert Meller. Is there evidence of COVID infection rates in patients on antipsychotic drugs? You mentioned chlorpromazine.
Chlorpromazine, yeah.
Yeah.
So this is something we're looking at now. We're trying to plug into the clinical networks where there is a lot of data streams of people who are on a lot of the FDA-approved drugs, whether it's ACE inhibitors, whether it's antipsychotics, whether it's protease inhibitors for HIV, and then seeing is there a less attack rate in those people who are already on these drugs, which is, it's a great way of looking at it and really fast tracking which ones are going to work and which ones aren't. And the problem actually is those clinical connections are all behind firewalls and HIPAA protected. And so there's not a lot of data streams connecting them together, but we're working on with government partners to try to untie that red tape and really get the data. It's a good idea.
Great. Thank you very much. And with that, let's transition over to our next speaker. Thank you very much, Dr. Freeman. Our next speaker, and you can go ahead and start setting your slides up, Dr. Agard. Our next speaker, Dr. Shirsty Agard, is an expert in maternal fetal medicine and is the Henry and Emma Meyer Professor Chair in Obstetrics and Gynecology at Baylor College of Medicine and Texas Children's Hospital. She serves as Vice Chair of Research for Obstetrics and Gynecology and is a professor in the departments of Molecular and Human Genetics, Molecular and Cellular Biology, and Molecular Physiology and Biophysics. She's a member of the Center for Reproductive Medicine, Digestive Disease Center, Eisenberg Center for Decision Sciences, the Center for Microbiome and Metagenomics Research, and the School for Tropical Medicine. She's also a co-director in the Baylor College of Medicine MSTP MD PhD program. Her clinical interests include emerging infectious diseases, preterm birth, diabetes and hypertensive disorders, maternal smoking and environmental exposures, and the detection and diagnosis of congenital and genetic anomalies. Additional translational research focuses include the role of the microbiome in pregnancy and early development and the impact of key exposures in pregnancy, such as diabetes, maternal high-fat diet, smoking, and environmental chemical exposures. Thank you very much for being here. Please welcome Dr. Shirsty Agard.
So thank you. I think fortuitously, we've all managed to pick up slightly different kind of elements in our introduction. So I'll follow that vein as we kind of move through. There we go. So in terms of the range of illness, I'm going to use a couple of just general talks or general points that'll relate back to some more subtle points as we move along. So in general, most of us are explaining it to our patients that the range of illness follows what we call the 80-15-5 rule, which means about 80% or so will have some degree of symptom or pre-symptomatic. Another 15%-ish will be asymptomatic, and 5% will be critically ill with case fatality rates following. And so that's kind of a pretty good rule of thumb that we're doing it. Speaking of asymptomatic people, I just want to briefly mention what we think of as the great testing debate, and that is really around different elements of testing. And so Chris brought up very nicely his thoughts around the LAMP assay. Most of us are still heavily banking on very good, solid clinical assays to make diagnoses. And part of the reason for that, it relates back to this issue of PPE use. And that is that we really need to know who is positive and who is negative, because it changes the amount of kind of PPE we use when we go in and out of these rooms. This is particularly important in situations like obstetrics, where we have otherwise very healthy people coming into the hospital all the time, and we can't treat them like they're all positive, and we can't miss cases that might be positive and end up with false positives in others, because it causes a tremendous amount of chaos around our management. It is both expensive and exhausting to try to work through who's positive and negative without knowing. So at some point, we really have to hit this compromise of knowing what it is. Chris alluded to this earlier, but over in the far right bottom column or figures, I've showed you where some of these different CT values are that we use as cutoffs. Most of us are saying, you know, when people are positive, they're clearly positive with CT values in that 20 to 30 range. And when we get back out beyond that kind of CT value of 30 on our RT-PCR assays, the most of the time we're kind of backing off of that, and we'll rerun it several times to determine true positive versus not. But that's really a lot of this issue around our reliability. And I've also showed you the different kind of gene areas that we target towards testing in most of our LDT or lab developed testing assays. I also just want to mention this timing of clinical symptoms, because I think it's important to understand a little bit more of some of the news that's coming out. Some people may have picked up that as of about an hour or two ago, China has suddenly identified another 1,200 some deaths that occurred that they believe are related to SARS-CoV-2. I think we're going to continue to see this theme emerge over time, over figuring out exactly what goes on. But in general, it really does look like it starts off as a mild illness ranging from 7 to 10 days. That's actually the time in which the viral production ramps up. There was a very nice study that was published in Lancet ID just over a week ago now that looked at that kind of viral dynamic time frame. And your highest rate of viral shedding is in that early period of mild symptoms. As your symptoms get worse, your viral shedding goes down. And in fact, interestingly, in that paper, their IgM and IgG came up concomitantly. And one of the reasons we're seeing a little bit of a difference around this is because about halfway through or so with the outbreak in China, that is when they kind of lost the ability to do their testing and started making clinical diagnoses. And so that timing of disease onset is a little bit skewed because of the lack of testing abilities in China. And so that skews it. This is an example of the first case in the US, and was very easy to track exactly how that occurred. And here you see a much more classical picture of that 7 to 10 days of asymptomatic or prodromal clinical illness. Mild symptoms move into heavier symptoms. And of course, we have the asymptomatic cohort I talked about earlier. There are some worthwhile statements to make about what really predicts death. And so it looks like there's a certain number that can predict death around this cytokine storm picture, in which your LDH and your D-dimer is going to go up as you start to have multi-system organ dysfunction. I'm going to go into this a little bit later on, but there are certainly a number of patients that we simply cannot continue to ventilate them. So they die of true respiratory death, very similar to what we experienced with H1N1. Not that many of us had experience, especially not in the US, with SARS-CoV-1 nor MERS, so we can't comment on that. But we had rather considerable experience, especially those of us that take care of pregnant women and children, with H1N1, and we see a very, very typical profile. There's also a group that appears to have largely cardiopulmonary collapse and death. And I'm going to illustrate that in a little bit when we talk about what's happening in some of our pregnant patients that we're just seeing emerge out in the literature now. But I just want to mention that some of these predictors of death are following very different pathophysiology, and that's kind of important to think about. Here I've showed it a little bit differently. This is the Lancet paper that characterizes well. We do know that when we have to trach people and put them on ECMO, it's highly unlikely they come off. And so those are very strong predictors of death. Some of that, of course, is severity predictions, and some of that may actually be questions about whether or not they get secondary infections. I did want to highlight Chris's comments earlier on about enterovirus that I'm personally intrigued by. Enterovirus, as some people know, causes a horrible myocarditis, especially in children and women, pregnant women. And so I'm a little bit intrigued about whether or not there may be a signal there worth pursuing, especially in our myocarditis deaths, that may be helpful for us to know that those may be some secondary viral infections that we're picking up. And it's not uncommon that those enteroviruses will be within hospital settings. If we had to pull together quite a bit of this different source data in terms of what's happening, you'll see that you have this viral response phase and the host inflammatory response phase, and those actually coordinate in an inverse response. And I think, you know, that's worthwhile just all of us keeping in mind that that's true with almost all of our RNA viruses, especially our plus-stranded viruses that go through an obligate double-stranded phase. I'm going to talk a little bit more about that when we get down into the microRNAs in a minute, but each of these potential therapies really come against each of these different points. Now, I'm just going to insert my opinion around what I think have been some clinical game changers and how they relate to potential opportunities for this group. I'm going to highlight a paper that came out yesterday and highlight a paper that came out just under a week ago, both in the New England Journal. I want to highlight the Icelandic population paper because I think it told us two things, and it'll lead us into our discussion a little bit more in pregnancy. What they found is they had kind of two cohorts. They had a targeted cohort that they were monitoring, and they had a surveillance cohort that they were monitoring. What they found is in their surveillance cohort, and this was largely out of Reykjavik, in their surveillance cohort, they had very low rates in women and children less than 10. In fact, in their second surveillance cohort, they had no children that picked up positive, and that was all under social distancing. Some people are interpreting this as biology, that women are more resistant to the infection and children are more resistant to infection. I'm going to urge great caution and remind us that we need to stay highly, highly humble right now, that we don't know a lot of this. To me, it's highly improbable that having 2 copies of the X chromosome is protective. I think this is much more likely to be social distancing and also poor reporting, and we'll give an example of that in a minute. But in general, in most countries around the world, this is the classic example of social distancing and cohorting of patients, which we work after all the time with public health. So if you have small children, it's more likely to have a woman at home with those small children. And so there is going to be less disease in that cohort. And you see that very, very well within this Icelandic population. So I think that's a critical paper for that. It also tells us something else important. We cannot, for economic reasons, stay sequestered forever. So we're going to have ongoing identification and isolations of all cases and their contacts. That's what the big take-home message is of the Icelandic paper. I want to just briefly also mention the remdesivir trial. It does show efficacy, actually pretty impressive efficacy. It's unclear if there's going to be synergism with hydroxychloroquine. It's unclear if there's going to be antagonistic with hydroxychloroquine. I'll just remind people that we ran a large study back in 2012 that showed if you used hydroxychloroquine alone with HIV positive patients, they actually did much worse. And so it may be important to combine hydroxychloroquine with an efficacious antiviral medication. And we can talk about that a little bit more in the panel. I'm going to move into kind of my area of clinical expertise and interest, and that is around pregnancy. So I just want to mention kind of three things. Historically, if we go over the last 100 years, pregnant women have always done worse with lower respiratory infections, bar none, no exception. In H1N1, I will just say in an editorial fashion, we have had a horrific pandemic in the last 10 years. It was H1N1. In H1N1, about 24% of the world's population became infected. Interestingly, despite the fact that pregnancy only represents 1% of the US population at large, in H1N1, 6.4% of all hospitalizations were pregnant women and 4.3% of deaths were, which is obviously a four-log-fold difference between that. So in SARS-CoV-2, 10.5% was the general population mortality rate. With pregnant women, it was 25%, 33% requiring mechanical ventilation. And SARS-CoV-2 is a little bit of a mystery, and I'm going to share with you data that I'm going to ask not be put forward in the next 3 slides because it's under press embargo right now. But despite over 200,000 deaths, we don't have people reporting deaths in pregnant women. We think that this is a reporting issue. This is not actually a true biology issue. But again, we stay humble and say we simply don't know at this point. So this is privileged data. I'm going to ask that this does not go up onto social media. But this is a cohort that we collected from Iran. These are heavily adjudicated cases of 9 women with severe disease over a 30-day span of time. So we did this in a nested cohort analysis. And within this set of cases, 7 of those women are now dead. 1 remains intubated, and while we hold out great hope, I think it's unlikely she will survive. And the final case is extubated after a prolonged intubation period. There's no rhyme nor reason we can figure out of who lived or died within this cohort. There was everything given from convalescent plasma sera to very good medications, including remdesivir. In 2 cases, the women were a little bit older than the average population and certainly older than a couple of the Chinese cohort analysis. And there was nothing to do with their blood type that could predict their death. And so this, we hope, will be in press soon and really highlights that pregnant women are dying of SARS-CoV-2 around the world. We're just all catching up. And I certainly know from all of my colleagues up in the Northeast that they're seeing pretty severe morbidity in pregnant women, and deaths are emerging there as well. There's also kind of another interesting observation within this cohort that we have, and that is why we have no evidence of vertical transmission. Again, we stay humble and unknown whether that's going to be true. We do have a high rate of perinatal morbidity and mortality. And so these babies are either dying in utero or they die ex utero with neonatal deaths as well. We had to reference this to something, and because we couldn't do large surveillance data, we referenced this to familial and household cohorts. And in each and every case of our 9 subject series, the pregnant women died and their husbands and children did not have severe disease and did not die. But this generates a couple of very interesting hypotheses. I will just comment that there's as much cardiopulmonary as respiratory death. We really think this is largely an underreporting issue. I will just state, from an epidemiologic standpoint, I'll remind people that in any case series of 9 that report 0 mortality, your upper confidence limit will be 33%. So you're far more likely to have guessed wrong than you are to have guessed right. There is considerable perinatal morbidity and mortality, and we're very intrigued by the possibility that this may be mediated by some antiphospholipid mechanisms, which are well documented in both hepatitis and herpes viruses, as well as in syphilis, and we can talk about more about that in the panel. With that in mind, I'm going to just quickly move to 2 vignettes that we worked through with our last pandemic, which was Zika. And we developed a marmoset consortium, and there's a variety of reasons we did this that have to do with how marmosets actually reproduce and the amount of time that they spend in their placentation phase. But suffice it to say, one of the things that we learned from this disease and were able to show very successfully is that some viruses do things we don't expect them to do, like replicate in the placenta. And the placenta, actually, we were able to eventually show serves as a reservoir for Zika virus replication. I will parenthetically mention that both ACE2 as well as at least one of the proteases involved in SARS-CoV-2 replication is well known to be on a placenta, and there's no reason to think that it can't be transported across the placenta. Whether or not it infects a neonate is a whole separate question. And we saw in Zika a very high rate of fetal death that actually informed our clinical management. And we now know that the most common outcome of congenital Zika infection is stillbirth and early pregnancy loss. It turns out that despite what was said initially about Zika, it is not trophic for neuroblasts. It actually will infect any number of cells during embryonic life. It's just that the phenotypic consequence of this ends up being brain destruction and subsequent microcephaly. I want to just bring this forward to the group. We did some RNA-seq analysis within both our marmoset cohorts and within some of our human cohorts. And one of the things we found that was rather interesting is inflammation varied very heavily based upon the tissue you looked in. So we could see, if we looked in the maternal circulation, really nice evidence of inflammation on a whole transcriptome analysis level. But when we looked specifically within the placenta, there was no evidence of inflammation within the placenta. We think that'll be highly interesting in SARS-CoV-2 if we can get some animal models in which we can look at different body sites and what's going on there. And we may see something both in the respiratory and the cardiopulmonary tract. And this is just a better example of that in which we bring it out. The final thing I want to just close with is also some work that'll be kind of vignette number 2 that we developed with Zika. And I want to remind people that in the process of replicating, these plus-stranded RNA viruses go through a double-stranded RNA replicon phase. And our bodies are uniquely poised as hosts to get rid of double-stranded RNA. That's the whole reason we have microRNAs. Their job is to get rid of these double-stranded RNAs and silence them out. So we did some work within Zika, and one of the things we showed was that when you underwent infection with Zika, you actually switched the arms in which you loaded on your microRNAs, and you would flip between a 5' strand loading and a 3' strand loading, especially in these regions that were responsible, we thought, for some of the Zika virus silencing. So I think that's something.
Dr. Agard, I just want to interject real quick and say we're just about to wrap up on time. So
Perfect.
if you can finish up fairly soon, and then I'll ask a couple questions. We can just spill over into the break a little bit.
Perfect. And so if we look at this different strand loading, that may actually have something to do with the transcriptional analysis that you see up. So this is Argo clip data that we developed with Zika, and we saw really interestingly some epithelial mesenchymal transitions as a result of those flips in the 5p, 3p loading of those microRNA molecules. And so back to SARS-CoV-2, I just want to remind people that despite kind of the sparse science, as was discussed in the last talk, the clinical trials are marching on. The time for science is now if we want to better inform all of us functioning clinically, but we're really kind of moving forward in quite a few different clinical trials. I want to thank the folks in the lab. In this work, Michael Yoakum brought us to the table through Todd, and Max Safarovic is doing a lot of our analysis. And with that, I am happy to take questions.
Thank you very much. So we're on break, everyone, now for 5 minutes. The next talk is going to be at 11:15 Central. And in the meantime, I'll just go ahead and ask a couple questions for anyone who wants to stick around for a couple minutes. Okay, the first question, do you see bacterial infections such as Staph aureus, let's say, heavily involved in sepsis in COVID-19 patients?
Yeah, so that's a good question. Specifically within pregnancy, we haven't. That said, you know, that's going to be a very, very tough signal to tease apart because we're typically putting folks on 2 and 3 broad spectrum antibiotics that are pretty heavy hitters. I don't know that we really know the answer to that. You do see a lot in the social media saying people aren't dying of COVID-2, they're dying of secondary bacterial infections. I'm not aware of the evidence for that. I think that's not a good line of evidence.
Yeah, sepsis and COVID-19 was brought up a few times. If you want to just say anything else there. Otherwise, I'll go to the next one.
Yeah, so there's of course sepsis, and then there's the inflammatory response to sepsis, which is part of kind of the cytokine storm discussion that's going on. And so most of us are managing these as septic patients with a septic response. In pregnancy, we don't use steroids for the reason of squelching or squelching that response. We use them to promote fetal lung maturity in women that are still pregnant. And in those situations, we're not really seeing a difference in maternal outcomes based on whether they got short burst corticosteroids or not.
Great. How about neurological effects, short and long term? Any comments on that?
We simply don't know yet. There is reports around neurologic effects. There are multiple kind of reports around some seizure activity. Some of that is accompanying multi-organ system dysfunction, but where that's going to settle out and whether there are some long-term neurologic sequelae, time will tell.
Are patients under immunosuppressors or anti-inflammatory drugs protected for less attack rate, like Crohn's, for instance?
We don't know. Those are the patients we're having stay heavily sequestered, and so we can't comment on where that data is going to fall out. I do know in a couple of the different case series in non-pregnant populations, they are actually more susceptible to death. But whether or not they're more susceptible to infection, we don't have the surveillance data to know that yet.
All right, final question.
Yeah.
Oh, sorry. Final question. There's been some data showing how lymphocyte counts, particularly T cells, is a poor prognosis factor during acute infection. Are there more people doing immune profiling of patients, both acute and convalescent?
Yeah, so definitely. So lymphopenia, we're seeing pretty routinely in pregnant women that come in with severe morbidity, and in our cohort of death, lymphopenia was very common. The leukopenia is a plus or minus, and the thrombocytopenia is a plus or minus. Like I said, we're very intrigued with the antiphospholipid pathway data that we're starting to observe. And so I think that's going to be an interesting route to go down, and that may explain quite a bit of the vasculitis that we see, as well as a microangiopathic disease.
Okay, thank you so much for that. Thank you very much, Dr. Agard. And with that, we'll leave off for about another minute or so we'll have our next speaker. And Dr. Wallace, if you want to go ahead and just get your slides shared on the screen, you're welcome to go ahead and set that up.
And that's what I'm working on. Share screen. Great. And there's slides. And is that working for everybody?
Mm-hmm. Mm-hmm.
Cool. Okay, great.
Okay, great. Yeah,
Thank you.
So we'll just give it a couple seconds and we'll start up.
No, that's great. Can you hear me okay?
Yeah. Yep.
Leo? Oh,
yeah. Hey.
Okay.
Yep.
So
okay,
now you can see me? Okay.
Yeah, perfect. Thank you. All right, I think I'll go ahead and start with introduction for you here.
Yeah, go for
it. So our next speaker is Dr. Doug Wallace. Dr. Doug Wallace is the Michael and Charles Barrett Endowed Chair in Pediatric Mitochondrial Medicine and Metabolic Disease, and the Director of the Center for Mitochondrial and Epigenomic Medicine at the Children's Hospital of Philadelphia, which is also referred to as CHOP. He's also Professor of Pediatrics in the Division of Human Genetics at the Perelman School of Medicine in the University of Pennsylvania. Dr. Wallace has studied the role of mitochondrial genetics in human health and disease for nearly 50 years. In the 1970s, he defined the rules of mammalian mitochondrial genetics, including demonstrating the maternal inheritance of the human mitochondrial DNA. He then used the mitochondrial DNA variation of indigenous peoples to reconstruct the origins and ancient migrations of women. He was the first to identify maternally inherited mitochondrial DNA diseases, thus founding the field of mitochondrial molecular medicine, and showed that naturally occurring mitochondrial DNA lineages contribute significantly to the risk of common metabolic and degenerative diseases, as well as AIDS progression. Dr. Wallace is a member of the U.S. National Academies of Science and Medicine. And with that, I think we can go ahead and get started. Thank you.
Thank you very much, Leo. And thank you, Todd and Afshin, for including me in the talk. I'm going to make a case that the mitochondria is an important aspect of the pathophysiology of COVID-19, and that maybe it gives us real alternative approaches for therapy. So looking at how SARS-CoV-2 virulence might relate to the mitochondria, first, I'm going to show you that viral proteins are extensively interact with mitochondrial proteins. Second, that COVID-19 individuals and groups exhibit differential sensitivity, and that's characteristic of mitochondrial DNA lineages. That males are more sensitive to females in COVID-19, and that is also very characteristic of mitochondrial diseases. That the elderly are more affected than the young, another factor commonly seen in mitochondrial diseases. We know now that the mitochondria regulate the innate immune system, both the inflammasome and interferon pathways, and that COVID-2 activates the inflammasome to drive viral dispersion, where it inactivates interferon arm to block viral RNA destruction. Excuse me. That the mild cases of SARS-CoV-2 result in short-term viremia, but in the more severe cases, we get long-time viremia with a severe cytokine inflammation. So what are some of the objectives we're trying to do at CHOP to look at this? First of all, we're trying to determine if mitochondrial DNA variation impinges on viral sensitivity. We are anxious to understand the action of the SARS-CoV-2 non-structural proteins and open reading frames. We'd like to understand the importance of mitochondrial variation in the severity of the disease. We'd like to develop a mouse model to study the functions of variation of the non-structural proteins and the ORFs after adeno-associated viral transduction. We'd like to look at using our mouse models to look at preclinical drug testing. We'd like to work with, say, people at University of Maryland to look at a model of SARS in the mouse, where we could look at our mitochondrial variants as they affect infection, and then use those for drug trials. So as you've heard, both SARS-CoV-1 and SARS-CoV-2, as well as MERS-CoV, originated in bats. The intermediate is civet cats for COVID-1, camels for MERS-CoV-2, still unknown for humans. And as you've heard, it's a 30 kilobase single-stranded virus that encodes the envelope and spike proteins, as well as a whole series of protease degradation products called non-structural proteins and open reading frames. Now, the mitochondria is unique because our human cell was formed by a symbiosis of two different bacteria about 2.5 billion years ago. One of the host bacteria gave rise to the nuclear cytosol. The other gave rise to the mitochondria. They have parallel information storage and retrieval systems. The nucleus now codes for almost all the structural proteins of the mitochondria, but the mitochondrial DNA still encodes for 13 polypeptides plus transfer RNAs and the ribosomal RNAs. And these polypeptides are critical in mitochondrial energy metabolism, making the mitochondrial DNA basically the wiring diagram of the power plant. The mitochondrial translational system is bacteria-like. It's initiated by N-formal methionine. It's sensitive to chloramphenicol and aminoglycosides, and it codes for key proteins in the energy-generating system oxidative phosphorylation, including 7 of the 45 proteins of complex 1, 1 of the 11 proteins of complex 3, 3 of the 13 proteins of complex 4, and 2 of the 17 proteins of complex 5. So basically, then we have to understand the physiology of the mitochondria. It has an outer membrane, an intermembrane space, and an inner membrane and a matrix. Reducing equivalents from fatty acid metabolism and glycolysis enter the mitochondria, in this case, from glycolysis through pyruvate to drive the tricarboxylic acid cycle, the purpose of which is to reduce the carrier NAD to reduced NADH. And these hydrogens are then burned by the electron transport chain, complex 1, CoQ, complex 3, cytochrome C, complex 4, to reduce to oxygen. Pumps protons out into the intermembrane space. They're used by the ATP synthase to bind ADP and phosphate, condense ATP, and to transport it out to do work by the translocators. The mitochondria also has a self-destruct system, the permeability transition pore, which when the membrane potential gets low, or mitochondrial generation of reactive oxygen species or the uptake of calcium become extreme, and they cause this pore to go from closed to open, release pro-apoptotic proteins, and degrade the cell from inside out. And so the mitochondria generates the energy, regulates redox, regulates reactive oxygen species, calcium, apoptosis, signal transduction intermediates, and the epigenome. Now, the organs that are most affected by the mitochondrial dysfunction are the high energy organs, the brain, the heart, the muscle, renal, and the endocrine and liver systems. The mitochondrial DNA is maternally inherited, transmitted from mother to all of her children, but the daughters transmit to their children. Male's mitochondrial DNA is selectively destroyed. Each cell has thousands of mitochondria. They replicate inside the cells. They accumulate mutations. We call that heteroplasmy. And when the cell divides, the distribution of the mitochondrial DNA goes along with the cytokinesis. So a heteroplasmic egg can give rise to tissues with different mitochondrial genotypes. And as the percentage of mutant mitochondrial DNA increases, the energy output declines until it crosses the minimum for that organ, and then you get a clinical phenotype. So the mitochondrial DNA then codes for small and large ribosomal RNA, a complete set of transfer RNAs that punctuate the genes, 7 proteins, ND1, 2, 3, 4L, 4, 5, and 6 for complex 1, cytochrome B for complex 3, CO1, 2, and 3 for complex 4, ATPase 6 and 8 for complex 5. And there are 3 classes of important mitochondrial DNA mutations, those that are maternally inherited and transmitted through the maternal lineage and give rise to disease. As an example, a mutation in the tRNA leucine gene at nucleotide position 3243. When it's at 30% heteroplasmy, it'll give you diabetes and autism. When it's at 50%, it gives you neuromuscular disease. When it's at 80 to 90% mutant, gives you a lethal childhood disease called Lee syndrome. We have then many other pathogenic mutations in the mitochondrial DNA that all affect this system. Then there are also ancient polymorphisms, and they define lineages because the mitochondrial DNA is exclusively maternally inherited. It can only accumulate mutations on radiating lineages. Therefore, by sequencing the mitochondrial DNA of any 2 people, it's directly proportional to the time since they shared a common ancestor. And that then allowed us to reconstruct the ancient origins of women. And for instance, this variant is found in 3/4 of mitochondrial DNA from sub-Saharan Africa. This is found in half of Europeans. There are also about 2,000 nuclear encoded mitochondrial genes, and those mutations are also relevant to disease. But if we put mitochondrial energetics in the middle of medicine rather than anatomy, we can see that we could have genetic diseases by mutations in nuclear genes or epigenomic changes. We could have changes that affect the mitochondria, the recent adaptive mutations, ancient adaptive mutations, and recent deleterious mutations. The diet we use, the amount of activity we have, or whether we are exposed to toxins, all affect mitochondrial function. If you limit mitochondrial function, you'll increase mitochondrial DNA damage and over time erode energetics, and that we believe is aging and why some diseases have a delayed onset and progressive course. The organs that give the most common disease are neuropsychiatric, cardiac, muscle, renal. And if you inhibit the furnace, then you'll build up the substrates, and that's then metabolic syndrome, diabetes, and obesity. And then finally, if the mitochondrial energetics is sufficiently impaired that the cell does not undergo apoptosis but necrosis, it releases then thousands of bacteria, because the mitochondria are bacteria, into the bloodstream, and it activates the entire inflammation system. Finally, cancer is all about regulating growth of the cell. So getting then to the SARS, the COVID 1 and 2, and MERVs are all very similar. They have 2/3 of the genome is this polyprotein that is then processed by proteolysis. And then they have the spike protein and the other structural proteins, but then also these important open reading frames. So we're very interested in how the proteolysis degradation products and the open reading frames might interact with the host. This is a nice diagram that one of our consortium members, Paco, presented the other day. But what it shows is that the polyprotein then is processed into a whole series of non-structural polypeptides, which become very interesting in the host interaction. So the work that we are doing started out with my receiving from a friend at University of California, San Francisco paper. First author is David Gordon, where they actually looked at SARS-CoV-2 proteins, and they then looked at their interaction with host proteins. And this is a diagram of their paper showing the different COVID-2 non-structural proteins and ORFs, and then the different genes that they interact with. So the first thing that I did is I went to each of these 332 genes. I did a literature search and determined the function of each one. And what that resulted in realization is that, about 55 of all the 332 proteins, about 17%, are genes that directly relate to the mitochondria. An additional 105 genes, or about 32%, are genes that modulate cellular function based on bioenergetics. So overall, we can envision about 48% of all the SARS genes are somehow affecting energy metabolism. And that, of course, makes sense because the virus is going to need a lot of energy to replicate. So what then I've done is I've classified all the different proteins that interact with SARS proteins based on mitochondrial functional functions. So the first here is mitochondrial protein synthesis. NSP8 interacts with these 4 ribosomal proteins. And then this protein, which is the asparagine tRNA synthetase of the mitochondria. The M protein interacts with the leucyl, tRNA synthetase, and ORF9C interacts with phenylalanine tRNA synthetase. So you can see that the virus is taking a lot of time to block protein synthesis. If we look at mitochondrial metabolism, NSP7 interacts with the assembly factor of respiratory complex 1, ORF9 with 4 other factors of complex 1, M with the coenzyme Q synthesis, ORF9 with complex 2 assembly, and this is one we'll come back to. ATBS5 interacts with the complex 5G protein subunit. M interacts with the electron transfer flavoprotein and the medium chain acyl-CoA dehydrogenase, key proteins and fatty acid metabolism. NSP12 interacts with the kinase for branch chain ketoacid dehydrogenase for branch chain metabolism in the mitochondria, and et cetera. So I can't see.
Dr. Wallace, I think you might have just accidentally got it muted here. I can unmute you.
Now.
Okay, that should be good now.
Yeah, so then we also have a whole series of interactions that specifically block protein import from the cytoplasm to the mitochondria. NSP4 interacts with these TIM complexes, ORF10 with another member of the same TIM22 complex, ORF8 with TOM70, which is the key protein for the outer membrane import. And most interestingly, the N protein interacts with the 2 peptides that make up the N-terminal import processing peptidase for all the proteins that come into the mitochondria. And there are other proteins that are specifically of the mitochondrial intermembrane carrier functions. There's also mitochondrial biogenesis proteins. This one affects the permeability transition pore. This is the CREB coactivator for PGC1 alpha. This interacts with mitochondrial biogenesis. This interacts with fusion and fission. And this is one of the sirtuins in the mitochondria regulating protein desuccinylation. Or there's a whole series of proteins on innate immunity, and ORF3A and E directly relate with the mitochondrial associated inflammasome, as does ORF8B. 3A, R6, and N are all involved in interferon production, and NSP13 all involved in the interferon arm of the innate immunity. And then a whole bunch of other interesting interacting proteins, MYC, HIF, protein synthesis, cytosolic, PKA, et cetera, et cetera. The point of all that is there's been a lot of effort by the virus to interact with the energetics. So why would that be? Well, the mitochondria is now known to be the key regulator for the innate immune system. If you damage the cell or have environal infection, it causes calcium to enter the cell. The calcium activates the TCA cycle, generating NADH. The NADH overloads the electron transport chain, making ROS. And then the ROS oxidizes the mitochondrial DNA, which is released into the cytoplasm. The oxidized mitochondrial DNA binds to the inflammasome, activates caspase-1. That processes so the pro-caspase-1 beta, pro-IL-1 beta and IL-18. Those are secreted. They interact with the toll-like receptors. They drive NF-kappa B and drive then the synthesis of the inflammasome response. But also the oxidized mitochondrial DNA binds to C-gas that activates through cyclic GAMP sting, which upregulates the interferon pathway, generating interferon that goes to the JAK-STAT pathway to drive interferon production. So the mitochondria then is central to regulation of the entire innate immune system. This is something that I've borrowed from another organization, another paper.
And I just want to let you know we've got about 3 minutes left for questions and for the rest of the talk.
So here we can see how the virus moves through the interferon system. So basically, I wanted to point out this paper where these people did a single cell RNA-seq analysis, and they looked at the T and NK lymphocytes. And what you can see is all of these proteins are upregulated mitochondrial proteins, and one of them is this ATPase6 gene, which is regulated by NSP6, and that, in fact, is a gene that we found mutated in patients. If we look at haplogroups, we find that, in fact, mitochondrial DNA lineage we showed a number of years ago directly relates to long-term effects of AIDS outcome and regulates CD4 levels. And so now what we're doing is we're synthesizing the SARS-CoV and NSP and RFs, putting them in expression vector, putting them into cultured cells that have variable mitochondrial function to look at the effect, putting them into mice through an AAV transduction system, and then looking at single cell RNA expression and mitochondrial and immune function. And so this just shows an example of a family and how the different phenotypes, the normal, diabetes, lethal childhood disease, neurodegenerative disease, all have different transcriptional profiles that we could modulate and affect the phenotype of these individuals. So finally, what we're really thinking about is how we might do surgery. How we might do intervention. And one of the ideas we have been thinking about, and I know that AFSFIN is also, is there's a microRNA 181, and that microRNA specifically targets and downregulates important genes of bioenergetics. So the idea is to create a therapy which uses this target sequence and then has many copies of it in an expressed gene, which will then sop up these microRNAs and thus alleviate the repression of the mitochondria and hopefully treat the disease. So how are we going to do that? Well, we need a mouse model for this disease. As you heard, there is now a human ACE2 receptor model available to the Jackson Lab. How are we going to get the mitochondrial DNA mutants into that model? We'll do nuclear transplantation of oocytes so that we can just transfer the ACE2 nucleus into the background mitochondria, and then we can use that for pharmacological and gene therapy experiments. So I'd just like to thank all the people that did this work, and thank you for your time.
Thank you very much. All right, the first thing I'll start off with, there were two questions, potentially treatment related, metformin and caspase inhibitors. Can you comment on either of those as potential therapeutic?
Yes, so metformin is a commonly used anti-diabetic drug. It definitely downregulates respiratory complex 1, but only to a small extent. People have hypothesized that that results in reduction of the high energy phosphates, creating AMP. AMP then interacts with AMP kinase. AMP kinase phosphorylates the master regulator of mitochondria, PGC1 alpha, and that upregulates mitochondrial function. And so the idea is that the way that works for diabetes is by by upregulating mitochondrial function and therefore decreasing the levels of the metabolites in the blood. that system is being looked at for cancer because some cancers are glycolytic, and that would drive it back to oxidative. And all the evidence for SARS-CoV-2 is that it's downregulating mitochondrial function. So any drug that might upregulate mitochondrial function has real promise, and that would certainly be metformin, which is already approved.
Thank you very much. And then the last question I'll ask, is there an increased correlation in mother/progeny outcomes versus father/progeny outcomes?
In mitochondrial disease or in COVID, SARS-CoV-2?
Yeah, I think for the COVID-19, but you can comment generally as well.
I don't know that I've seen any data that would look at that, but that would certainly be interesting to do. What we're doing right now is we're taking the RNA-seq data, we're aligning it as are other people, looking for the mitochondrial polymorphisms, and then trying to determine the haplogroups and see if haplogroups correlate with the predisposition to the disease or protection.
Okay, thank you very much.
Thank you.
Our next speaker is Dr. Afshin Beheshti. And Dr. Afshin, actually, first I'm just going to announce if you don't mind, the schedule. The next speaker is Dr. Afshin Beheshti. Then at 11:55 Central, we have Dr. Kathleen Fish. And then we have another 5-minute break following that, followed by 3 more speakers. And we'll send out more information in just a sec on that for those who are wondering about the schedule. So our next speaker is Dr. Afshin Beheshti. Dr. Afshin Beheshti completed his PhD from Florida State University in physics and made a transition to cancer, systems biology, and radiation biology for his postdoctoral training. In 2014, he became an assistant professor at Tufts University School of Medicine and Tufts Medical Center, where he continued his research as a systems biologist, studying various aspects of cancer, including microRNAs, aging in cancer, cancer drug targets, and development of novel immunotherapy. In April 2017, Dr. Beheshti joined NASA to be part of the Gene Lab project, assisting with developing the platform. In addition, Dr. Beheshti also has obtained grants, including a Trish grant and a space biology grant, where he's conducting research on how microRNAs will impact space biology and potential use for countermeasures to mitigate space radiation and microgravity. Lastly, Dr. Beheshti currently holds an adjunct assistant professor position at Rutgers Robert Wood Johnson Medical School and has a visiting researcher appointment at the Broad Institute. Welcome, Dr. Beheshti.
Thanks, Leo. And I want to thank all the speakers so far. It's been a great session. And yeah, so I'm going to talk about microRNAs. And I think Doug did a wonderful talk before me and also kind of set me up for microRNAs to speak about now. So let me make sure the slide advances. Okay, so what are microRNAs for people who don't know what microRNAs are and why study them? So classically what we thought, let's see if I must, yep. So classically what people had thought before 1990, early 90s and early 2000s is that, you classically had DNA transcribed into RNA and translated into protein. But right about here, around 2000, well, the first microRNA discovered was in worms in 1992, but then right around, there's a big lull, silence where no one paid attention again until 2000s, early 2000s. And once people started paying attention, they started noticing what previously thought that these small 22 nucleotide fragments of RNA was formerly thought debris, that they're actually very important in the biology. And the new understanding is now basically this, which says, microRNAs can be involved in every aspect of what was previously thought to be the classical view. So what are microRNAs? MicroRNAs are basically one type of non-coding RNA. And they basically have 22 nucleotides. They're very stable at this size. They can freely float in every bodily fluid in your body. And well, meaning freely float where they're attached to an argoprotein, or they could be in exosomes also. So either way, they get in and out of cells and organs, and they can either cause good things or bad things to happen in the body. And microRNAs are known to be involved in every aspect of disease, including COVID-19, which I'll get into. Which in my theory you can get into, and people are starting to find out it could be. And microRNAs are not only found in mammals, but in every living organism, plants, and so on. And the interesting thing about microRNAs, they're highly conserved between species. So that's going to become important for COVID-19, when I'll show you in a second. So what is my view of microRNAs? So I'm a systems biologist, as Leo had said. And systems biology view kind of looks at it, a holistic view, top to bottom view. And looks at it as not as an isolated system. So in this case, a microRNA example, if you look at the top panel here, you see that, classically, a lot of people look at microRNAs at one at a time. And when you look at one at a time, like I'm using an example of LET7A, which is one of the first discovered microRNAs to cause, in mammals. And what basically, let's say in the cancer example, people classically think LET7 is a tumor suppressor. So in that case, if they're just looking at that microRNA in their system, looking at it, they're going to decrease the health risks. But, it's never just one thing in a disease. It's always probably multiple things. So then let's say, you look at another microRNA, and in your system, you're only looking at two microRNAs. One is miR21 in this example, which is which is thought to be a cancer-promoting microRNA or an oncomere. And then there's also, the LET7A. So let's say if someone was just looking at these two in a system and they had equal influence on each other, one being the suppressor, one being the promoter, they're going to say there's no difference. Those microRNAs don't really have anything contributing to that disease, which might not really be the case. So in a systems biology view, what we really is a whole system of microRNAs as a signature associated with a certain disease or an impact from the environment. And whether the balance of these microRNAs are going to be, going more promotion or inhibition could cause this to tip to one way or the other to decrease or increase health risks. So that's how I view and that's how I tackle microRNAs. So again, a systems biology approach, another way I look at it, I'm using a tumor example here, which also can apply to any kind of diseases like COVID-19. Classically, when they look at cancers, like you would look at tumor and the microenvironment only. And people concentrate on those things, which is important. But really, when you have any kind of disease, such as a tumor, or in this case, COVID-19, it's impacting the entire body systemically. So why would that happen? One theory that I have is possibly a factor like microRNAs are circulating throughout the body and causing a systemic impact on the host of whether it's a cancer, in this case, COVID-19, to cause it to have, to impact the entire body as a whole. And as I said earlier, this, for circulating microRNAs, because of their size, it can either be in exosomes or free floating. And based on this, they can be conserved across, the multiple organs in the blood to actually cause, impact in multiple places in the body due to that disease. So I'm going to bring up a few examples of how I've done microRNAs and then dive into, the COVID-19. Just so you know that, the way I've been looking at microRNAs and the way impact microRNAs have is not just on COVID-19, but it sort of has the same impact on multiple diseases or multiple arenas of science. So in one case, I'm going to bring up a lymphoma model we looked at. We were able to predict the microRNA circulating signature associated with diffuse large B-cell lymphoma. And in this case, what happened was basically this is on human samples, and like long story short, we did mouse model first. We found a microRNA signature associated with this in the mouse model, and then we translated to actually patient samples. And if you look at down here, you see these are the healthy patients, and this microRNA signature was taken all from the serum of all the patients, so all from the blood, so the circulating factors. And indeed, what we saw is like in healthy people, this signature associated with lymphomas, they're basically non-existent. But once you have the patient samples from either like pre-treatment or progression or on treatment, then they're significantly regulated and upregulated. So this could be a nice prognostic marker or biomarker for this disease, and it detects it long before any known mechanism that we have to actually detect lymphomas. Another example, which I work at KBR and NASA, so I do a lot of space biology research. So I like to show one actually example of space biology work, and then move on to the virus and the proof of principle concept, how it works into virus too. So in space, we have the space radiation impact, the different impact astronauts as a whole. And then we also have microgravity and so on. And if you do a long-term space mission, of course, you're going to get a high dose of radiation, which can impact cells. So again, using the same concepts, I went ahead and predicted a microRNA signature. And what we did was basically then quantify this with space radiation simulated experiments in mice, serum and blood from that, and microgravity experiments. And I won't go into the details because we're about to publish this paper too, but you could always contact me offline. And Sharina, she's just got accepted to graduate school, did a lot of this work, promising graduate students, so keep an eye out for her papers. But what we basically saw is this, that we indeed in the circulating blood with an increasing amount of radiation, this LET you don't have to worry about, but it's basically the increasing impact that energy causes damage on the body has an increasing correlation to microRNAs that are circulating. And in a sense, so again, it's a circulating microRNA signature, and with increasing impact of the space environment, we see that microRNAs associated with the space biology signature is going up. So how does this relate? And this is a little idea of LET, but how does this relate to viruses and microRNAs? So there's been several papers out in the past few years talking about how viruses use microRNAs and vice versa, how microRNAs can try to inhibit viruses. So one thing viruses do, this is just one of the reviews out there, which I like the figures on, that talks, and there's many reviews talking about this, that viruses actually use or hijack microRNA machinery. So what they can do is they can hijack the machinery either for replication or even to evade the immune system. So some of the previous coronavirus work on microRNAs, which this paper had reviewed, talks about that, basically how microRNAs for SARS, this is the COV version, use microRNA machinery to evade the immune system and then further replicate. So make sure my time's okay. Yeah, okay. So then, and the other feature is that microRNAs are highly conserved. So, that in my theory could be possibly used for easy transmission between species. A recent paper that was just put on bioRxiv actually is a computational paper that actually predicted the microRNAs that could be associated with SARS-CoV-2. And this is a very nice paper, which I'll get into in a second, but they actually predicted some microRNAs based on computational methods that could possibly be involved. So going to our research with our covert group that, I talked about earlier and Doug had talked about. So Doug talk really nicely came into this talk. So we initially started analyzing the data, and I had some people here that helped me. Rob Miller from a group had helped me, had done some of the pre-processing for this. The bottom slide got cut off. But what our initial data had shown, which Doug had shown nicely could be involved, is mitochondrial suppression happening. And again, the calcium's up, as Doug has said. So this is like a common theme that happened. And we found that happening in the initial analysis that we have done in the public data that's available from Todd's lab. And Krista and Michael Yoakum have found. So again, I won't go into this too much, but because Doug talked about it a lot. But one other symptom that, is interesting that people have in COVID-19 is the loss of smell and taste, right? And that's, again, related to mitochondrial suppression and then calcium uptake, as we see here. And then I won't go into this stuff here because Doug talked about it nicely, why the elderly population is being affected and diabetes and obese people are being affected more than others, which Doug showed that. So I was able to use my bag of tricks and figure out a predicted microRNA signature based on the data I just showed you that's showing mitochondrial suppression. And what I predicted is these microRNAs. So we look at them as like, okay, I might recognize these from other diseases. I see MIR155 from the lymphoma signatures in there, but it's suppressed. But how does that relate with viruses? And what we see is this. So when you actually start digging the literature, you look at this. So interestingly enough, like MIR10, I mean, you look at specific literature associated with SARS-CoV-2, the COVID-19, it says if MIR10A5B, which is one part of the family of MIR10s, if it's abundant and upregulated, it acts as an antiviral mechanism. But here, based on my data, I'm predicting it to be inhibited, which is not good for the host, but good for the virus to replicate and move on. That bioRxiv paper that had the computational method that predicted a lot of microRNAs related to COVID-19, they said this guy here, MIR2392, should be activated. Interestingly enough, they also reported that this is heavily involved in interspecies transmission, they predict, and replication of the virus. And I predicted, based on the data we initially had, to be activated. These two microRNAs I don't have any literature reference next to, people haven't really associated with SARS-CoV-2 yet. Maybe they are, maybe they're not, but I think since I'm here, they probably are. The inhibition is causing maybe the virus to be able to replicate in the body and affect the host better. And again, these other microRNAs are showing similar things in the literature. The inhibition of them is bad for the host, but good for the virus. So how does this impact, other functions of specific genes? And we see again the mitochondrial suppression happen, targeted by this MIR2392 activity, and the suppression of these also help further decrease the mitochondrial activity. And interestingly enough, when you want to predict how does it impact other functions that are known to to impact the host by SARS-CoV-2, indeed these microRNA suppression activations causes increased inflammation, which Doug talked about happening in COVID-19 patients, decreased immune response, increased fibrosis, which has been, some people have been reporting to happen in the lung based on this virus. So these microRNAs are heavily involved with a lot of activity that's being seen to occur in people getting infected with COVID-19. Interesting enough now is what can we use these as a target? And Doug had talked about MIR181, which is not here, but I could include this in this analysis, and it is involved. So there's some of the predictions I made in the past also. But which of these microRNAs can we use as a target or as a therapy? And interesting enough, there's two aspects of this. One is you got to rely on your statistics that you have. Two is rely on previous knowledge. So although let's say MIR155 is being suppressed here, I wouldn't want to increase the activity again, because although maybe that increasing activity for SARS-CoV-2 might be beneficial, earlier, if you remember from my lymphoma work, MIR155, if you increase that, could be possibly part of the whole signature of lymphoma. So then you could cause side effects of cancer, which we don't want. So that's why you whittled it down based on previous knowledge, previous experience, and previous work that we have done, or me and others, my collaborators have done. So one good target right now might be MIR2392, or MIR124, or maybe even MIR10, because you want to increase that. So if you want to look at further functions of what these impact, one thing Doug talked about, which was a nice lead into my thing, was the toll-like receptors. See, they're heavily involved with that function. There's other functions here. This is some prediction made from microRNAs on a gene ontology pathways. And we see it, people who are familiar with mitochondrial activity and inflammation and things and factors related with SARS-CoV-2 like that, we see definitely that there's, all these functions are really heavily related with those activities that we see in COVID-19 patients. And ACE2, as Chris and others have, Mason and others who again talked earlier have spoken about, these microRNAs do actually target ACE2 also. So it's here. And one thing to note is that MIR2392, you don't see a connectivity here. So one of the downsides to these pathway prediction tools is if there's not that much literature out there to connect them to known genes, of course you're not going to get connectivity. And the higher the number of, you go with the microRNAs, the less is known about them. So MIR2392 right now, there's not much out there, not much people know things about it, other than some virus literature that, these bioarchive people are showing.
Afshin, we got about 2 minutes to finish up and ask questions.
Okay, thanks, Leo. I'm going to finish in 2 minutes. So going back to this bioarchive paper, I'm going to briefly go some of the facts here, but again, ORF8, Doug brought up, and other covert members like Todd has really looked at ORF8. MIR2392, they have predicted is really heavily involved with ORF8, which again is pointing to things this might be a good target. So what are we going to do next? More analysis from Chris Mason's RNA-seq data that he talked about, which we started. More samples to collect, validate microRNAs possibly in the blood to see how it's circulating. The third thing, now we've already started with the Nushri and the Vinu, is designing antagomeres. So you have the MIR2392 is upregulated, and why don't we design an antagomere to target that? And they have methods to that, and they've already started designing that, which is wonderful. And then, as Matt and Doug talked about their models, since they're part of our covert consortium, they're gonna test it in their models to see how that impacts it. There's the other members that are probably watching in our covert model that have other models that we're gonna test with that are here, like Waldemar, Mark, Ray, and Dick. So hopefully we might have some results soon to maybe say microRNAs might be the way to impact or bring the host from having this impact of COVID-19. So these are overall conclusions. And basically I want to say the only way we're going to solve this, as I said in my opening remarks, is working together, everyone working together as an open science platform. Instead of creating the problems, hopefully we'll solve them though. So thanks, Leo, you did great at keeping the time. And also I want to thank Trish they have provided a supplemental fund for my grants and KBR to actually providing some funds for me to actually help, for my efforts to work on this project with Covert and everyone in Covert, which is here. We're a team here and we're all working together. I couldn't be doing all this stuff without them either. And no one, it's a great team, as I say. And anyone who was interested in joining, please contact us. But yeah, so Leo, thank you.
Thank you very much. All right, the top rated question is, have you compared the differences of microRNAs between CoV-2 and other viral infections, say influenza? Are they similar patterns or completely different? And also maybe to other coronaviruses like SARS-1?
Great question. Yeah. So when I compare them to other, so there's, these microRNAs specifically that I've shown that I'm predicting, they're different patterns than other viruses. And then when I look at the literature, everyone's reporting a lot of the microRNA virus experts that are the papers out, they say, for example, for influenza, there'll be a different set of microRNAs that should be associated than let's say MERS or Ebola or or the HEF1 virus. HIV virus. So that's basically the idea is every disease, or in this case, every virus infection, in my opinion, should have a different microRNA signature associated with them.
How about using that for diagnostics? How would that work? Could you make a
So
test
that's
for
the
it?
perfect question. Yeah. So that, I think that, in my opinion, based on the previous work I started the talk with, is that's definitely what I would go for, because if the microRNAs are circulating through the body, that's an easy way. And if the signature is conserved throughout the body, it's going to be the perfect way to use it as a diagnostic to actually see if the patient's infected long before the symptoms are there. So in that case, if it's circulating through their blood draw, or maybe possibly, this is what I'm hoping to get from new samples from various members like Sonia and John Cook at Methodist that they're collecting, they're going to be hopefully collecting soon. Is for example, is it in the urine, saliva? And if this microRNA is truly conserved, like I've shown with my previous work within these different bodily fluids, then maybe all you have to do is, take a urine sample or a blood sample. And then you would know that if they're infected or not 1 or 2 weeks ahead of time. And we still have to show this stuff. This is all a theory and hypothesis right now based on my previous work. But I'm hoping that maybe we could start showing this soon, since they're going to collect samples and we can actually quantify this stuff.
Great, thank you.
Yeah, thanks.
All right, Dr. Fish, if you're ready, we can go ahead and have you load up your slides.
Yes, definitely.
Great. Our next speaker today is going to be Dr. Kathleen, or Katie Fish. Dr. Katie Fish is the Executive Director of the UC San Diego Center for Computational Biology and Bioinformatics in the Department of Medicine, and serves as the co-director of the ACTRI Center for Accelerating Drug Development. Dr. Fish spearheads the development of a vibrant, leading-edge vision for computational biology, bioinformatics support, and infrastructure at the UC San Diego Health Sciences. She has a strong record of collaborative research in computational genomics as a consortium multi-PI for the Omics Data Generation Center, the ODGC, for the NIH Common Fund Acute to Chronic Pain Signatures Program, leading the data management and analysis within the ODGC. She's also the co-director of the NIH San Diego Digestive Diseases Research Center, Microbiomics, and Functional Genomics Core, leading the computational biology core for the center. Dr. Fish is interested in investigating the epitranscriptomics of host response to COVID-19 infection, stemming from her experience investigating epitranscriptomics of myeloproliferative neoplasms and integrated multiomics of HPV-related head and neck cancers. Welcome, Dr. Fish.
Great. Thank you so much, Leo, for the great introduction. And thank you, Afshin and Todd and the entire COVID-19 International Research Team for inviting me to speak here and for being so open and collaborative in this way. So I'd like to first introduce you briefly to the UC San Diego Center for Computational Biology and Bioinformatics, of which I'm the director. We are a collaborative research center in the UCSD School of Medicine. We work both within UCSD and outside. We specialize in all computational genomics assays, primarily focused on the downstream interpretation. We have a great team. Some of them are on the call today and are part of this consortium, and have also been part of the Gene Lab working groups. And so I thank them for the great work that we do and hope that we can contribute to this collaborative. So today I'm going to give you the
Katie?
Yep, sorry.
Sorry to interrupt. You have a blank screen.
Oh no. Okay.
I thought I just mentioned that.
Not good. All right, hold on. Let me stop the share and then let me reshare. Okay. That's not good.
It looks, I'm sorry to jump in, but it looks like there is a drawing app or something like that that I just saw, but maybe just there you go.
You can see this now?
Perfect. Yeah.
Okay. If I play it, can you see it? Can you see that?
Yep.
Yes.
Okay.
Perfect.
All right. So really, back to this. This is the team, UC SD CCBB. And so today I'm going to give the epitranscriptomics perspective on, which is the part that we're working with the team on. And this is very early work, so this is going to be mostly theoretical at this point. And I'd really like to get in the later discussions feedback on thoughts on how this may be involved in COVID-19 infection. So epitranscriptomics is, the broad field of epitranscriptomic RNA regulatory mechanisms. And these play a great role in RNA homeostasis that can influence transcript fate and also different regulatory processes of the cells without actually altering the ribonucleotide sequence or the DNA sequence. There are, in this diagram here, I'm showing 3 main types of epitranscriptomic modifications that are modifications directly onto the RNA. One is methylation, so this is M6A sequencing. RNA editing, which is what I'll be talking about in more detail in a moment, where it's actually editing in either A to I or editing or splicing of the different RNA transcripts. One of the reasons this is interesting is that epitranscriptomics allow rapid modifications to extracellular inputs in a cell. So this allows cells to change very rapidly. Examples of this here on this diagram, extracellular input, in cases of heat shock, in diurnal cycles, microbiome, can cause different modifications, which then result in different cellular consequences. The one that we're going to be focusing on here is RNA editing. This is known to be a response to pathogen invasion, and this has been shown to enhance A to I editing by ADARs. One of the mechanisms that's thought to be involved in this is the recognition of self versus non-self in the cell, and controlling innate immune response. And so this is something we'd like to look into more to investigate the differences in responses to COVID-19 infection. Some of the different specific functional roles for RNA editing in cells. There can be non-coding editing, and these are responsible for enhancing RNA stability. Exon editing, this can be changes in mRNA translation, so actually differences in the proteins that are coded coming from these editing sites. It can be involved, so as we heard from Afshin, they can be involved in microRNA regulation, so disrupting 3' UTR binding if an edited site is there. So that could all be very well connected there. And then also intron editing can be involved in pre-mRNA splicing. And so by analyzing the RNA editing signatures of COVID-19 infection, we hope to be able to elucidate some of these functional consequences. Just kind of a side note that I found interesting when preparing these slides was that RNA editing is now becoming, talked about as a new therapeutic, in addition to CRISPR. They've developed some interesting RNA-guided editing tools that can actually be used to identify and correct RNA modifications as a therapeutic. I'm not going to talk about that, but I thought that was an interesting aside. There's this Nature feature this February talking about this if you're interested in more detail. But the relevance here and why I'm interested in exploring RNA editing. So first, we at the UCSD CCBB do a lot of RNA editing analysis for various collaborations. So we have the pipelines developed, but also to understand the host response to COVID-19 infection and to identify variability in clinical response to see if there is a role of RNA editing and potential development of therapeutics. So RNA editing has been implicated, maintaining self-tolerance, as I mentioned, preventing autoimmunity and the baseline condition, and also controlling cytoplasmic antiviral immunity through double-stranded RNA recognition. We heard about that earlier, so that was a good lead-in. There have been a few studies in influenza A and in dengue virus, where it's been shown that viruses can usurp ADAR1 for their own replication, and ADAR1 is involved in RNA editing. And one study to date has looked at, and this is in bioRxiv, has looked at RNA editing directly on the viral RNA. What I'm interested in doing as part of this team here is to look at the host response, to look at the editing within the human DNA or the human RNA. So this is a demonstration project that's been done on the data kindly put up by Krista and Todd and the team on the OSF website. This is based on the Zhu et al. Paper. I am going to note here, I have a big asterisk that this is a demonstration project. It's only 5 samples, and the RNA was not prepared with human host transcriptomics in mind. And so I don't want any biological interpretation to be done here, but it is a demonstration project of what we plan to do with the larger data set on human transcriptomes. So the first, we downloaded 5 samples. These were from samples from bronchoalveolar lavage fluid at the early stages of the outbreak. We identified RNA editing sites. So first we aligned them, then we call variants on them, and then we cross them with RNA editing databases to identify RNA editing sites. And then downstream of that, we integrated this with the gene expression and the SARS-CoV-2 human protein-protein interaction map that Dr. Wallace showed earlier. So I'm just going to briefly show how we put these into context here. So these are the RNA editing sites that we found in these patients, but again, the asterisk is here. Please don't interpret these. We did find some common editing sites among individuals. They happen to be genes that are involved in innate immunity. However, this is very early stage, and we hope to expand on this as part of this team. Dr. Wallace showed this great map earlier. So this is a protein-protein interaction map that came out of Nevin Krogan's lab at UCSF. One of our CCBB team members, Bryn Rosenthal, helped contribute to this paper. And so using this, we put these, or we hope to put the gene expression, the human transcriptomes, and RNA editing sites into context in proteins, human proteins that are interacting with viral proteins, which is what this map shows here. And so this, again, demonstration project, but is one way to use the interaction maps to overlay and integrate data. So in this case, I went and I overlaid the gene expression that was posted, that was analyzed by Krista and the team using the Callisto transcripts per million, overlaid those with the human interacting proteins of the SARS-CoV-2 viral proteins, and then denoted RNA editing sites that were observed in this data set. But again, do not interpret this at this point, but hopefully in the future, once we get the larger data set and are able to do the broader analysis on many samples, we hope to be able to elucidate the host response to SARS-CoV-2 infection and to identify if RNA editing is playing a role in this cytokine storm of a certain subset of these patients. And so with that, I will wrap up and open it up to questions and the break. Thank you for your time.
Thank you very much. And just a reminder for everyone, the Q&A box is open if anyone has any questions for Dr. Fish. And I can just lead off with one. For the methylation, when you gather the methylation data, can you go over the different ways of gathering the methylation data, like from the sequencing side perspective?
So methylation data, so I'm specifically talking about RNA editing, but I believe actually that Dr. Chris Mason is considering gathering those types of samples using MeRIP-seq. But I'm not prepared to talk about that on my side at this time.
Sure. All right, well, everyone, we have I don't see any questions yet in the chat. I think I'll just go ahead and get us back on time, and we can return back to any questions from this speech during the panel session, since you'll be on the panel. So let us know what questions you have, and we'll return during the panel session. Thank you very much. And now, everyone, that's going to go ahead and kick off a 5-minute break. Also, for those of you who were wondering about the schedule, that's been posted in the chat, so you can see it there. And so we'll see you back in about 5 minutes. Thank you very much, Dr. Fish.
Hello, Todd. Can you hear me well?
I can hear you. I'm
Yeah,
not Todd.
Yes, we can.
Welcome,
Right.
Don.
Um,
I'll pass it over to Leo quick. I think I'm not sure
I
if
just want to
Katie's
make
still.
sure that I can present my slides here.
Yep, you're good. Go ahead and try them out if you need to, but we're good.
Let's see. If I go into presentation mode, what happens? Are you seeing the presentation? Or are you seeing, what are you seeing?
It's just going to share slides. Did you try sharing your slides yet? There's a button on the bottom which says share screen.
It says screen sharing failed to start. Please try again later. Let me get in.
Do we have screen sharing disabled right now? There you go.
Okay, what are you seeing?
We see your slides as they usually are on your desktop, but not in presentation mode.
Now what do you see?
Now we see presentation mode and a lovely picture of Wuhan.
All right.
Dr. Fish, we've got a couple minutes. Sorry, I actually think I got a little bit ahead of myself. I've got two questions for you. If you're around, we can take those now before the panel session.
Oh, sure. Yes.
Okay. Yeah, great. The first question is there a difference between human and coronavirus RNA editing?
We haven't looked at that yet. So I think we'll definitely want to look at that.
Mm-hmm. Okay. And the second question is, are these edited RNAs detected with RNA-seq?
Yes. So if you have high quality RNA-seq that's deeply sequenced, because you need to be able to call variants, then that's the starting material for RNA editing.
Mm-hmm. Great. And actually, here's another one. Do you know if there's a correlation between the number and location of viral mutations with the RNA editing human protein to protein results?
We haven't gotten there yet, but we hope to look at that.
Mm-hmm. Okay. All right. Great.
Okay. Thank you.
Thank you. Thank you.
Yeah, I just want to just speak for 1 minute. This is Todd Trangan again, and I wanted to say for questions, thanks for everyone who submitted questions. Please keep them coming in for the speakers, and apologies for those that can't upvote. But as you submit questions, some of the participants can upvote the questions, and then 3 or 4 of us are actively looking out for the questions with upvotes to ask them. So please keep those questions coming. They've been great thus far, and I will continue to answer questions, and others will as we can. So thanks everyone for submitting questions.
I also want that before we start is if anyone's interested in contacting us, we'll give you our information at the end for Todd and I. So stay tuned for those, just in case you wanted to reach out and are interested in participating in the covert, our team here. Thanks.
All right, I think I'll go ahead and get started with Don's introduction here. Our next speaker is Dr. Don Milton. Dr. Don Milton is a professor of environmental and occupational health at the University of Maryland School of Public Health. He has 40 years of experience in occupational and environmental health research and clinical practice with a focus on bioaerosols. Among his contributions are a mathematical model of airborne infection transmission risk, using simple carbon dioxide measurements, and 3 patents on exhaled breath bioaerosol collection and analysis methods. A recent paper in Nature Medicine using his methods demonstrated the potential for surgical masks to limit shedding of seasonal coronavirus aerosols from infected patients. Welcome, Dr. Milton, and if you're ready, you can go ahead and get started.
Excellent. Thank you. It's a pleasure to join this group today. I'm learning a lot about the biology behind what we're seeing happening on this global scale. And of course, everybody knows this started off in Wuhan.
Yeah, Dr. Milton, you're just letting you know, we can't see your face. If you feel like turning your video on, feel free to.
Well, let's see. I would have to, how do I get out of here so I can even see anything?
That's okay.
If in the show for a second, and to get back to controls, and I don't know why it's not showing me the controls. I can't start video. Okay, there we go. All right.
Yep, perfect. Thank you.
All right. So back to the show. So as you can see here, Wuhan is a beautiful place where this awful virus got started. And we are now 4 months or so into this pandemic, and we're still trying to understand how transmission happens so as to be able to really effectively control transmission without continuing to damage our global economies. And so we can start by thinking about what are the modes of transmission of respiratory infections generally. And many respiratory infections, including this one, are mainly seem to be transmitted by extended close contact. But what happens during extended close contact? Well, we touch things, we share objects, we can also share ballistic droplets generated as we talk or cough or sneeze. And there's been a number of papers recently talking about the bolus of hot gas that emanates from an oral sneeze that can travel quite a distance. But also happening in close proximity is that there are fine particle aerosols generated from our speech and our breath, as well as from coughing and sneezing. And these are much more concentrated close to a source than they are at greater distances if there's some ventilation. So when epidemiologists say that close contact appears to be associated with transmission, it is sometimes assumed, maybe often assumed, that that means fomites, contaminated objects, and large droplets. But it can also mean fine particle aerosols. And one of the things that distinguishes fine particle aerosol exposure from droplets and fomites is where they deposit. And they can deposit much more deeply in the lung than can the large droplets and contact transmission. And that may also be important. The route of entry may be important in the presentation of the clinical appearance. Now, this paper that is one of the earlier papers about COVID-19 published in the New England Journal talked about asymptomatic patient 1. And initially the thinking was, well, this person who came to the business meeting from Shanghai transmitted while she was asymptomatic. But it later turned out, in fact, she had symptoms earlier. And that wasn't necessarily an asymptomatic transmission. But what is much clearer in this paper is that patient 1 didn't develop symptoms for several days after exposure to the visitor from Shanghai. And during his asymptomatic period, he clearly infected 2 other people, one of whom was infected very early on in his incubation of the infection, within hours, it would appear, from when he was exposed. This is very worrisome, and there's been increasing evidence that there are asymptomatic cases who never manifest symptoms and are an important part of the transmission chain. So also there's been this look at the CT scan showing this ground glass appearance, and many of these patients are symptomatic, but some of these changes are being seen in young people with no symptoms. There is another paper showing a 10-year-old with a CT scan with a little bit of this ground glass appearance and no symptoms. What does that ground glass appearance tell us? Well, it tells us that there's infection out in the distal airways in the lung. That's why we have this appearance. We don't see it as being more proximal in large airways. It's in the distal airways. And what is happening in the distal airways is that you have airway closure and reopening events. And this has been now worked on for over 15 years. And this very nice paper from Lydia Marwaska's lab in Queensland really laid out what we think is happening. These breath aerosols were first recognized, in fact, at and characterized a little bit at Los Alamos in doing respirator fit testing with people wearing tight-fitting full-face respirators. And they couldn't understand why some people couldn't get a good fit test, even though they seemed to have be wearing the respirator properly. And it seemed that what was happening was there are some people who are super shedders of droplets from the lung. And it appears what's going on is that those people have lots more of this airway closure. And we'll come back to that again. I hope we have time. Why there are some things like singing that end up generating a lot more aerosol, and that may be part of the mechanism. So again, about 15 years ago, I worked with a team that developed this device we call the Gesundheit 2, because there was something called the Gesundheit at Fort Detrick in the 1960s for collecting cough aerosols. That was a static sampler. This is a dynamic sampler where we are pulling air through this cone at about 120 to 150 liters a minute. And in the throat of the cone is a 5 micron impaction surface, a nozzle, a slit to a surface impactor to collect the larger droplets and aerosol particles. And we then shoot steam into it and chill it immediately to grow droplets and collect everything bigger than about 50 nanometers. And we use this to study surgical masks, but also to really study and characterize the aerosol from flu cases. And in one paper, we showed that people who weren't coughing were still shedding a fair amount of influenza RNA copies, up to well over 10 to the 5th in some cases, and that we could culture this virus from the aerosol using this device, so that there was infectious aerosol being generated by people who weren't coughing, who just sat there for half an hour, recited the alphabet 3 times during that time, and that was the only speaking that they did. So is this relevant? Maybe that's important with flu. Is it relevant to coronaviruses, and particularly these novel ones? So one of the things that's been of concern was this paper from South Korea in 2016 showing that they had positive culture of an air sample outside of a negative pressure room. Now, this was a makeshift negative pressure room. It had a portable negative pressure machine stuck in there blowing air out the window. And so maybe a better designed room would work better, but not only were they detecting RNA, they were culturing MERS in the hallway. So we are concerned about that. We know in SARS-1, there were fecal aerosols in Amoy Gardens that infected large numbers of people. So there's a lot of concern about this. There are now a bunch of papers out in preprint and a couple in peer-reviewed publications demonstrating that there's probably infectious aerosol coming from SARS-CoV cases. And the one that has perhaps gotten the most traction is this paper from the Santarpia et al. At Nebraska, where they were measuring in a quarantine unit and in the isolation unit with more severely sick patients on various days after onset of infection. And they were picking up air samples in the hallway outside of the patient room. They were picking up virus in the air. And the personal samplers worn by the people doing the air sampling were also showing virus, especially in a room where somebody was getting a treatment that may be considered an aerosol generating procedure. So that 100% of the personal air samples and 2/3 of the ambient air samples in the hallways were positive. And this is in a well-designed facility, suggesting that with a lot of air changes and directional airflows, and you're still seeing it, is very concerning. Here is something from a paper that I'm involved with in kind of helping interpret and design these experiments in the high containment units in Singapore, where size selective samplers were placed on tripods on either side of the bed. The air exhaust is behind the head of the bed. And one of these patients was asymptomatic and not coughing, and we still found virus in the air in fine particle aerosols in these rooms. Just for a little perspective, the dorms at the University of Maryland College Park, we published this this year. We have some dorms that have nice ventilation or reasonably good ventilation. I mean, you'd really like to be up at 15 liters per second per person, but we've got something. But only 4 of our 39 dorms look like this. This is a dorm next door that's probably more typical of dorms on our campus, where we have essentially nil in terms of ventilation. And we are very concerned about how do we repopulate our campus when we have no ventilation in many of the residence halls. When you turning to the issue of what can surgical masks do, that was actually the reason we developed this Gesundheit 2, was to test out what ear loop surgical masks could do as source control. And you see here that for flu, it pretty much knocked out all of the coarse particles, but it only dropped the fine particles somewhat. Now, this is a log scale, so this is actually about 2/3. This is one of these cases where one of the reviewers said, well, it only dropped it 2/3, it's meaningless. And the other reviewer said, oh, it dropped it 2/3. This is fantastic. You should make a big deal about this. In this study, we had each person who was studied first wore an ear loop mask and then was tested again without the mask. So we had paired data. In this study that was published 2 weeks ago in Nature Medicine, Nancy wasn't able to get people to stick around in Hong Kong to give 2 samples for half an hour back to back with 10 minutes in between. So some people wore a mask, some people didn't wear a mask, and a few people did both. And I think that is part of why her data looks a little different. But again, for flu, it's actually pretty similar, that it pretty much knocked out the large droplets, and it did something to the fine droplets, but perhaps not as much. For rhinovirus, it didn't seem to work very well for either, although it may have knocked this down a little bit, but it wasn't a significant effect for either coarse or fine droplets. And for coronavirus, it looked like it worked great for everything. I have some questions about if we'd had paired data, would it look better? One of the reasons we hadn't gotten around to publishing this for 5 years after she did the work was we were not really able to explain this, but timing is everything. Now it's in Nature Medicine. So there's often people asking for absolute evidence that airborne transmission plays a role in transmission. But we don't ask for that kind of proof for contact and large droplet transmission. And in fact, there's no evidence that any of these viruses are actually transmitted by direct contact or large droplet spray. Even with rhinovirus, where it's been attempted, there has been evidence of airborne transmission in studies done in Wisconsin. Studies trying to do this in Virginia were not able to reproduce that, and they were not able to get transmitted by fomites. The only transmission that happened by direct contact was when somebody sneezed on their hand and their hand was still wet. Somebody else touched their hand and then immediately inoculated their nose. That worked. Last time I shook hands with somebody whose hand was wet, I tried to remember to wash my hands before I scratched my face.
Dr. Milton, we've got about 3 minutes left for the questions.
So that is my last slide.
Okay.
This is basically describing that in different sources generate different size particles. The aerosol state is very dynamic, where particles can shrink or grow. They have physical fallout. They have biological decay. And then where they land in your respiratory tract depends on what size they are as they enter and how hygroscopic they are and how quickly they grow. Thank you.
All right, awesome. Thank you, Dr. Milton. First, the highest question voting is, when you talk about extended close contact, what exactly is the time scale of extended? What does that constitute, extended close contact?
Well, we see about 10-20% attack rates in households. There's a lot of extended close contact in households, but it's not entirely clear. In rhinovirus studies, they've needed to be in contact for many hours.
How about the impact of temperature and humidity on the airborne transmission potential?
Well, there's not much data on coronavirus in particular. And in influenza, most of the data suggests that at high and low relative humidities, the virus survives pretty well in aerosols, but not so well in mid-range relative humidities. But a recent study from Lindsay Marr and Seema Lakdawala, where they aerosolized influenza virus in a conditioned media with from airway epithelial cells, found that in fact that protected them against desiccation and drying out in that mid zone. So we don't really know. Some of the seasonal data, like what Matt Freeman showed you earlier, suggested, and some of the data from Michigan suggests that there's a reduced transmission in the summer, but there's also a seasonal data from China that suggests that some of these viruses actually circulate just fine in the summer too. So we really don't know how this virus will behave.
All right. And finally, how about from a public policy perspective, say people having to wear, like being forced to wear a mask in public or asked to wear masks in public? Do you think that's necessary? Do you think work sites, restaurants, public settings will have to change in order to accommodate appropriate ventilation and things like that?
Yeah, I think that there will be a lot of change in this over time. And there we'll need to figure out how you have enough distance, how much protection you can get from masks, and use all of these things together to keep control until we have medical countermeasures.
Okay, and final question. What about things like people outside playing sports or trying to get like physical activity during the current confinement? Thoughts on that?
Yeah, I think that jogging while wearing a mask is not necessary and unusually uncomfortable, and I don't recommend it.
All right, thank you very much, Dr. Milton.
But I wouldn't ride in a Peloton in a bicycle race. Not a good idea. You're drafting. If the guy in the lead is infected, you're going to get it.
Thank you very much. And with that, we can go ahead and prep our next speaker, Dr. Fernando Gonzalez. Dr. Gonzalez, if you want to go ahead and start sharing your screen and your video and unmute yourself. Thank you. Our next speaker, Professor Fernando Gonzalez, works at the University of Valencia, Spain, where he is full professor of genetics and leads a research group on molecular epidemiology. He's also appointed to Physiology, a research institute on public health, where he has been working for over 20 years in the molecular epidemiology and evolution of pathogenic viruses and bacteria. His group has obtained the first genome sequences of SARS-CoV-2 from Spain. Please welcome Dr. Fernando Gonzalez.
Okay, thank you. Thanks, Leo, and thanks, Todd, and everybody for inviting me here.
And we can't see your actual face. I don't know if you want to try to start your video up.
Let me.
No, I see it, Leo. I see it.
Okay. Never mind. Sorry. Okay, you're good.
I am here.
Okay, thanks.
Okay. So thanks, as I said again, and I will start by thanking the members of my research groups, I would say in a minute, because what I am going to talk about is a joint project with several Spanish groups, mainly based here in Valencia, in the East Coast, the Mediterranean. And well, I think that the introduction, there have been so many talks earlier than me that the introductions can be really, really short. And the questions that we are all interested in is how have we come from this virus to the current situation, and how long it will take to solve it. And we are all interested in how to stop this. So in my view, and during my research, we have approached all these and similar questions from a very different point of view to those that you have shown so far. And the questions come from the work of many scholars and well-known scientists, although they are quite old in the traditional sense, because the question, our approach is to answer these questions through the lens of evolution. And that doesn't mean that we are not going to use similar tools, but our approach, our concepts, our ideas about how to tackle these problems are slightly different from those that you have been using in your talks and in your daily life. Why evolution? Well, this is not the first pandemics, and most likely won't be the last pandemic. This is not even a major pandemic in terms of effects on human beings, and we have a long history of pandemics since let's say the last 10,000 years approximately. But in the last 10 years, there has been a dramatic change in the way that we approach and we study and we fight these pandemics. I would also include the AIDS pandemics, because although it started earlier than this epidemiology 2.0 era, it has extended until nowadays, and it will unfortunately continue for a while. And I just wanted to mention that the theory and the concepts behind the application of evolutionary theory have been developed, especially in the last 25 years, but most in the last decade. I won't be mentioning any of those, but those are underlying the tools and the results and the interpretation of them that we make. How is this? Well, we share with many of you the use of high throughput sequencing. And you know that high throughput sequencing is something of the last decade in the daily use of many labs. And there are many applications of high throughput sequencing. You have seen many of them. Most of you are also familiar with the bioinformatic pipelines, tools, and programs that allows us to actually analyze this huge amount of data that we are obtaining in the lab. What we usually differ is what we use those data for, and that can make a difference. Many of these applications go almost straightforwardly to the clinic, but the applications that we are mostly interested in are in the reconstruction of close or long evolutionary history of the organisms that we are, or the samples or the sequences that we are dealing with. Okay, so this is what I will be talking about in the coming minutes. So the first use of the sequence of a coronavirus 2 of this new pandemic was to learn that it was a coronavirus. And for that, the authors that published the first report on this new virus, they had to go to to build a phylogenetic tree. In the first phylogenetic tree, they already showed that it was closely related to bat coronaviruses. But, and as we will see in a minute, this does not mean that the human coronavirus comes directly from bats. In fact, previous coronaviruses that are known to affect humans are the primary host are bats, but there are intermediate hosts which in some cases have been identified, and in the current case has not been identified with precision yet. There is a very recent report showing that the most closely related coronavirus in the wild comes from another bat, but it is also closely related to coronaviruses found in Malayan pangolins. The authors of this report don't claim that they have found the origin of the human coronavirus to be the pangolin nor the bat. They suspect that there might be other intermediate hosts because none of these pangolins was found at the Wuhan fish market where the outbreak and the posterior pandemic started. There have been also some claims about the non-natural origin of this coronavirus. I think that the American president has lately claimed some strong views about the artificial origin of this, and many of these claims have to deal with the specific sequences found at the receptor binding domain and which are shared by the human, the bat, and the pangolin coronavirus. And apparently it seems that this provides a perfect, along with the peribasic cleavage site, provides a perfect way of entry into human cells. And probably you know much better than I do about the specificity of this interaction. And in a sense, it is like an exaptation. An exaptation is an adaptation to a different function, but which comes almost like designed for the current function. And this is probably the case for this SARS-CoV-2 entry site. Okay, another question is that this is not an easy virus to analyze. In its evolution, there has been a lot of recombination. And what we don't know yet is whether these recombination events, at least 5 different recombination events are shown here in this preprint, are directly involved in the evolution and emergence of this virus into humans, or these are regular recombination events that happen to be there, and the adaptation to the human host and the transmission among humans has nothing to do with this recombination. Recombination is a recurrent feature in the evolution of many viruses, and it allows them to jump from adaptive peaks to another adaptive peaks in the adaptation landscape. And it has also always to be considered when providing or designing new therapies about to deal with viral infections. So that is something that we have obtained with one or a few genomes, but probably most of you are already familiar, not with these few, but with the now thousands of genomes that are being deposited this database, HISAID, which is designed for the large scale analysis of well, pathogens, but especially of viral pathogens. It was started about a decade ago as a result of the 2009 influenza epidemics, as a way to have a global surveillance of influenza. That changed the minds of many people working in the genomic epidemiology and they decided to share openly their data, their raw data, before even analyzing them. And this has led to an impressive amount of information being deposited there. You can see the summary that I put in the slide about the number of sequences for human influenza, from human influenza viruses. This is a huge effort. Currently for human coronavirus 2, there are about 8,500 complete genomes. And this is their latest evolutionary tree. Well, this is a couple of days ago, evolutionary tree built from those sequences. So our project is more local in scope. This is a Spanish project, which is led by Iñaki Comas and myself. And we are just starting the project. We started sequencing coronavirus genomes from our city, from Valencia, a few weeks ago. But the project that we started as something in our region soon started to grow, and now it has developed into a full national project with more than 40 hospitals involved. Our goal, as you can see, is to obtain about 20,000 sequences during this first year of the epidemic. This is something that we will have to see how it develops. Our initial goal, in any case, is to obtain about 4,000 genome sequences. We have already 1,600 samples. We have completed 100 sequences, which have been already deposited in HISAID, and they are being analyzed in NextStrain. We will see that in a minute. And during these last weeks, we have devoted a lot of time to put all this in running. It is not that simple to start a project like this, and we have had to change a lot of things from what was our usual workflow. In addition, you know that in Spain, in the last 5 weeks, we have been under strong containment measures, and work in the lab for coronavirus is allowed, but not everything is easy to be done under these conditions. So how have we done, and how are we planning to do this? Well, we are relying a lot on the work by this network, the Arctic network, in which they have provided the resources and the documents for the lab wet and the bioinformatic analysis of the sequences produced. Initially, we were doing sequencing with Oxford nanopore mean ions. Now we are shifting to Illumina with MySec, but using still the same Arctic version 3 primers, which will produce a complete genome sequence after priming and after priming and sequencing small overlapping stretches of the genome, so we can finally obtain complete genome sequences for an alignment like the one that you can see at the bottom of the slide. This is something that has to be taken into account when analyzing the raw reads that we will be sharing immediately after we obtain them. So what can we do with this information? And well, I think that many of you already have visited this web page, the NextStrain web page. This is a super bioinformatic tool. It reads, or it takes sequences from the previous database from HISAID. The tool allows to process those sequences. It selects some of them. It discards some others because of quality reasons, because of coverage reasons, and then it includes continuously new sequences into the analysis. The analysis is very complete, but one message that I would like to bring here for you and for everybody is that behind this analysis, there is a lot of theoretical developments of which you need to be aware of in order to make adequate and appropriate interpretations of the results. For instance, we have done, thanks to Giuseppe Dauria, one of the members of the research group, a community for the Spanish sequences. And if we run, let me show you in a minute.
Dr. Gonzalez, I just want to let you know you've got about 3 minutes left for questions.
Yeah,
and for
I will be going fast.
Yep.
Hopefully.
Okay.
Yeah, thanks. Yeah. Okay, so I'm using two screens and it's difficult for me to get sometimes the mouse from one screen to another. Okay, so if we go and play the Spanish sequences, we get like this picture in which the Spanish sequences start, some of them in Valencia, others in Madrid, and apparently the whole of Spain is seeded by these two spots. Okay, so some people have done this type of interpretation based on next strain analysis, and I just wanted to warn that this interpretation has always to consider what's the sampling behind this analysis. With a low sampling, you cannot make any definite conclusions about the way in which a pathogen has entered a region or a place. The same goes along with the different ways in which the virus has entered into Spain. There has been multiple entries, and we have already analyzed this for every country which is represented in next strain. And if there is something that we can conclude is that SARS-CoV-2 does not care about borders, political nor geographical. It is permeable to all the borders. Building phylogenetic trees apparently is easy because we have tools, but we have to be very, very careful about how we interpret these phylogenetic trees. Sometimes you will see this type of trees in which we already see the different lineages of the Spanish samples. But if we, instead of using the cladogram view, we use a proper phylogenetic tree, we see that the picture is very, very different. And this is interesting because it also allows us to spot weird sequences like the one that I am pointing here, in which the number of substitutions that we found there makes it very unlikely to be a reliable sequences. There are a few tens of sequences in the HISA database that we shouldn't consider as reliable because of the analysis of this. One thing that we are very interested is in using this information for the epidemiologists and the public health, and we will be analyzing transmission chains and the contact tracing. And this is the first initial outbreak here in Valencia of the coronavirus. We are currently analyzing, they have analyzed up to 7 generations of transmission, and we have sequences from most of these patients that we will be comparing to their epidemiology analysis. Just to end up, this is part of the things that we are doing, and the plan, our plans for the future, of course, go along with other population genetic analysis of natural selection, acting in the viral genome or doing phylogenomic analysis. And well, I think that this shows a little bit what we are up to, and we will be going to do in the coming months. Thanks a lot for your attention.
Thanks a lot, Fernando. That was really great. I'll take this one, and then I'll let Leo jump in. But
Sure.
it's really great to have the perspective from Spain. So thanks so much for joining. And I'm going to use this opportunity to ask you a question that you asked me 12 years ago on my PhD committee. So Fernando was a member of my PhD committee when I was in Barcelona. And so at the time I was building methods to align and compare multiple genomes. And he asked me, okay, so you've built tools, but what do you think you can learn from comparing all these genomes? And I didn't know, I don't think I had a great response to that question, but I will ask you the same question, Fernando. What do you think we can learn by comparing all of these genomes that you've shown? and how can that help us in our fight against COVID-19?
Well, I've been asked that question quite often in the last 4 weeks, let's say so, because after obtaining the first genome, then many people ask it, and what is that useful for? And there is a short and a long answer. I will use the short one just because of the audience. Surveillance based on genomic information is essential. So I think that the most important tool that we have now for surveillance are genome sequences, be this for checking for new virulence-related mutations, vaccine escape mutants, or simply badly designed primers for diagnostics. All that has to be based on genome information. So obtaining that is essential.
Thanks so much. I wish I would have had that answer when you asked me that question 12 years ago, but now I have it for future reference. And then I guess the last thing I want to say, I just want to thank you for sharing data, because we have been analyzing the genome since the beginning, and it was amazing to reach out to you and you immediately provide data from genomes. So thank you so much for that. This is definitely a community effort. There definitely are not any borders on this. So thank you so much.
Thank you.
Yep, thank you. And we'll just do one quick question from the audience in the interest of time, and we'll move on. Are the sequences that you work with, do you always do de novo assembly or use a reference to guide them? And any thoughts there?
No, we use one of the Wuhan sequences as a reference for mapping the Arctic protocol. That's not, I mean, it might be feasible, but it's simply much easier to have them mapped to the reference genome, and the amount of variation that they show is so low that I mean, I think it is the best alternative.
Okay, great. Thank you. And with that, let's go ahead and start getting the next speaker ready, who is Dr. Sylvan Costas. Dr. Costas, if you want to go ahead and just start sharing your slides and video and audio.
Can you hear me?
Yeah.
Yeah, I can't share. I think it says that the previous speaker needs to stop sharing.
Okay.
Okay, sorry.
Yep. And in the meantime,
as soon as
sure.
There we go. Thank you.
Yep.
Sorry.
Thank you. Our next speaker, Dr. Sylvan Costas, is the project manager for Gene Lab, which initiated the analysis working group focused on analyzing space omics data. This Gene Lab working group started looking at COVID-19 data when the epidemic spread worldwide. It became clear a new group should branch off and focus entirely on COVID-19, at which point COVIRT was created. Dr. Costas has a PhD in nuclear engineering from UC Berkeley and has been doing computational biology for the past 25 years. He'll give a brief overview on some of the modeling directions that this group could take, trying to merge epidemiological data with findings this team will uncover using omics techniques. And as a final note, feel free to reach out to him after this talk if you want to be included in the modeling effort. Welcome, Dr. Costas.
Well, thank you. So I want to thank first Todd and Afshin for giving me like a few minutes to discuss some modeling perspective for this COVIRT. I'm listing below the people who are actually doing this work. Let me just make this disappear. And so we have Dr. Daniel Tizzi from University of Sao Paulo in Brazil, Dr. Vidya Manian from University of Puerto Rico, myself, NASA Ames Research Center, and then we have Dr. Catherine Conley, who is also a NASA Ames astrobiologist, visiting scientist at the Geophysical Lab. We also have Dr. Kai Hernandez, who's a professor at the University of Chicago. And again, this group is being built. The presentation I'm giving you today is really things that we've been thinking about. It's work in progress, and your perspective are most welcome. I'm the project manager for Gene Lab. You can probably see that in the background of my video stream. I used to work before at the Lawrence Berkeley Lab, where I did a lot of modeling, and I'm a senior scientist for doing human research program NASA work. So, we're looking at this data in a simple way. You've seen this data multiple times, but I'm not a big fan of looking at the number of confirmed cases because I think it's a highly biased variables because it really depends on the way you do the testing, the policy of the current state or the current country, the type of test you use. And so what I've been focusing on in the data that we can download from the web is really early kinetic. And here to give you an introduction of the matrix I'm going to use here today is I'm using the 12 first time points of the total number of reported deaths from COVID-19. And when you do this, you clearly see a very classic behavior where you have a nice log. So this is a log scale. I mean, it's a log scale for the y-axis, I'm sorry. And you can see those nice straight line of the initial incidence. And what you can see immediately is the different type of doubling time from these different countries. So here I'm illustrating Spain with a 1.9 days doubling time at the beginning. The time points here are all normalized so that we start about 4 or more deaths, so nothing before. And that helps us compare the different curves. And then you can see at the bottom, we have South Korea, which started with a pretty low slope to start with, and really managed to really break down the pandemic or the epidemic. This is the kind of measurement we can do. Here you can track the number of deaths on that first time point, the doubling time. You can also compare the doubling time towards the end of this 35 course. So here again, I'm only focusing on the 35 first days from that first time point. And you can see how some countries have been very successful at really reducing the doubling time significantly. One thing I've been looking at is the positioning of the full confinement in this curve, and then when we start seeing the curve bending. And for the most part, it takes about, depending who does it, it takes about 15 to 25 days for to see a bending from the original growth curve. One thing that is really disappointing is the US, because we were very lucky. We started with a very low doubling time, and we didn't really take advantage of this. Actually, on the contrary, we actually are the only one who actually show an uptake. And I think, the USA is a bit complicated because we are made of, really 50 small countries. And so to be fair in this assessment, I think what's really reflected here is the fact that there's different cluster taking off at different time. So in a way, the US is actually a great system to study here in this data, because they really are, each state becomes a different cluster. And even I'm going to go further, each county is really what we need to focus on. Just something that I've been, tweeting about over the past few weeks is really this concept of, what we did has really paid off already. And you can see here, I was trying to project from this early time response kinetic, how far we would have gone after 35 days in terms of the number of deaths. We're looking at deaths here. And you can see actually that, from the initial onslaught of the death rate, the most successful country is actually Spain, because they were on their way to millions of deaths, and they very quickly bent that curve. Italy did a good job, so estimated here, and then you can see here that +7 is the time it took them to start going in full lockdown from that initial 4 or 5 deaths. So they were pretty quick at reacting, and they were starting with a very fast doubling rate. Italy did pretty good too, but they started their lockdown at 13, and they saved 295,000 deaths. And you can go on with this metric, 88,000 for China, 30,000 for France. South Korea, not much, but South Korea was starting with a very slow doubling time, and they actually didn't take any lockdown procedure. The one thing that's really a bit alarming again is that with this metric, USA actually didn't save life. Actually, it has an increase of 7,500 lives. So, again, take it with a grain of salt. It's just a way of looking at the data, but different from what you may have seen. Okay, this is the work that Daniel Tiesi has put together. You've seen some of this curve before, but that's interesting. It shows the mortality rate as a function of country, and we see that actually the mortality rate is quite variable between people. The definition here is the total number of deaths confirmed for COVID divided by the total number of confirmed cases with COVID. I'm going to go over this in a minute. This is the same type of data. Very quickly, you can see how there's a very large difference between countries in terms of the mortality rate as a function of time. Something that I thought was very interesting that Daniel put together here is if you look at the last year 2019 influenza, pneumonia mortality, and then other form of death for different states. So here we're looking at Washington State. This is the behavior from last year. This is the behavior from this year. And actually, if you see, we actually see a little bit of a lower number of deaths overall. And if you compare, and so what's interesting, if you look at the pneumonia cases for recently, and then here I placed the shelter in place for Washington State, which was March 23rd. And if you move these guys compared to, previous year, actually it looks like we have more pneumonia than we used to have in the previous year, which I think is going to be probably confirmed in the future work of many people that, we probably are misdiagnosing some of pneumonia to COVID-19. You can see this COVID-19 taking off here. The other thing you can compare is the other thing you can compare is those other type of death, and you can see a very large reduction of other type of death since shelter is in place. So you probably have read some of that also in the news, but it's interesting to observe it here in the data. So if you now look at New York, which has been really New York State, the number here are different. The onset of COVID is so drastic that you can't really make the claim that the number of deaths is lower than the previous year. It's actually really clearly, there's other graph showing that actually it's the biggest amount of deaths. It has passed the death rate that we've seen during 9/11. But again, we still see some features that I showed you previously, like shelter in place was on March 20th. And again, we see some kind of higher pneumonia level. And again, the other type of death has dropped. Again, it has to be done for a longer time to have significance here, but it's interesting. Okay, so as I was making the point, I think working with the US is interesting and working at the state level is interesting. So same exact approach, focusing on the initial kinetic to really compare different states. One thing I did here is actually I went further. I looked at the county level, and each of these dots have a color for different state, but multiple counties here, all the counties we could find. And we're comparing here the doubling time as a function of the density for these different counties. And you see something that you would expect. The doubling time goes smaller as you have a higher density of people. So, let me give you a few pointers here. The thing that's really surprising is that, so first, those 3 points here are Philadelphia, Suffolk, and Hudson as county. And if you focus on that group here, I just was looking at, what is made of these guys, which have very low density of people, but still have a very fast doubling time. What's going on there? This is the list of those this county, sorry. And a lot of them are in Massachusetts and in New Jersey. Again, this really reflects maybe some differences in the way people handle the crisis, and it may also be strain related. I'll come back to this. One very interesting point, I'm not showing you New York City has a density of, let's see, it's a huge density. On this scale, it's at 65,000, so it would be way off. So I didn't show it, but sorry. But it follows the exact same trend. It's a very high doubling time, fast doubling time. But the really point that stands out is San Francisco County, which has a very high density, relatively speaking, and has a very low doubling time. And so this suggests that San Francisco has done a very good job here. So, kudos to San Francisco. Matrix and confounding factors. So we've been trying to look at total deaths as a function of density. That's kind of like my positive control to make sure that the county data we have makes sense. And sure enough, the more dense you are, the more deaths you see. Nothing special here. The thing that really surprised me, when you look at the mortality rate as defined in previous slides as a function of density, you see that the very low density counties have a very low mortality rate. And I don't believe it's correct. And this is really one point I wanted to bring in this presentation is the mortality rate is highly biased from the way, remember when you do the mortality rate, you're dividing by the number of confirmed COVID cases. And again, I think this number is biased. And it reflects probably that in low density areas, they are not doing as much testing, which then makes sense. You're gonna have a high death rate versus the control. And again, confirmed cases here as a function of density, you see this very drastic increase. You expect this, but I think it's more than what you think. So we need to do more work on this, normalized by the total test per county and compare confirmed case to inhabitant per county is work we're trying to do, but we don't have this data yet. I need the information, so I'm trying to dig this. I compare mortality rate to doubling, and there's really no strong correlation, which really suggests that those two metrics are really reflecting different things. Doubling time might be more reflecting transmission and virulence, and the virulence might be more the mortality rate. So more to be done, but the mortality rate needs to be corrected for this bias. Okay, I'm going to move it to Cassie, if you want to unmute yourself and present your slide.
Sure, and my slide is actually following very nicely onto the prior presentation, as well as the work you're doing here, because one of the things that I'm interested in is how difference in location and differences in people's behavior could be influencing the transmission and mortality of the virus. So just to repeat information that was said earlier, this is a particular, on the left side of the slide, there's the radial diagram showing in green a particular isoform of a particular genotype of the spike protein from the Pacific Rim, and on the right side of the slide is from Europe. This is just one location in the viral genome. It's one example. The clades have incorporated all of the locations in the viral genome. And then on the map at the top left of the slide, simply repeating the observation that on the Pacific side of the US, the West Coast, you get mostly sequences from the Pacific Rim, and on the East Coast, you get more viruses that are coming into the US via Europe. In the middle of the country, it's mixed. And actually in the radial diagram, the top half of the radial diagram where the branches are fairly sparse for the US viral sequences that are in this map, in the phylogram, those are mostly from the Midwest and from the less highly sampled parts of the United States. On the lower right, the very dense bit is shown on the right side of the lower left of the full radial diagram, is shown on the right side of the slide. On the upper part, all those orange sequences all the orange dots are from Washington State. In the curve down on the lower right of the slide, this is that lower right of the radial diagram where you see all the sequences coming in from Europe. The green dots are from, as shown in the map in the middle, from the New York area, and those are all replicating. There's a huge amount of viral sequences that were all sampled around March 18th. Whereas in the Washington State, the sampling started much earlier. So you start seeing things that were diverging around 5 February, according to this particular branching. And of course, there's all the caveats about the branching structure. But then you start seeing a lot of samples being entered that were sampled around February 26th. So there's a big difference is when the samples were taken between the West Coast samples and the East Coast samples. And in addition, so that may be sampling bias. It may actually be something to do with the virus activity. But in this 18th March and later lower right-hand sample that came from Europe, there are clades, or there are subsets, branches on the tree that are sampled from the orange from Washington State and the yellow from Idaho, I believe, if I remember correctly. So those might provide examples of replicates of the virus propagating through a community, but different communities. And at least with the Washington State, you have the two different major clades of the virus, the A clade and the B clade. And these may provide tests for evaluating sort of experimental samples for evaluating whether it's just behavior or actually virus spread or sampling bias. So looking at from the next strain data, one can identify subsets of the data that might be useful for further modeling. So, thank you.
Thank you, Cassie.
And Dr. Casas, with that, the schedule now has us at a 5-minute break. If you guys want to go ahead and just kind of overflow into that for another, maybe 2 minutes, and then we'll see if we can get a question in. And for those of you that want to take a break, go ahead. And in 5 minutes, we'll have our next speaker, Catherine Coonrod.
Okay, so we're already in the break. So I'll be quick here. We've been trying to link the divergence from the next trend to the mortality rate and doubling. Right now, we're not seeing it much, but the problem is the next trend data don't have a county location yet, or we could not find them. So we're really looking at the state, and so trying to link the divergence of the strain or other kind of property from the virus to phenotype like doubling and mortality rate is difficult right now, because we need to really get to the county level to have a better match between the cluster of infection and the measurements that we're doing. We're going to use some agent-based model, another approach. And I guess I'll let Vidya present her last slides. well, I guess Vidya is not able to go on. She's just proposing here to use spread detection using flow propagation and prediction of spread. I don't think I will do a good job at presenting our work, and since we're out of time, I'll just then move to the final slides. If you have questions, we'll be happy to take them. And again, if you want to join us, if you have any ideas to include some concept for modeling, contact us. Thanks.
Thank you very much. Vidya, I don't know if you can talk now if you wanted to mention anything on that slide. Are you able to unmute your microphone now?
Theo, I think oh, there she is. Okay.
So yeah, I just wanted to work with community modeling. So we saw that because of social distance, most of the countries were able to lower the curve, right? So here we model a network configuration where each individual has a prescribed degree distribution, which gives rise to links connected at random. So here we have a social network. I mean, not all of them are infected nodes, but we could include those and give weights to the edges based on, how contagious each individual is. We could have virulence signatures included in the social distance as well. So we want to be able to predict the degree of spread in the future as we relax our social distancing. And we could model counties with communities, and communities could have households where the network is interconnected as a click, and each click will have a random link to another household network. So we can train a deep learning network with these different configurations that we have right now from our social distancing scenarios in an individual county with different communities. And we can train the model with the current levels of degrees of spread, and then use it later in the future as we relax our social distancing, where we will have a more denser graph, and then we will be able to predict the spread. So right now we use k-nearest neighbor, and we can expand it to detect communities which overlap and also non-overlap, right? So as we go out in the future, relaxing social distancing, we would have communities that overlap, and we would have a network that's more dense. The next slide, please.
Yeah, and I think with that, I think we need to go ahead and start getting the next speaker set up. Yep, thank you all very much for that talk. Thank you, Dr. Costas. Catherine, are you ready? You can go ahead and start sharing your slides and unmute.
Okay, perfect.
Show your video. Yep, great.
Okay. Does that look right to you?
Looks like I'm seeing presenter view.
Better?
And now it looks good.
Okay, great.
All right. All right, I think I can go ahead and start your intro. Our next speaker is Catherine Coonrod, who is a fifth-year PhD student in Dr. Rebecca Richards-Cordham's lab at Rice University. Her thesis work is on developing point-of-care HPV, DNA, and mRNA tests for use in cervical cancer screening programs in medically underserved areas. Recently, she's been leading a team from the Richards-Cordham lab working towards the development of a point-of-care COVID-19 test. Welcome, Catherine.
Thanks so much, Leo. I've really been enjoying all the talks in this symposium. They've been really interesting, and I'm grateful for the invitation to present on behalf of the Richards-Cordham lab at Rice University and our collaborators.
Yeah.
I'm focusing my talk today on COVID-19 diagnostics for use in medically underserved areas. Reaching critical testing capacity globally remains a major challenge. So South Korea ramped up early and comprehensively and has accordingly managed the number of cases they have seen by most reports. In contrast, the United States, among others, have arguably rolled out testing too slowly and hit several roadblocks in the rollout, including challenges with primer design and supply shortages. In Houston, where we're located, for example, some hospitals have reported shortages of viral RNA extraction kits one week, swabs the next, etc. The challenges faced in high-income countries are expected to be to look much worse in low and middle-income countries. Basically, everywhere, testing capacity needs to be dramatically increased, as several speakers have touched upon today. To respond to the lack of available tests, there is a lot of test development happening globally. The Foundation for Innovative New Diagnostics, or FIND, is maintaining a tracker of tests and their regulatory status. As of yesterday, there were 265 molecular tests in development. 82 of the 265 molecular tests are either fully automated for use in a laboratory, for use near the point of care, or for use at the point of care. 11 of these tests have US FDA emergency use authorizations. To frame the discussion on the diagnostic landscape and what's required to increase testing capacity, it's helpful to consider specific testing contexts. The solution for expanding testing in high complexity clinical labs, for example, will look very different than the solution in rural areas or in labs without consistent power and climate control. This list, spanning from high resource settings to medically underserved settings, encompasses a subset of the sorts of places where expanded testing capacity is critically needed. Starting with high complexity labs, one of the most important considerations is increasing throughput and minimizing hands-on time for laboratory technicians. Going from the manual CDC reverse transcriptase qPCR assay to an instrument like the integrated Roche cobas test shown on the right, for example, allows for about the same order of magnitude of tests per day, but with much less hands-on time, allowing for technicians to work on other tests at the same time and reducing errors in setting up manual PCR reactions. The cobas assay is one of several tests being designed for high complexity labs, and it targets 2 SARS-CoV-2 genes and relies on a large laboratory analyzer. In lower complexity labs, automated instrumentation is helpful to free up laboratory technicians, similar to in high complexity labs. Self-contained test modules are critical to preventing workspace contamination, which can lead to subsequent false positives. Per test cost and throughput should be moderate at this level. And one good solution here is the Gene Expert SARS-CoV-2 assay from Cepheid. This test relies similarly on RT-qPCR and targets the N and E genes of SARS-CoV-2 with a sample processing control built right into the cartridge. The limit of detection is reported as 250 copies per milliliter. And depending on the throughput of the specific instrument, you can see there are several options here, from an instrument that accommodates 1 cartridge to an instrument that accommodates 16. 1 through 16 samples can be run every 45 minutes or so. The test is exceptionally easy to run, requiring just 3 manual steps that take a minute. One of the best aspects from my perspective of the expert systems is that there are over 23,000 systems that are placed globally, largely as part of TB programs and including in medically underserved areas. Assuming test kits will be subsidized and reliably delivered, this system could expand geographic access to testing. The subsidized cost of other expert assays, for example, for TB, HPV, or HIV, range from about 10 to 20 US dollars per test. So the test will likely remain too expensive for widespread use in low-income countries. And further, the reliance on consistent electricity and climate control limit the settings where this test is useful to lower complexity centralized labs. True point of care tests are needed outside of centralized labs. This means the tests need faster turnaround times, lower throughput, and lower instrument costs. One promising solution here that has received an emergency use authorization is the Abbott ID Now test. The test relies on nicking enzyme amplification reaction, or NEAR, an isothermal amplification technology, and it detects down to 125 copies per mil in under a half hour. The test requires 8 manual steps and tests are run 1 at a time. Unfortunately, at least one of our colleagues who has clinical experience with this test is concerned with false negatives, which could be related to sampling at that clinical site, or it could be related to the test itself. Supply of test cartridges and per test cost also remain unclear with this system. There are a number of gaps that remain for medically underserved area. The per test cost of existing solutions remain too high. Weak supply chains will make delivery of test kits and spare parts challenging. RNA purification is required for many point of care approaches. There remains a high degree of reliance on electricity and/or climate control. The tests require highly skilled technicians, and/or the reaction vessels are not contained, leading to workspace contamination. Our work in the field of cervical cancer technology development positions us well to think about expanding testing capacity in medically underserved areas. Over the past several years, we have worked on screening tests that include sample to answer molecular assays that either take the form of a paper and plastic lateral flow device or nucleic acid amplification tests, including instrumentation that is appropriate for use in resource-limited clinical settings. Sorry about that. Further, we have developed instruments for diagnostic follow-up testing in the format of real-time high-resolution imaging to provide actionable clinical results at the point of screening, allowing for immediate treatment. Finally, we have worked toward capacity building by designing training tools and support to increase the number of providers who are trained to carry out the full spectrum of cervical cancer prevention activities. Furthermore, Rebecca Richards-Cordon, who leads our lab, as well as Rice 360 Institute for Global Health, has been working toward building capacity to supply technology and consumables, as well as training biomedical technicians as part of her work on NEST 360. The goal for NEST 360 is to deliver a suite of life-saving technologies for newborns across the African continent in the next 10 years. This intervention is projected to reduce 50% of newborn mortalities that occur in health facilities. Rice 360, along with many global partners, are designing technologies, training biomedical technicians, delivering the technologies, and conducting implementation research. With our background in point-of-care diagnostics development and technology delivery, we have been thinking about how we can leverage those skills to contribute to increased COVID-19 testing capacity. So thinking about point-of-care molecular testing, there are three main steps to go from sample to answer: sample preparation, amplification, and detection. The options listed here are not exhaustive, but include some commonly used approaches to this sort of technology development. The options we are pursuing are highlighted in blue. We aim to develop a test integrated sample prep that relies on isothermal amplification and real-time fluorescence detection. Dr. Chris Mason described the same assay this morning, so I'll keep the description brief. New England Biolabs has developed.
Hey, Catherine, we're having a little bit of some mic issues right now. I don't know if that's, I think that's coming from you, but it sounds okay now.
It sounds okay?
Yeah.
Okay.
Thanks.
I'm not sure what that was, but hopefully it has stopped.
Okay.
Okay, so I'll pick up with Dr. Chris Mason having described this assay this morning. So in short, New England Biolabs has developed a loop-mediated isothermal amplification, or LAMP assay for SARS-CoV-2. And they're partnering with several groups to develop the assay into a validated clinical test. On the left is a colorimetric readout, which was described by Dr. Mason, where reagents change from pink to yellow through a pH-mediated dye in the presence of amplified DNA. The benefit of the colorimetric readout is that the only necessary equipment is a single temperature heater. On the right is a fluorescence readout for the same assay. With fluorescence, instrument complexity is higher, but the benefit of the fluorescence approach is reduced pH dependence, simplifying sample preparation requirements. So with our work, we are validating the fluorescence assay. The quantitative LAMP assay can be run on a standard qPCR thermocycler where available. In addition, we are working with an instrument design and manufacturing company called Axon, located in Australia, to validate their isothermal amplification and real-time detection instruments for this application. These instruments are available at a fraction of the cost of a thermocycler and can accommodate between 1 and 16 tubes. Our strategy is to start with the 2 instruments on the left, which accommodate 16 or 18 tubes, and which are currently commercially available and rely completely on standard consumables. The workflow for these instruments does require a manual sample transfer step for sample preparation. Following validation with these instruments, we plan to transition to a 1-tube instrument shown on the right. And this instrument utilizes a custom consumable that integrates sample preparation directly. An example sample to answer workflow with one of these instruments is shown here. Following sample collection, the swab is placed into a lysis buffer, then transferred to amplification reagents. Isothermal amplification and real-time detection will occur within about 20 minutes at 65 degrees Celsius, leading to a positive or negative result readout. Some practical considerations for test development include sensitivity. We are concerned with reports of false negatives with the rollout of new point of care tests and need to be sure we achieve adequate sensitivity. We will continue validation during test rollout to monitor any changes in sensitivity relative to ideal laboratory conditions. Second, we're considering robustness of both the assay to inhibitory substances and of the instrument to wear and tear. Thinking about supply chains, one of our main questions is, will the consumables compete directly with standard of care tests, and will that lead to shortages? And also, can the consumables reach our collaborators in Mozambique, Malawi, et cetera? And then finally, thinking through some regulatory strategies, the FDA emergency use authorization is necessary but not permanent. What are the short and long-term regulatory solutions to ensuring test access? I've outlined the technical validation for test kit manufacturers for the emergency use authorization here. And I've included FDA's expectations for a developed test, and am happy to discuss more if there are questions on this, but we'll be brief for the sake of time. The 4 key sets of experiments relate to establishing limit of detection in contrived specimens, demonstrating inclusivity for all published SARS-CoV-2 sequences, demonstrating non-reactivity with off-target organisms, and clinical evaluation, where clinical specimens are preferred, but FDA does provide guidelines on how to validate with contrived specimens as needed. Our team's development plan includes a clinical study at MD Anderson here in Houston in collaboration with Dr. Kathleen Schmaler and Dr. Kayur Patel, along with their incredible teams. We are validating reverse transcription quantitative LAMP with synthetic and clinical samples in our own lab. And we are developing and evaluating point of care friendly sample preparation approaches to integrate into a sample to answer workflow. In preliminary validation experiments, our lab has demonstrated a limit of detection on the order of 100 input copies of purified viral genomic RNA isolate, which matches preliminary results from NEB. You can see the amplification curve shown in the lightest blue here of the positive samples. Our next steps are to develop and evaluate a sample preparation method and then to identify the limit of detection as previously described in the emergency use authorization format with contrived specimens. After EUA submission, our plan for scale includes collaborating with industry partners for kit and instrument manufacturing, evaluating test performance against gold standard reference methods in Houston at private and public hospitals, and conducting a pilot evaluation with clinical partners in 5 African countries where our existing research infrastructure and collaborations are strong, including Mozambique, Malawi, Kenya, Tanzania, and Nigeria. I want to take a minute to thank all of the amazing teammates we have working on this project. I especially want to thank Dr. Rebecca Richards-Cordon for her leadership, Dr. Mary Natoli, Megan Chang, Chelsea Smith, and Cy Paul for pushing the research forward. Dr. Richard Schwartz and Vivian Mack for all of their research and administrative support. I also want to thank Dr. Kathleen Schmaler and her team at MD Anderson for making miracles happen and setting up a clinical study so quickly. And finally, we have a lot of technical support from our industry partners at Axon and NEB, and we are also so lucky to work with some amazing astronauts who astound us with their technical knowledge, including Dr. Kate Rubins and Dr. Ellen Baker. And finally, I'll leave off by reiterating our goals for this project. We aim to help implement sustainable solutions in collaboration with our partners globally, including the clinicians and laboratory technicians who work here at this public hospital in Maputo, Mozambique. Our solutions need to provide high quality laboratory results in a format that works for our partners in medically underserved areas. And with that, I just want to say thanks so much for the opportunity to share our work. And at this time, I'd be happy to take any questions.
Great, thank you very much, Catherine. Okay, let's start with this question first. For the current COVID-19 test, or just testing, let's say, is there the process of enriching or culturing the virus in the sample to get the sufficient amount of virus for the test?
In our test, no, it's just lysis and amplification. And really culturing the virus is it's harder to get safety approvals for that sort of work. So for the purposes of our test development, that's not something that we are considering.
Thank you. Are you familiar with the Gene Expert system by any chance? And would you happen to know if that is FDA approved at the moment?
Yes, so I mentioned the Gene Expert system as a good solution for a lower complexity centralized lab. Let's see if I can bring that back up. From what I understand, the expert system has FDA emergency use authorization right now, and I don't know what plans they have to get full FDA approval. But having used the expert system for other tests, I can say very confidently it's very simple to use, and I think it's a great solution. And the main challenge here is the per test cost.
Mm-hmm. How about, do you have any thoughts regarding what is contributing to false negative rates on the different tests?
Yeah, I think there's still a lot of research being done on the best swab types and actual swab products. So it's, from what I know about the tests themselves, I'm not sure if it's a clinical sampling issue or if it's the test itself. It could be any sort of inhibitory substances, especially when you move away from full RNA extraction. It's hard to control for anything that you might be leaving in the sample through your sample preparation phase, which could inhibit the downstream amplification reaction and limit your sensitivity.
Great, thank you. And a lot of these tests are fairly quick. Any idea on why it might take so long, like 4 to 7 days turnaround to get a report on some of these things, like in the real world?
Yeah, well, I think the main answer is sample backlogs. So like LabCorp, for example, at first I think said maybe a 1 to 2 day or a couple day turnaround. And then as more and more, as they started to get inundated, it became 4 to 7 days. So the time to run the test there are very few that take longer than like 3 hours from sample to answer, but it's the volume that these labs are being inundated with that causes the backlog.
Great. All right, well, thank you very much for your talk, and thank you for that.
Thanks so much.
Yep, Dr. Purcell, you're gonna be up next. If you wanna go ahead and start sharing your screen and audio and video. Thank you. Our next speaker is Dr. Jamie Purcell. Dr. Jamie Purcell is the Principal Scientist and Acting Director of R&D and Clinical Affairs at Great Basin Scientific in Salt Lake City, Utah. Jamie leads development, clinical evaluation, and commercialization of in vitro diagnostic tests on the Great Basin Scientific platform. Great Basin is currently developing a sample to result IVD test for the detection of SARS-CoV-2 viral RNA direct from patient specimens. And I'll just say, these talks are all going to be on diagnostics, so if there's some questions that are kind of similar on each, feel free to ask those again for these future speakers. All right, please welcome Dr. Jamie
Sorry, I was muted. Can you hear me okay?
Yeah, you're good.
So that was a really great talk to follow up on. And I apologize, my camera is not working. It's more of a distraction than a benefit at this point. So I'm just leaving it off. So I apologize everyone for that. But Catherine just gave a really great overview of a lot of the same things I'm going to touch on. So I guess you'll get kind of more the commercial side of this perspective. And so as mentioned, I run, slides don't want to advance. There we go. So I run R&D at Great Basin Scientific here in Salt Lake City, and I also do all of our clinical affairs. And I consider myself a scientist first, but my role in the company and in the scientific community is to translate these emerging science and technology advances into something that can be used clinically and is practical and is accessible for diagnostic patient diagnostics, testing, and patient management. So I'm going to give a very similar talk to what Catherine just did, but with a little bit of a different perspective. So when I was asked to put together a talk on RT-PCR, I really felt like I needed to put together a talk on in vitro diagnostics as a field. And so classically, I would say in vitro diagnostics really consists of three major testing products, which are antigen-based tests, antibody-based tests, and then what I work on, which is nucleic acid amplification tests, or NATs. We're also seeing these great emerging technologies coming into the diagnostics field, such as CRISPR, which I think we're going to hear about next, and NGS. But to date, from my knowledge, there's not a lot of broad spread clinical utilization of these technologies yet. So I'm not going to dive very deeply into anything in those fields, but I'm excited that we're talking about it. So I kind of wanted to give you an overview of what it looks like for those of you that don't go into clinical testing labs regularly. Like, what is it like when you walk into those laboratories? And if this slide doesn't give you a little bit of anxiety, then I haven't demonstrated it well, because especially in centralized labs, large reference labs, large hospital labs, they have a multitude of testing platforms for molecular-based detection methods. You have anything from the point of care systems up to these large, gigantic integrated systems like the COBOS system that Catherine just talked about. And you're not going to see point of care systems in a centralized lab, but you could see them in the hospital. So within the same care centers, you've got a centralized testing lab within the hospital, and then in the ER, you can see a point of a care system. And then there's got to be collaboration on the testing and the data and the patient management between those two diagnostic modes or nodules. So generally within a centralized testing lab, when I walk in, I see the full gamut of molecular diagnostic platforms. So you have everything from the near patient platform. And again, I am a scientist. I am not in marketing or business. So the way I striate a market is based on the types of tests that are being ran on the platforms, not necessarily how they fit into the quote unquote market. And so from my perspective, a near patient platform is anything that's going to give you sample to result or load and go. So a lot of these platforms, which is where the expert, which was just touched on in the last talk, would fit. These are sample specimen directly into a disposable cartridge or some type of a consumable for testing directly into the instrument and reporting. They generally work within 60 to 90 minutes. In cost, they're in the moderate to low range. But the compromise with using a near patient platform is your test volumes. You'll also have in the same clinical lab, these large systems that can handle up to 384 specimens at a time, but they tend to be very expensive. The per test cost can be low, but the batch sizes need to be large, so the turnaround time can be large. These are the systems that handle high volume, but they also suffer from the backlog issues that Catherine just touched on. And then within those same exact labs, you've also got what we're seeing, the commercial tests, which is where a company is just providing reagents. They don't have a system essentially. So these are almost platform neutral in that you have some type of an amplification machine in your lab. They're going to sell you the reagents to put on it. You have to put the two together. It's very hands-on, it's very intensive. And then the final category inside a lab is a lab developed test. And these generally emerge when there's absolutely nothing else available, or what is available is too expensive. And so labs are having small R&D teams within their labs that are actually developing tests for use only in that facility, and they're validated within that facility. And again, they're sourcing everything themselves. They're building those tests themselves. They're not really, other than working with commercial vendors, they're not really regulated in any type of way. So as you can see here, it's a very complicated market. There's a lot of different systems. There's a lot of potential here for testing. And again, it really depends on what type of test, how much you can afford to pay for it, what your test volumes are as to what types of these instruments you have in your lab. And so if we get more specifically into nucleic acid tests, so specifically RT-PCR tests, which is what I'm here to talk about today, so for viral detection of SARS-CoV-2. And as Catherine mentioned, there's several ways that the sample gets into the test. Everything coming in is going to be a clinical sample. A little bit later, I'll talk about the wide variety of clinical specimens that are applicable for this specific infectious disease. But the first thing you have to do is open up your viral particle. You can do that mechanically with heat, with enzymes, with chemicals, a combination of mechanical and enzyme, a combination of heat and chemical. There's a variety of ways that companies do that initial lysis step. And then again, as she touched on, you either isolate or you don't isolate, or purify or you don't purify your nucleic acid at that point. The majority of people that are purifying are using a bead purification based technique, which is where they're enriching for the nucleic acid, washing away any contaminants, and then they elute the isolated clean nucleic acid off of the beads. And then what they've got is a very nice clean sample with minimal to no inhibitor compounds in there, and it's usually enriched. So that means it got concentrated for the nucleic acid. And then the next step is, of course, reverse transcription, which is where we turn the RNA into DNA. There are several ways that you can do this. Traditionally, RT takes place using an RT enzyme or reverse transcriptase at a specific temperature, usually low. Then there can be a cleanup step or a wash or a denaturization, and then you would do your amplification. You'd add your PCR reagents and do your amplification. Over time, we've shortened that into what we call one-step PCR, which is now where you have your reverse transcriptase and your PCR polymerase and all the reagents required for both of those in the same mixture. And then you just do your RT at a specific lower temperature, inactivate your RT enzyme, and then perform your PCR. There's no stopping in between, there's no cleanup, it's just all integrated. And then very recently, emerging technology right now is this concept of zero-step PCR, which is actually where you're only using one enzyme. It's a very heat stable reverse transcriptase that can also do PCR. So it's kind of this newly great engineered enzyme. And so you just go straight to PCR. You don't have to do RT anymore. So it's basically RNA to PCR in a very abbreviated period of time. And then when we talk about PCR or amplification, there are a myriad of techniques for doing this too. We all are very familiar with PCR, but as we've touched on today, I've heard multiple of these brought up. There's a lot of isothermal technology still on the market and emerging. So we talked about LAMP. We talked about NEAR, and then there's also HDA, helicase-dependent amplification, and TMA transcription. I don't remember what that stands for right now. Mediated, excuse me, transcription-mediated amplification. So there are a variety of technologies that are using isothermal techniques. And then finally, detection, and again, we've touched on this. You have fluorescence probes, fluorescent dyes, a variety of hybridization types of chemistry available. You have visual readouts. There's a myriad of ways to detect the DNA. And then finally, for a lot of these large integrated systems, there's a step where we've removed the user intervention from data analysis, and the instrument itself is actually performing the data analysis and making what we call the diagnostic call. So there's a lot of algorithm development and diagnostic test development that goes into that final phase, which is turning the DNA detection into an actual actionable result for the patient. And so if we take a step back, and this got touched on much earlier today, this is what the genome of SARS-CoV-2 looks like. The first RT-PCR test that I know that came out, or at least the first well-publicized one, was the one from the World Health Organization. It's a triplex, which means there's 3 targets simultaneously detected. In the RNA-dependent RNase gene, the E, the envelope gene, and the nucleocapsid gene. And then shortly after that, the US CDC released their design, which was actually 3 target regions, again, a triplex in the N gene. And interestingly, I think a lot of us kind of know some of this, there were some design issues with this assay. So as of late March, the CDC has actually revised their recommendations and they've dropped the N3 target. And I can tell you, I have this assay in-house, I run it all the time. N3 is definitely problematic, so I'm glad it got dropped. But the N3 target has been removed. So now the current US CDC cleared test is just the N1 and N2 targets. And then over here on the left, I took a little screen grab yesterday from the WHO website of some of the other released by other countries. And you can see there's a smattering over the entire genome. And then the asterisks at the top kind of designate where I know either commercial tests are being developed or academic researchers have developed tests in these regions. So pretty much we have tests spanning mostly the whole genome. We have a lot of diversity there for the diagnostic targets. And then I wanted to transition over and talk about this in a little bit of detail because I have a unique perspective on this, being I think one of the few people on the call that's heavily working on the commercial side of this. So what is an emergency use authorization? Why is that important? And how is that impacting our pandemic response? So the conditions for an EUA is that you have to have an emergency situation. It has to be declared a public health emergency. There has to be no adequate or approved available alternatives. And what's interesting is these are only temporary approvals. So it justifies emergent use, but that doesn't mean that that test will continue to be a viable commercial product once the pandemic has ceased. And that's really important to understand for manufacturers because there is some risk with seeking an EUA. And the idea is generally you'd get your EUA out and then you'd go for the formal approval, which I'll give you a little compare and contrast on the next slide. And this was interesting to me. They actually released 2 pathways in the guidance for EUA. So the first was for commercial test manufacturers, which is a formal submission through EUA. And then the second one, which I wasn't familiar with this being a pathway through the FDA, but what they have allowed is clinical use of laboratory developed tests if they're developed in a state and then only being used to test patients in that state, which is really interesting. So and a lot of this had to be enacted very quickly in response to what happened, what was, the status in New York. Things just weren't fast enough for that response. So the health labs in New York were given the right to build and validate their own tests to their own criteria, and then they didn't require an EUA submission to test New York patients with those tests. So that was really interesting. The other really interesting part of this guidance is that serology testing is not, and I would like to repeat that, it is not being specifically regulated under the FDA. They are saying as long as you're labeling your test that it should not be used as the sole basis of diagnosis, that it is okay to just go ahead and release serology tests. If you want your serology test to be used as an actual sole diagnostic and the basis for diagnostic decisions, then it does need to go through EUA. So that was kind of an interesting distinction. And then we have a lot of recent historical precedents for this mechanism of initiating an EUA, the Ebola virus, Zika virus, and then even, I don't think people remember this, but coronavirus, MERS in 2013 had an EUA. So we have a lot of familiarity with these mechanisms in the diagnostic industry. And so again, Catherine went over all this really well. So I'm just going to highlight more specifically how this compares to what a normal product would have to go through. So as she mentioned, limit of detection, I found it really interesting that you actually don't have to use the virus itself. Normally you would have to use your target organism. You're allowed to use RNA or the inactivated virus. And I also found it interesting, you only have to submit your most challenging matrix to the FDA. So if you're going for multiple specimens, you only have to give your most challenging matrix to the FDA, which I thought was interesting. Clinical evaluation this is where it differentiates extremely from a normal 510K, which is the general clearance to manufacture and commercialize a test in the United States. Normally, a clinical evaluation is based on patient specimens, and the number of specimens run is related to the prevalence of the disease. You want a multi-regional clinical trial base with multiple sites. You want to get a broad sampling of the populations. The clinical studies are very extensive, and all that is required for these tests right now is 30 contrived positives and 30 contrived negatives. They would like to see it in an artificial in a real clinical matrix, but they're even allowing simulated clinical matrix. So this is pretty much a very, very, very low bar. And I will caution everybody, but there's no such thing as sensitivity and specificity measurements on a test that hasn't been through a clinical evaluation, because we use that term, clinical sensitivity is defined by your test's ability to compete head-to-head with a previously cleared test. And then how well you do versus that test determines your sensitivity. So I think we sometimes confuse sensitivity with limit of detection. And so you're validating your limit of detection, but not necessarily your sensitivity until you've actually run a head-to-head pairwise like clinical study, which aren't required right now for these tests. Inclusivity we touched on in silico. They aren't even requiring us to test anything other than the one thing that's present. Cross-reactivity, which is non-SARS-CoV-2 organisms. There's an in silico portion, and then there's also a wet testing portion, which again, Catherine touched on, but it entails 13 viruses. All of the human coronaviruses are included in this data set, and then 13 bacteria, fungus, all that are found in the upper respiratory system as normal flora, or things that would cause similar symptoms. And then they've also got microbial interference, which are non-upper respiratory types of bacteria and viruses, and then interfering substances. And these are things like nasal spray, cough syrup, allergy medicine, things that could be in a clinical specimen that aren't necessarily relevant to the disease, but could impact your sensitivity and specificity.
Dr. Purcell, you've got about maybe 2.5 minutes left for questions
Okay,
and wrapping up.
Great. So then I will just cruise through this bit. I have different numbers than Catherine because I pulled my stats from a different place, but basically this is how the status of things looked. So if you think about this, we've had about 70 days as an industry to respond to this. We've got 35 tests cleared in the US, so that's about 1 every 2 days. That's pretty good. If you look at the breakdown, there's been 18 cleared in the, authorized in the US that are just reagents and kits. This requires the lab to also source purification reagents. They must have instruments. They must be sourcing everything individually and highly trained users. There are 6 systems. These are the large systems where everything is fully integrated, but the user has to add everything. And then there's 8 sample to results that have been cleared. And in those 8, 3 of them have been given the wave status, which is kind of how we distinguish point of care. So technically 3 would be in the point of care area. And then there have been 3 serology tests cleared. Of the granted specimen types, this is a big deal. The first saliva test of its kind ever that I've known of in molecular diagnostics got cleared a few days ago. That's a big deal. We've only got 1 test on the market right now for sputum, which is interesting. And I haven't seen a single test hit the market for BALs, tracheal aspirates, or stool, which is surprising to me because the FDA actually called for those specimen types. So we have a bit of a gap there. And then if we go back to this question, are the needs being met? I mean, if you take that data and you spread it out across all of the potential instruments within the labs, I think we're doing a pretty good job of covering all of the bases. As you can see here, every major category of testing system does have something right now authorized in the US for use. And then I wanted to touch on our system. So our system is a single use, it's a near patient system. It's a single use test cartridge. Everything's fully integrated. It's less than 1 minute of hands-on time. We have 5 FDA approved products. Our bench type analyzer is portable. All it needs to run is a standard power outlet. Our tests are all about 90-minute tests, 75 to 90 minutes. It's very easy to operate. No one needs training to use this. Unskilled users can use it. The training total setup time is about 20 minutes. And every necessary reagent you need, including all of the waste, is self-contained within a disposable test cartridge. So that helps minimize the contamination risk, which allows us to enable our product usage in non-lab environments. This is what our cartridge looks like. I'm not going to go over it in any detail, but basically it's a molded plastic. It has microfluidic channels. We have all of our reagents in liquid on the cartridge, and then everything else is freeze-dried. And then through a series of chemical reactions on the cartridge, we're able to perform lysis, amplification through RT-PCR, detection. We use a hybridization-based detection technique. And then we have a software algorithm integrated into the instrument that takes that detection image and analyzes it into a test report that gets delivered to the patient. And then we are currently developing a SARS-CoV-2 test in collaboration with a bunch of people. The idea here is that our panel will have 3 to 5 targets on it. So if there's ever any drift in the genomic sequence that invalidates a particular target. Thankfully, we'll have several others on there, and I think that's the idea behind all the multiplexes. And then my last thing I kind of wanted to just throw out there for everyone is looking forward, like what are the next steps of IVD? Because there's responding to a pandemic, and then there's like the next phase, which is like, okay, we've responded, now what? And so we want, you know, to make everything widely available, which means reagent and system availability. Again, Catherine touched on the fact that we're having severe limitations in our availability to get reagents. And then there's the nosocomial considerations. We need to have co-screening for co-infection susceptibility. Recovered, reinfected, asymptomatic. What does that mean from an IVD perspective? And how can we build tests that are able to differentiate between these categories? And then of course, the deep dive technologies that will come online, which really helps us with epidemiology, the variant tracking, and things like that. And so I think there's still a lot of work left to be done even after this initial phase of response is done. So that is the last slide I have, and I think we may have a few seconds for questions.
Yeah, I think in the interest of time, I think we'll probably go ahead and move forward to the next speaker. That's okay. That's okay. Yeah. Thank you very much for that presentation, Dr. Purcell. And Dr. Gao, are you here and ready to start? You can go ahead and share your screen and unmute yourself. Okay, great. Our next speaker is Dr. Sherry Gao. Dr. Sherry Gao is the Ted Law Assistant Professor in the Department of Chemical and Biomolecular Engineering, doctor at Rice University. Dr. Gao's research program lies at the interface of chemical biology and biomolecular engineering with a primary focus on biomolecule discovery and their applications to human health. The main research interest of her group is to discover and develop advanced genome editing agents and delivery systems and apply these genome editing tools as next generation therapeutics to clinical treatment of human genetic diseases. Please welcome Dr. Gao.
Sorry, I forgot to unmute myself. Okay, now it's
You're good. Yep. Okay. Yep.
Okay. Okay. Hi, everyone. Thanks for this great opportunity. And also, I really very surprised to see we have a lot of participants. And myself actually learned so much from all aspects about COVID-19, and this is definitely something everyone is very caring about at current stage. So thanks, Leon, for the introduction, and also thanks, Todd, for the invitation for my chance to give this talk about the CRISPR-based COVID-19 diagnostics. So really thank Jimmy just mentioned that CRISPR is an emerging technology that could be applied to COVID-19 and in general for viral diagnostics too. So just a little bit background about myself that Neil already introduced. So we look into our lab, we look into a lot of genomic information, and we try to use very useful enzyme tools to perform synthetic biology applications, and then particularly applying to human diseases for medication as well as for other applications in the laboratory. So I have been worked with CRISPR in the last, let's say, 7 years, starting in 2013. Ever since my postdoc research now, after I started my own independent lab, I do continuing very interested in CRISPR technologies too. So my lab has done preliminary research about CRISPR diagnostics, but today I think this talk I will be giving a more of the general introduction about the current stage, and then since this is a more emerging technology, so this is a more overview of what the CRISPR diagnostics, especially towards COVID-19 treatment tests. So this is a picture, I think we will see this picture for COVID-19 multiple times. So one thing I, again, want to emphasize here, this is a single RNA genome virus that is in a positive stranded direction. So I want to emphasize that test is very important because this disease, COVID-19, initially will have some flu-like symptoms. People will feel very nervous to see whether I'm getting a flu or I'm actually getting COVID-19. So the symptom onsets average on 6 days. That we currently really facing this difficulty that the diagnosis cases probably is far less than the infected actual cases. So moving to this slide, I think multiple people have already shown, but I again want to show here is this is a phylogenetic analysis about the different COVID strains back in January 2020. And then on the right side, now people started to have more of the sequenced strain of COVID-19 specifically. I think this is around March of 2020. So starting there, we started to see two dominant types of the strains. One is this L type, one is of this S type. And then what I'm really interested in is really see what it called this single nucleotide variants, because this single nucleotide variants is something that CRISPR technology can very easily adjust its working system towards the changing in the single nucleotide variant sequence. So in terms of L and S, and then now I think gradually just in April, I think less than 2 weeks ago, this publication in PNAS also pointing out that in addition to the two of the original S SNPs, which is changing one of the amino acid and the other one did not change the amino acid sequence into the S form, but now there's another mutation already changing the glycine to valine to cause this type C in the coronavirus strains. So here's a summary that I think many people has already talked about nucleic acid-based COVID-19 test. This is also the most popular testing currently. And we have already heard about RT-qPCR as well as isosomal purification. Genomic sequencing, again, this is evolved next generation of the next generation genomic sequence. This is less available towards the common diagnostic. So today, my talk will be most focusing on the CRISPR technology that using these two Cas homolog protein, one called Cas13, one called Cas12. So I will have one actually primarily focusing on Cas13 since due to the limited time. But again, I will have one slide just showing Cas12 application has a publication just showed up yesterday prior to our symposium today. Okay, so we have lots of clinic diagnostic applications. Here I'm showing you a figure and then summarize some of the advantages and as well as some of the less advantages, the disadvantages of this current available diagnostic in addition to CRISPR. That if you can see, all of them involve some sort of the machine. If you see NGS needs a very expensive machine, not saying that qPCR, because of occupancy of the machine, this is one of the problem that why the result coming out so far behind the testing date. So, so now really goes into the point about what is CRISPR. And we are talking about two CRISPR homolog proteins. One is called Cas12, one is called Cas13. So here I'm showing the crystal structure of Cas12 as well as the crystal structure of Cas13. So the difference is about, oh, I think I have definitely have a typo here. The difference about this two protein system is Cas12 is an RNA-guided DNA endonucleases, and this is totally wrong. And Cas13 is the RNA-guided RNA endonucleases. And then please definitely correct this in your memory. So what this two system does is both of them require RNA guide, which we call the cRNA. So this is a short RNA sequence to guide the protein to the location where you want to perform the cutting. So that's the original function of both for both Cas13 and Cas12. So you will see this complementarity region. This is where you tell the protein where to perform this endonucleases activity. So as you can see, both of them requires complementarity of the binding, and then we'll cut the string of desire. So in the case of Cas12, again, is a DNA endonucleases to cut double-strand DNA, and then in Cas12, it's cut single-strand RNA. Okay, so one of the, in addition to the common feature about the Cas12 and the Cas13, I think the other less familiar feature to the public probably this effect, what we call collateral effects. So the collateral effect means when there's a Cas13 protein get expressed, and then it gets combined with its GAD RNA, which is a crate RNA, has this blue region to tell the protein where to go and perform the cutting. So in the Cas13 case, to cut the single strand RNA. So this green molecule is a target RNA for cutting. So after complementarity of the binding, this system gonna be active and perform cutting to break the target single strand RNA into two parts. So what is less known to the public is after it's cut the target DNA, this machinery does not stop. What it do is start to really non-specifically to cut any RNA available adjacent to itself. So this seems a disaster in terms of genome editing, because you don't want your system to cause any of the off-target editing. However, this turn into something can be very useful towards in vitro diagnostics. So for example, here I'm showing Cas12 also has this property of collateral effects, which it means upon binding of the target double-strand DNA, and it performs double-strand cutting. However, the system still continuously cuts any single-strand DNA reporter which is available. So in Cas13 case, it's going to continue to cut any single-strand RNA molecule that is available. So people has been invented this really smart way. What if I just put single-strand RNA or DNA reporters? So in this case, you have a fluorophore and you have a quencher. Before the Cas12 or Cas13 activity, this probe is not going to show in any of the fluorescence. However, upon cutting, this fluorescent probe is going to be released from the quencher and then showing a readout of fluorescence. Okay, so this is the collateral activity. Why this is very important? Because if there's one molecule of the on targets and it causes a machinery turn on of Cas13 or Cas12, and then this non-specific binding is magnitude, orders of magnitude or more, this is a very good collateral effect that cause amplification of the signals. Okay, so this here, I think in the rest of maybe around 10 minutes, if I still have that many minutes, I will talk about 3 of the cases that use Cas13 or Cas12 to make the diagnostic for virus, not specifically for coronavirus, but this has been developed back into 2017. So what it does in the cases using Cas13, it use double-strand DNA or use single-strand RNA. That's your molecule target that it combined the isothermal amplification using this RPA enzyme, that if this translated into double-strand DNA, it get retranscribed back into the single-strand RNA target that can be utilized by the Cas13 detection. So this system has been developed by Feng Zhang in the MIT and the Broad Institute that using this as a report as Sherlock version 1 to cut the fluorophore and the quencher and to report the detection of virus. So one of the things I do want to point out, this system can be used for RNA and DNA detection. However, for RNA detection, it does need translate into transcribe into DNA and transcribe back into single-strand RNA in cases of Cas13. So this is the flow chart as much as I described. So after signal amplification and then in vitro transcription back into the RNA target, this system gonna turn on, and then you can see with no target and with target, you will see a clear fluorescence difference for Sherlock version 1. So further, one characteristic I want to point out, Sherlock system can tolerate one single nucleotide mismatch. However, if you have 2 or more mismatches, this target and the recognition collateral activity will be diminished quite a bit. So this has been applied into diagnosis of the different Zika virus from a different origin. Okay, so further improvement for Sherlock version 2, that's Feng Zhang has also again developed this system that utilized 4 different of the Cas13 protein and also use all different fluorophores to achieve this multiplexing detection of the virus. Okay, and if combined with this CSM6, which is the effector to enhance the Cas13 activity, this is gonna further boost the reaction sensitivity quite a bit. And this has been published just 2 years ago. So in addition to the sensitivity and the multiplexing property, this feature also been developed into a flat lateral flow detection. So now you can visualize the assay result by just reading, and you don't really need any of the instruments. So so this has been also applied to have a detection minutes of around 10 to 100 copy per microliter for the input. So it's quite a bit sensitive. So here is a paper I just read it probably just within this month. It actually compared the Sherlock method with the RT-PCR. So it generally does believe that the Sherlock method, when it detects 2 genes, and then if both of them shown positive, that's gonna report as the positive result. Whereas the RT-PCR seems to have a much less sensitivity compared with the Sherlock method in testing of the clinical samples. However, one of the concerns does raise because the high sensitivity, and then it raises the problem of the specificity, whether this can be really specifically target the sequence, because as again, you can see the tolerance of one base pair mismatching cause a problem of false positive, that this result actually confirmed by genome sequencing. It does not have the COVID-19, however, Sherlock showing that this is a positive result. Okay, so this slide, I think a lot of you, including Todd, is very expert in terms of machine learning designing of this primers. I think in this slide, I just really want to mention that primer design in RT-PCR as well as in CRISPR system, this is very important. I hope this again answer Todd's question to his advisor. This is very important for you guys to study of the metagenomic property to lead us for primer design in RT and as well as CRISPR systems. Okay, finally, there's could be ways to improve, for example, bioinformatics, as I just mentioned. However, also I want to mention that the CRISPR system, generally people still use isothermal amplification, which lengthens the process quite long. Even though CRISPR technology can detect a signal within, let's say within 30 minutes, however, this probably needs another 1 hour to finish all the sample preparation or even longer. So finally, this is the paper I just added. It published yesterday. And then this paper using clinical samples as well as other synthetic samples for COVID-19 detection, but it used Cas12. So Cas12, we did not go to too much of detail, but the principle is more or less similar. It detects the single strand DNA input instead of single strand RNA, as I just mentioned in the Cas13 signal. So what I want to summarize here, and also this is gonna finishing up my talk. And I want to say in my opinion, again, compare with the RT that is widely used and also other detection method, CRISPR could have more of the advantages in terms of that it does use, even when this use isothermal signal amplification, but it does not need a thermocycler, as well as the because of the single nucleotide matching with the target of the pre-RNA and the target RNA, the specificity can still we consider is pretty high. And then lastly, if such as this assay developed here, that lateral flow straps can be really developed for reading out of the result instead of a fluorescence, this could be another improvement for because of the CRISPR technology.
Dr.
So this
Guy,
is the
you've
last
got
slide.
about Okay, yeah, perfect. About
This
about
is
2
my
minutes
last
left.
slide.
Okay, perfect.
It's a summary slide. I just in case some of you are interested in comparison of some of this technology available, I have a nice summarized here. This Sherlock is the main talk of today, and then we talk about detector in the last slides. And there are a couple of others commercially available right now. And I also point out that one of the things they develop is a lateral flow reading out. But when you use a lateral flow, you do lose some of the sensitivity, not the accuracy. But if you use fluorescence, this Sherlock tend to be very highly sensitive in terms of the range. But again, we need to be aware of the false positive coming out with this highly sensitive test. Okay, so that's what I want to talk today, and thanks.
Great, thank you very much. So we'll try to get one quick question in here, and actually the top question I think is a very nice one. So that's all on the diagnostic side for CRISPR, but how about on the viral therapeutics side? Can you talk to the therapeutic side of CRISPR?
Yes, definitely. So as you know, that CRISPR has been developed into to its potential to treat a lot of genetic disease. So especially, and I think my postdoc advisor, David Liu at Harvard, he developed this base editing technology, which has a single nucleotide reversion to reverse single nucleotide morphosis diseases in all this human genetic disease area. So I want to say is we are still addressing a lot of problems for CRISPR. For example, its accuracy, for example, base editing improve the precision of the modification. But for off-target, that's another thing we're really worried. But if you're specifically talking about Cas13 and the Cas12, whether this can be applied to the therapeutic sides of the of COVID-19 specifically, and I think there's also papers coming out. Researchers has been done stuff. I think the one of the recent research I've been seeing is the paper. I think it's still in bioRxiv, but it's talking about targeting a common region of all the coronavirus. So in terms of you need to developing thousands of antibody to treat possibly to treat this disease. However, I think in the CRISPR, you can use one of the common region of the coronavirus, and then hopefully this is a solution to destroy the RNA for the virus in one shot. So, but then again, this is a very preliminary, and then definitely when this can be developed into clinic, I think we'll see this technology. I will see only being very fast developed within the next 7 years, less than 7 years. So we need a longer time.
Great, thank you very much for that. And Dr. Monahan, if Dr. Gao, if you don't mind stop and sharing your screen, and Dr. Monahan, if you want to go ahead and start sharing your screen. Our next speaker is Dr. Lee Monahan. Dr. Lee Monahan is a postdoctoral fellow working in Professor Aaron Darling's lab at the University of Technology, Sydney. He's a biochemist and microbiologist by training, and for the past 5 years has worked primarily on developing novel sample preparation methods for next-generation DNA sequencing. These include low-cost alternatives for existing library preparation methods, as well as new techniques for functional genomic studies in bacteria. Most notably, Lee has played a key role in the development of MorphoSeq, a synthetic long-read technology launched in 2019 by spin-out company LongAs Technologies. Thank you very much. Please welcome Dr. Lee Monahan.
Can you hear me?
Yep, perfect.
Thanks, Leo, for the introduction.
Thank you.
And thanks to the organizers for giving me the chance to speak today. Basically, this may be a slightly unusual talk in that I'm not actually going to be sharing any data with you today. What I will be presenting is an idea, and this is for a method that we are currently developing, which we're hoping will be able to provide not only massively parallel diagnosis of COVID-19, but also whole genome sequencing of the virus. And the reason we want to present this now, even though it's still largely untested, is we only have a small team of people working on this at UTS, and we really like to fast track the development of this method, partly through feedback and collaboration with people that have complementary expertise, and also just by putting the idea out there for people to take it and run with it. So apologies if I'm covering any background that's already been done to death today. It is currently about 5:30 a.m. Sydney time, so I actually missed out on a lot of the earlier talks, but I'm keen to check out the recordings later. To effectively manage this pandemic, it's been suggested that really we would ideally want testing to be done at a whole population scale. But there are significant challenges involved in scaling up current detection methods like RT-qPCR to that kind of level in terms of throughput, as well as access to reagents and instruments. Certainly people are working to address those challenges, and some of that work has actually inspired parts of our approach as well. But really to achieve population scale testing, several groups are now turning their attention to DNA sequencing, and that's probably not surprising given that the current sequencing platforms generate massive amounts of data at fairly low cost. So there are various methods that are now under development, including LAMP-seq, the Octant swab-seq method, and the Hussein et al. Protocol from Howard Salis's group. And these are all fantastic ideas, but one thing they have in common is that they target only a handful of viral amplicons. And so I mean, that's perfectly fine for diagnostic purposes, but we wondered whether we could leverage those ideas and actually expand them further to actually generate whole genome sequences as part of the process. Now the SARS-CoV-2 genome is about 30 kb in length, and so the cost of actually generating the sequence data itself to do whole genome analysis, at scale it starts to become almost negligible on a per sample basis, but it adds a significant amount of value over a simple positive or negative diagnostic, because it gives you a much richer data set that also enables you to do phylogenetics and to better understand the spread of the disease. So if we could do this at scale, it gives us the chance to get a much more complete picture of transmission chains. And when that's combined with contact tracing and other data, it should now allow epidemiologists to design much more targeted interventions rather than the full scale kind of lockdowns that we're currently seeing. So even if we're not able to replace existing methods for diagnosis, if we can come up with a strategy for high throughput genome sequencing, then this will provide a lot of additional benefits that are kind of independent of that. I should also say that phylogenetics and epidemiology is certainly not my strong suit, but we're lucky to have Aaron Darling working on this project, and he'll really be driving the kind of data analysis side of things. So the method we are developing is a massively scaled approach for generating genome sequencing libraries directly from patient samples at low cost. One feature of our method is that we use magnetic microbeads coated with DNA probes to capture viral RNA. And so we incubate these beads in a buffer that lyses the virus and also enables the viral RNA to bind to the probes. We then use a magnet to basically pull these beads out of solution and effectively purify the RNA at the same time. The beads also contain some adapter sequence, which we need for the downstream sequencing, as well as sampled barcodes. And what that allows us to do is to pull many of these barcoded samples together at this stage in the protocol, perhaps hundreds of samples, into a single reverse transcription reaction. And that helps to streamline the protocol as well as reduce costs and supply chain demands. So once we have cDNA in this pooled sample, we can then use that as a template to do a multiplex PCR reaction to generate fragments that are ready for sequencing. And we're planning to use Illumina sequencing for this. So for PCR primers and our original probe sequences, we designed these such that they will create overlapping 200 base pair amplicons that span across the entire SARS-CoV-2 genome. And we used the primal scheme primer design tool for that. We also introduced additional sample barcodes during this PCR stage, shown in gray here and here. And this gives us the potential to combine many of these pooled samples together prior to sequencing to further scale the method from hundreds of samples up to thousands or even millions. So of course, we don't actually know whether this is going to work, but there are a few things that give us at least some confidence. So we previously have experience in developing protocols to generate custom single-stranded DNA constructs on streptavidin-encoded beads. We can leverage that here. We also envisage that we may be able to prepare these beads in advance and supply them to testing labs preloaded into multi-well plates, which would simplify the process. There are also established methods for doing bead-based RNA capture in the literature directly from crude samples, such as human mRNA capture from cell lysates. Our method could also be used with purified RNA as well, which would also simplify things. And one scenario might be for labs that are currently doing, say, qPCR-based diagnostics on extracted samples. They may be able to use our method to kind of supplement that with whole genome sequencing data for positive samples. And then finally, this primal scheme multiplex PCR approach has been really well established for viral genome sequencing, and it is currently being used quite extensively for SARS-CoV-2 as well, albeit at lower throughput. So where are we up to at the moment with this? So far, we've developed a draft protocol, which we deposited on protocols.io. We've also started work on some initial experiments using synthetic viral RNA spiked with human RNA, and also using different buffer conditions that would be compatible with real swab samples. So we're hoping to test and optimize various parameters, including specificity, establishing sensitivity and limits of detection, and also looking for sample crosstalk using negative control samples, which is something that may happen during our reverse transcription reaction with the combined samples, although we do have some ideas for how to mitigate that if it is a problem. We also want to look at the coverage of reads across the genome and perhaps go back and titrate the levels of our different capture and PCR primers to try and normalize for genome coverage. Once we have some kind of working protocol, we're then planning to test that on real patient samples in collaboration with the University of Melbourne. And Aaron's also planning in terms of diagnostics to try and train a machine learning classifier to call samples as positive or negative based on the number of viral reads and where they're mapping in the genome. So lastly, I'd just like to thank everyone that's worked on this project so far, as well as the I3 Institute, who provided some funding to get this off the ground. Please feel free to contact Aaron or myself if you'd like to discuss anything further, and thanks for listening.
Thank you very much, Dr. Monahan. And with that, since we're a little over time, I think we're going to go ahead and have a short break. We are scheduled for a 10-minute break. We're going to pick back up here at 2:45 p.m. Central Time. And I will see you then. We will have 2 panel discussions coming up next, where we'll ask questions from the audience and a mixed bag of questions to each of the panel members and to the group. Thank you.
Yeah, and I'll just thank Lee again. Thank you for getting up so early to present these kind of exciting thoughts and ideas here. So thank you so much for that, and also for sharing some of the protocols and stuff that you just mentioned. So, and I just kind of like to stop here and also thank all of this, before we move kind of to the panel session, thank all of the speakers. I thought that was just an incredible lineup of talks. So thank you to all of the speakers, and thanks to Leo for keeping everyone on time and us on schedule. So just a round of applause for our speakers and Leo. Thank you.
Hey, this is Afshin. I'll add a slide here for the break, Leo. So just in case people come back or leave, they know if we're still on break or not.
Yeah, thank you. Also, one final note, we have the Q&A is still actively going. So if you want to try to get a question in for either of the two panels, the clinical or the computational panels, feel free to go ahead and post a question. And we'll try to get that in there. Thank you. All right, so as everyone's coming back from the break, I'm going to go ahead and start introducing our panel members for the clinical panel discussion. For this panel, we have a few new and returning guests. First, we have Dr. Sonia Viapol. Dr. Sonia Viapol is assistant professor at Houston Methodist Research Institute in Weill Cornell. Houston Methodist was the first site in the nation to perform convalescent plasma therapy for COVID-19 and one of the first 5 sites for remdesivir clinical trials. Dr. Viapol's team will work on understanding the connection between SARS-CoV-2, inflammation, and the gut microbiome using animal models and clinical research. As a quick reminder, we have returning Dr. Don Milton, a professor of environmental and occupational health at the University of Maryland School of Public Health. And we also have returning Dr. Shirsty Agard, the Henry and Emma Meyer Professor Chair in Obstetrics and Gynecology at Baylor College of Medicine and Texas Children's Hospital, as well as serving as the Vice Chair of Research for Obstetrics and Gynecology. And finally, we have Dr. Jem Maydon, who is a research associate in the Mason Lab at Weill Cornell Medicine, the Institute for Computational Biomedicine, and World Quant Initiative for Quantitative Prediction. Dr. Maydon published the first large-scale urban microbiome study by looking at the microbial populations across the New York City subway system, along with other microbial surveillance projects around the world. GEM's main focus is on integrative genomics in cancer and other studies with multimodal data, such as his work combining multi-omics aspects in the NASA Twins project. So welcome to everyone and thank you for joining. And the way that we're going to do this is I'm going to go ahead and just start asking a few individual questions, and then we'll ask some general ones to kind of open it up towards the end. So to start with, Don, a question coming back from the audience for you is, what about some kind of, you talked about your Gesundheit machine or the G2 for collecting aerosols and analyzing them. What about something like a miniaturized G2 for other people to use? Any thoughts on that, or if other people wanted to do something similar?
Well, yeah, there have been other devices developed where you have people blow through a mouthpiece into something. And some of them, there's a device called the Pneumacheck, which is designed to try to exclude air from the dead space, and then collect on a filter would be ways that if you can extract from a filter, or maybe use a gelatin filter or some new novel filters. We're looking at with a bioengineer at some novel biologically inspired filter materials that might enhance recovery of virus. That, you could do something like that. The Gesundheit is designed for a particular purpose, which is to really try to characterize how much aerosol does a person generate by having enough flow to capture, even if they have a surgical mask on and the aerosol's coming out the sides, that you're still going to get it into the cone. And so it's got a particular niche, and also we designed it to try to facilitate culture. I think there's going to, if there hasn't already been, some success with gelatin filters in culture, but it's harder than if you can collect directly into liquid.
Mm-hmm. Dr. Agard, a question for you. Coming from the audience, hydroxychloroquine and drug interactions may enhance cytotoxicity. The age group that is most affected are people over 60 years who may be taking one or more prescriptions for chronic conditions already. Do you have any thoughts on that?
There are two thoughts around the toxicity with hydroxychloroquine. One has to do with the QT prolongation, the QT interval prolongation. So the current recommendations that almost all of us have are to go ahead and do an EKG and make sure somebody's not at risk for long QT syndrome. There's a second issue based upon the Brazilian experience that may be that people experience additional toxicity. As I noted, there was a study back in 2012 that was published in HIV positive patients that showed worsening toxicity. In fact, that study was halted if hydroxychloroquine is used in isolation without antivirals. Whether or not that's going to hold true entirely in COVID is yet unclear. We use a lot of hydroxychloroquine and their equivalents in pregnancy, as in other states with people who have lupus and other phospholipid disorders. And so it's not unheard of. There's some additional issues around some of the medications it gets combined with, again, largely around the long QT interval problems, including azithromycin.
Thank you very much. Dr. Villapol, I'm going to go ahead and ask you two questions here. The first, given your work on the gut microbiome, do you know of any current link between COVID-19 and the effect on the gut microbiome? And then the second, along the lines of your neurological work, can you discuss at all the long-term and short-term neurological effects of SARS-CoV-2? Is it able to infect neuron cells? And if so, are there any specific drug tests going on for such infections? And you may be on mute potentially right now. Actually, I'll just unmute you.
So,
I can hear you now. Perfect.
Good. So the novel coronavirus can show some they show in COVID patients that they cause gastrointestinal symptoms, and as we know that the intestinal change that associates with the gut microbiome alteration. So definitely that is something that we had to explore and look at not only to thinking about treatments, but also for diagnostic. And curiously, these patients, they show 1 or 2 days before the onset of fever, they show this diarrhea and this kind of the change on the gut. So that is something that we should explore. And related with the second question that you asked me about the neurological manifestations, that definitely the data from Wuhan, they show that around the 40% of the patients, they show headaches and nausea and vomits and also confusion. In the population, that the early population, the more severe infection and symptoms, they show cerebral thrombosis and ischemia, or like cerebral hemorrhagic. So definitely there are a link and has to be explored. There are several hypotheses, but we thought that, for example, that the coronavirus can bind the receptors, the AC2 receptors, that there are endothelial cells, and then the vasculature in the brain contains these cells, the endothelial cells, they contain these receptors. So probably that is one of the targets. So there are other things to explore. And when you ask me about the long term, a lot of viral infections, they were associated with neurodegenerative disease, with Alzheimer or Parkinson. And we cannot exclude this one. We don't know if these COVID patients, they have high risk to development neurodegenerative disease in long term. So it's something that we have to study and look at that. Yeah, thank you.
Dr. Maydon, audience question here. Is there a relation between DE genes / enriched GSEA categories and severity?
Yes, we actually do see some more differentially expressed genes in the high viral titers patients. And in relation to that, we also see more pathways related to interferon response and some other known biological response to flu or dengue or other viral infections. So we do see as more enriched in the high viral load patients. Actually, low viral patients seem to have a minor response compared to the others. But I also know that other researchers have shown that viral titers do not seem to significantly affect the duration of the course of the disease. So it might be that just some of the subjects we sequence with high viral titers are actually in a different stage of infection. So they are having a different response than the low viral titers.
Back to Dawn, quick question for you. Is there any estimate on how many particles, like for COVID-19, required to actually transmit during aerosol transmission for a successful infection?
No, I don't think we have any data on that. There may be some data on what sort of MOI you need per cell to initiate infection in cell culture. And if as with other defective or RNA viruses, you tend to have a lot of defective particles. The multiplicity of infection may need to be greater than 1 to have a high probability of initiating infection, which would say something about how many viruses you might need in a droplet for that droplet to be carrying an effective payload. In influenza, there's some data suggesting that it's somewhere around 3, and that might be true. It's an RNA virus, so we'd expect it to be maybe similar to another RNA virus in that respect.
Thank you. Shirsty, I've got 2 more questions here from the audience for you. The first one is basically, I understand that doctors are prescribing erythromycin along with hydroxychloroquine to treat. Are you or anybody else worried that maybe there's like a broad overuse of this and worried about development of antibiotic resistance from overuse?
Yeah, so I think ideally, no. People aren't, let me rephrase this. almost all medication treatment is done under either compassionate use or under heavy ID approval. It's typically limited to only severe cases in inpatient settings. The concomitant use of the azithromycin with the hydroxychloroquine is we're really after kind of the autophagy inhibitors with that, and trying to use antiviral as a way, or use autophagy inhibitors as a way of preventing that viral replication. And that's reserved for very sick people. And so, you know, given the choice between not surviving and potential risk downstream of antibiotic resistance from azithromycin, I think most of us will take the potential risk of azithromycin because these are being used in exceedingly ill patients.
Mm-hmm. And a final question for you. Do you think that the increase in pneumonia deaths is due to COVID-19 cases being misclassified as influenza-like illnesses or bacterial pneumonia, both, or something else?
Yeah, so I don't think there's a lot of evidence that they're bacterial pneumonia. If you look at kind of the classic changes we're seeing on the features we're seeing on chest imaging, I don't think these are misclassifications. There's a very clear progression with an early ground glass appearance on both sides of the chest, and then they may or may not get a consolidated pneumonia, but very quickly they go to ARDS, which is much different than you see with a bacterial pneumonia. We did have a very bad flu season, especially in the US. We had a horrific flu season this year. So I think we're going to have to be a little bit cautious about not over characterizing everyone who is very ill or dies as just having died from COVID. We're going to have to be a little bit cautious with that because this has been a horrendous influenza season across the board. So that's going to be something we'll have to sort out, you know, months and years from now. But I think we ought not be too quick to overcategorize that.
Mm-hmm.
Our vaccine was not effective this year, and actually Australia gave us hints of that last year, that was probably going to be the case.
And Jim, one more question for you. Do you mind just describing it again really quickly? We're about to have a computational panel, so maybe to kind of also set up for that, can you describe the end-to-end process from sample collection to extracting RNA to sequencing and quality control? And actually, I might add in there also, there was a question from the past that we didn't get to answer from an audience member, which is for the clinical perspective, how does it interfere potentially, or how does it work given the biosafety levels when you're working with potential patient samples or samples that you collect?
Sure. So all our samples so far have been coming from our hospital's pathology department. So we get inactivated virus, and from there we get the RNA extracted, and basically we follow regular ribodepletion RNA-seq protocols to do some of our analysis, or otherwise we use the isolated RNA for lab assays or other experiments we are doing. So the QC process is, you know, similar to all other sequencing projects so far. We didn't need to have a BSL because it was already handled by pathology based on, you know, clinical specimens. And computationally, we check the data. So our data so far is coming all from nasopharyngeal swabs, and you might, as you may expect, that we are not just getting the virus, but we are getting a lot of human DNA or human RNA in there, as well as other microbiome data. So based on that, we had to clean the data to separate the reads into first, is it human? Is it coming from SARS-CoV-2? Or is it coming from another microorganisms, which it can happen. We have some of the, you know, regular presence of the epithelial cells, other bacteria, and other viruses, as well as we have some of the, in some cases, certain taxa that might be causing some kind of comorbidity as another infection or pneumonia or another viral disease as well. So we clean those out, and basically for viral evolution, we can just look at the reads that map solely to virus, and we can just look at the human RNA for host response and other data that we are interested in. And the other microbiome data can be used for other purposes as well.
And just to clarify, I think sometimes this is a little bit confusing, and rightly so. So it's a live virus that's a BSL-3. Once it's either embedded in tissue, or it's in Trisolve, or any number of, then it falls down to a BSL-2. So it reclassifies based upon the tissue and the specimen that you're working with.
Great, thank you. And let's wrap this panel up. I want to do one question kind of posed to everyone. I think this is a really good one from the audience. Again, on the clinical perspectives, the question is basically, how do you translate from any kind of in vitro research to then any kind of actual outcomes, let's say. So why don't we start with you, Sonia, and we'll just go around. What are your thoughts on translating from basically research to then how it's going to translate either for your personal work or just in general, and how easy it is, let's say, or hard, or things like that?
Yeah, no, definitely. Everything needs to be connected in some of the bench work we can extrapolate and reproduce in preclinical work with animal models. And for example, now some of the companies, they have the AC2 floss mice that we can use now for these studies that were not available before. So all the kind of the tools that we can go step by step and go in deep and check all the possibilities that they're always good, and the clinical trials is always the last step, of course, but we have to maintain the safety and the effectivity of all the kind of the treatments that we have, and we can do that as in the lab and the bench side is wonderful, and always thinking about the translational science and who that can affect the patients.
Mm-hmm. Don, do you want to go, and then maybe Shirsty, and then finish with Jem if you have any thoughts finally?
Well, we're looking at things, I guess, like the talking about what does the NIAID data on survival of virus in the lab mean? And, they picked one set of conditions. And obviously in the real world, there's lots of different conditions. But it gives us a range of what the half-life of survival is. And I think that's meaningful. And yeah, it'll be pushed up or down a little bit by warmer or colder temperatures and different relative humidities. But it's not going to just suddenly vanish because of a change in temperature or humidity. And that's important. And when we think about what does it mean in terms of the airborne survival of it having a half-life of an hour? Well, the half-life of just air in your room is basically driven by air changes per hour. So if your office has 2 air changes per hour, the half-life is half an hour, which is shorter than the half-life of the virus. So really ventilation is a big factor in whether there's airborne transmission or not.
Yep. And Shirsty, do you want to follow up on the translational component?
Yeah, so I think there's really 2 kind of key aspects to translation. So I think sometimes when, especially those of us are discovery scientists, think about translation, we really think about where innovation is going to fit in here. And I'm not sure that COVID's a great example of an innovation problem. It's a public health problem. And so kind of the first level of translation is really how do we solve 2 competing public health issues. One is around preventing transmission of the virus, especially as we come down off this curve. But the second is we have to get the economy up and going. And it's not just because of we need to have jobs, it's also because we're going to hit a cusp pretty quickly where all of the other patients we take care of every single day who don't have COVID-19 are going to have worse death and outcomes. COVID-19 doesn't cure cancer, it doesn't cure preeclampsia, it doesn't cure preterm birth. And if we can't get some of these public health issues solved, we're going to have huge confounding of effects here. So I think issues that Don talked about in terms of the built environment and how we're going to come around that, issues around understanding how can we quickly isolate people, so find those people who are infected and their contacts, and so that we can get things back up and running in a good phased in approach that's safe. You know, the term has been used, we need to let the science drive the public health. The problem is we don't really know what that is right now. And so to me, some of the major translational breakthroughs are going to be, how can we rapidly track and facilitate this? That's going to be dependent upon really reliable testing and a large scale approach. I'm not sure a vaccine's going to save us here. If we look back at SARS-CoV-1, those neutralizing antibodies in SARS-CoV-1 patients were gone by about 2 years post-infection. So I'm not sure a vaccine's going to get us there. I think it's going to be good old-fashioned public health, identify, isolate cases and their contacts.
Thank you. And okay, we're pretty much out of time. Jem, do you want to make any final notes on any of that?
Yeah, I agree with Dr. Agard. So we are right now working with some collaborators who are trying to find drugs, but I feel most of the impact will be on basically these public health measures, because if we can reduce the amount of transmission and the amount of people who are getting sick and basically stop the health system from getting overloaded, I think that will be the best thing we can do. And that's why we are trying to work on, these available, cheap, quick testing. I think when testing becomes more available, then we can do, smaller scale quarantines, maybe not just in a country scale. I think that'll probably keep the economy going, as well as keeping the infection at a controllable level.
Yep. All right. Well, thank you very much, everyone, for agreeing to do this and being here. Thank you. For our next panel, this is going to be a more computational panel. We have Dr. Rob Meller. Dr. Robert Meller is an associate professor in the Department of Neurobiology at the Morehouse School of Medicine in Atlanta, Georgia. He's part of the translational stroke research team investigating racial disparities in stroke and stroke diagnosis. He runs the sequencing facility at the school and has expertise in blood transcriptome profiling and the use of mathematical modeling for diagnostic prediction, with current research funded by NIH and NSF. We also have Dr. Allison Heath, who is the Director of Data, Technology, and Innovation at the Children's Hospital of Philadelphia, also known as CHOP, where she is responsible for providing technical and collaborative leadership to build platforms for data-driven discovery. She supervises the data products and platforms team, which performs basic and applied research in data-intensive computing, bioinformatics, cloud computing, and related areas. Since joining CHOP in 2017, Allison and her team have been working on solving big data challenges in the unique context of pediatric diseases, including leading the development of the Kids First Data Resource Center. We also have Dr. Deanne Taylor, the Director of Bioinformatics, also at CHOP, and Assistant Research Professor at UPenn. Her interests range from genomics to metabolic modeling, and has worked with Adam Resnick and Allison Heath as part of the Kids First Data Resource Center, is part of the Human Cell Atlas, and also collaborates with the NASA Gene Lab. We have returning Dr. Katie Fish, the Executive Director of the UC San Diego Center for Computational Biology and Bioinformatics, and the co-director of the ACTRI Center for Accelerating Drug Development. And our last panelist is Dr. Krista Turnus. Dr. Turnus is Science and Technology Advisor at Signature Science in Austin, Texas. She's provided technical expertise and managerial support for a number of the company's genomics and bioinformatics projects, including being a co-PI on IARPA's FUN-GCAT program. In addition to FUN-GCAT, she's led multiple large multi-institutional teams developing and optimizing metagenomic workflows for microbial forensics, biosecurity, biosurveillance, and public health applications, and is currently working to analyze RNA sequences from human genomes, human microbiomes, and environmental swabs as part of the covert research team. So thank you everyone for being here and welcome. And along the lines of the IARPA FUN-GCAT program, why don't we start with Dr. Turnus? So going back to an audience question here, engineered versus non-engineered, what computational methods would you say are out there, and how could these potentially be used to tell, or can we tell if this was engineered or non-engineered, let's say? And I don't hear you, so it's possible that you may be muted. Let's see. Krista, are you there? There you go.
Yes. Can you
Okay.
hear me now?
Yes.
All right. Sorry about that.
No problem.
Right. So there has been a lot of rumors going around about whether or not SARS-CoV-2 may have been artificially engineered in a laboratory. And I have not seen any evidence yet that looks credible for that claim. I think there that SARS-CoV-2 has not been engineered. Typically, when we're asking the question of whether or not a pathogen has been engineered, we look for synthetic constructs or regions of the genome that do not follow a pattern of natural evolutionary variation. And I think we've seen a lot of great presentations here today of phylogenetic trees and understanding of how this virus naturally evolved. And no evidence yet to show that there's anything abnormal about the sequence that couldn't have been derived naturally. In particular, there was a rumor about HIV being engineered into the virus, and I think that has been debunked at this point, in that those sequences that were being claimed to have been derived from HIV are actually shared with many different viruses and not unique to HIV. So that is not true.
Mm-hmm. Thank you. Deanne, can you describe the typical computational platforms and procedures, let's say from raw reads to gene expression, for analyzing, let's say, swab or lung tissue samples?
It depends on what the experimental question is. A typical RNA-seq experiment would require, correct extraction of the data on the laboratory side. For example, depletion of DNA or somehow selecting out of RNA from the sample, and then having that go to, usually manufacturers produce those kits, like RNA-easy or something like that, and then going to a library preparation, and then on something like sequencing, like on Illumina or some other platform. And once we get the data, it's in a series of raw reads with quality scores. There are various types of analysis software that can then align, or what we call map, the reads to the genome. And the genome, depending on the organism, we use something called HG38 now for humans. If you're aligning RNA or other types of sequence to to microbial organisms, there's other methods for that. Then we quantify the reads, we quantify the counts or the number of features present. In that case, we would use a genome reference, some sort of reference that tells us where the genes are. And then we count how many of these fragments have mapped to each gene. And then we quantify those using a combination of, using quality scores and other methods to make sure that what we're seeing is real. And then using those quantified genes or features, we could then do comparisons between groups. So at the end of it, you end up with basically a vector, or the equivalent of Excel, a column of gene counts for every sample. And then you can do comparisons from sample group to see what kind of differences you have between groups. So that's a typical workflow. And depending, if you're doing single cell, it's a slightly different workflow with different processing steps along the way. But what I just described is a typical bulk sequencing workflow.
Mm-hmm. Thank you. Rob, does gene expression for the same genes vary significantly across sample type? Are you able to speak to that?
Yeah, so we know that some tissues, by definition, they will selectively express certain genes. So for example, neuronal tissue to have the phenotype of a neuron, they will express specific neuron-specific genes. There are some genes though that seem to be quite commonly expressed amongst most cells. And then you'll probably find that some of the non-coding RNA seem very specific to different cell types as well.
Mm-hmm. Thank you. Allison, question for you. Given the need for rapid data sharing between institutions with what's going on across the country and across the world, what are your thoughts there as far as new cooperative strategies for big data and data sharing specifically in response to COVID-19, and any thoughts you have about basically rapid data sharing?
Yeah, I think it's been really fascinating to see, at this point in time where we've really built all these platforms that are connecting to each other, both in terms of the national landscape and the international landscape. There's a lot of, interesting initiatives going on. And I think COVID has really shown that the urgency of speed, a lot of times when we're building these platforms, we aren't necessarily talking about, new processes or new ways, but how do we accelerate the current processes that are already happening naturally? And it's, I think, been really great to see that a lot of times, if you have some urgency, that a lot of the barriers are not technical a lot of times. We have a lot of the technology now today to do this very rapidly, very quickly in terms of the data side. And really rethinking, you know, how do we do this kind of in a manner that really accelerates the impact of these different platforms. And it's going to be really great to see. I think we're starting to get some of the data in that can be publicly accessible, and how do we get, you know, more eyes on that and have kind of rapid turnaround on how we can analyze and share this data. And I think there's just been a lot of initiatives. This is one. I know there's a big group of folks working with NCATS to share kind of the clinical data. And we have all these platforms to do them both in a secure and in a public way, depending on exactly what kind of data we're managing.
Mm-hmm. And Katie, I was wondering if you maybe wanted to follow up at all on that as far as data access and sharing goes. And then also I was wondering if you could speak to the role of epitranscriptomics, again, just sort of in solving the COVID-19 puzzle, if you want to follow up any on that.
So, yes. So I think that the open data sharing, this is a really great, this whole symposium is a really great example of how allowing for, you know, open access with a tight group of researchers really accelerates the science, right? Because a lot of these things, as was just mentioned, we have the tools to process them rapidly now, but there are, you know, computational hurdles. There are, I mean, just downloading the data, finding the right data set, making sure it's processed in the right way. We just had this discussion yesterday on the Slack channel. Those are the barriers that take the time. But if, you know, these are posted, we have a whole team looking at them, we can really ask the biological questions very rapidly, which I think is a really great thing that everyone's doing. So I really appreciate that. And again, I think the same for epitranscriptomics or any of these techniques that can be based upon these, you know, basic data types, because, okay, they've already been pre-processed. So now we don't have to spend, you know, a few days per sample running the same pipelines over and over again, right? We can consolidate, run a streamlined pipeline, and then everyone can use, you know, standardized data products from that.
Yeah, thank you. Let's go back to Krista. Along these same lines, what publicly available data sets are out there? Do the publicly available data sets, do they have things like metadata and things like controls? Are those available, or just raw sequencing data? What's available?
Yeah, so I was interested in that question a couple months ago and started trying to collect the initial RNA-seq data sets that were coming out of Wuhan, China. And that was really valuable. So first of all, thank you to everyone who is making their data public. That is incredibly valuable for all of us. And RNA-seq data sets in particular from COVID-19 patients are a really rich data source of potential therapeutic targets. So, you know, bottom line, thank you for making your data open. I completely agree with everything that Katie just said. But the data that is publicly available right now does have some limitations in that the samples were prepared in different ways before sequencing, and those aren't always directly comparable depending on the way that the library prep was done. We also have a limited number of controls, so not every study posted corresponding controls for their COVID-19 patients, which makes it very difficult to do the statistical analysis when you don't have those controls. And overall, we just have a very limited number of samples. So as you can imagine, the statistical conclusions that we can come to are limited when you have such a small sample set. So we're really looking forward to getting our hands on more data that Allison and others are helping us to organize and distribute throughout the group. And I'm sure it'll be available to the wider scientific community soon too. But again, it's all about making that data open and sharing and communicating with each other about the data itself.
Yeah, thank you. Rob, what mitochondrial signatures, if any, have you seen in the COVID-19 RNA-seq data?
So we did do some analysis of the data that Krista just mentioned. These were samples from China. We did see some changes in some of the mitochondrial gene expression patterns. The problem is, as we say, because the controls were made in a slightly different way, we're kind of a little reluctant to basically bet the house on the results that we see at this stage. They are interesting, they're promising. We also do see some interesting point mutations as well within the mitochondria that might suggest some changes that could be involved in the disease. But clearly what we need is just more data at this stage. There were very few samples, there are very few controls, and to really get the depth of data to allow us to make a confident interpretation of the data statistically, we just really need more data. And some of the samples that Chris described earlier on today, the numbers that they're able to generate for these further studies should actually then give us the statistical power to make confident predictions based on the data that we see, or at least more confident predictions based on the data that we expect to see.
Great, thank you. So let's wrap this up with one final question. Allison, we'll start with you, and then we can check anyone else who wants to follow up on this one. What are your thoughts on essentially just data-driven discovery for COVID-19 therapies, just in general, and for helping in the effort for COVID-19? Do we have, like, for instance, do we have enough publicly available data to perform data-driven analyses and discoveries, or what are your thoughts there?
Yeah, I mean, I think the other panelists have already kind of emphasized that, you know, we have initial sets of data. You know, I think having more of that, and I think obviously the control question that a lot of people are raising, I think is important. I think I'd probably hand that off to others to answer that way, but you know, from my standpoint, especially I think linking that to what's happening clinically is going to be important. You know, it's always one thing to look at the genomic data and understand the landscape, but I think we've always found when we're looking at that data, really having, you know, what's happening, you know, outcomes, symptoms, and things like that to really relate that to has always been an important aspect, as well as, you know, kind of multimodal data. You know, we've been talking a lot about the RNA-seq data here, but, you know, I think there's a lot of other genomics, there may be imaging, lab tests, other things that we can bring to bear, other molecular assays. So I think that will be important as well. But, I think others can definitely speak towards this topic.
Yeah, if we can just do real briefly, we're running just a little bit behind time. So if everyone could maybe just give a real brief follow-up to that. Katie, maybe starting with you, if you have any follow-up.
Yeah, so I think that's exactly right. Having a prospectively designed study, I think, is very helpful. So in this case, with Dr. Mason's samples that will be coming, having the controls, having everything run on the same platform will be very helpful instead of piecing together, you know, a few samples here, a few samples there. But as we've said, I mean, it is still very valuable effort to mine publicly available data that is out there.
Yep. Krista, did you want to?
Yeah, I would just add that I don't think we can afford to not do data-driven discovery for this outbreak and this virus, because there are so many unknowns and so many things we're still trying to understand. One of the powerful ideas behind data-driven discovery is that you don't go in with any assumptions. You just see what the data tells you. And I think that is really needed right now for COVID-19.
Rob?
I think with the larger expanded data sets, what we're going to see is a shift as well from the traditional discovery approaches to using more AI type approaches. Once your data becomes large enough, AI becomes super powerful. And that's where I think we're then going to start seeing some very important discoveries that we wouldn't have thought of because of just the way that we look at data typically within our own area of knowledge. I think when we then have the ability to take a data set or large public data sets with enough metadata, you can then use some computing to really go and find patterns that we just aren't seeing because they're just not so obvious. And that's in using the entire omics field to fill in this data. So we've then got as complex data on the omics side as we do with the patient data. And that's when I think really AI type approaches will really revolutionize the way that we're going to apply data for identifying targets for medicine.
Great. And with that, Deanne, if you want to add any final comments, and with that, we'll go ahead and wrap up the panel. Allison and Deanne are actually speaking next, so we can transition straight into that. And then Deanne, I'll let you take over.
I'd like to add, if I could add, one of the things that we don't have is enough data, and I'm not very hopeful that we'll ever have enough data after this unless people are taking samples and freezing them right now prospectively. I'm worried that we're not going to have enough data to do enough host viral response studies. And I think what we're lacking in the world, and probably in our nation and in the world as a whole, is a prospective standard operating procedure between the major centers of research and hospitals to say, next time we have this kind of outbreak, that this is what we're going to do as a group. So have something that the NIH has, or the Department of Homeland Security, or somebody with an agreed on set of money, an agreed on set of sample collection protocols to have in place. And if an emergency committee has to come together at the first sign of one of these outbreaks and come together and say, this is what we need to do this prospective study, and send it directly into the research hands of this panel of people that can then go and spider out and determine, and start petitioning out research resources, that would be the best thing that we could do in the future. Because this won't be, someone else has pointed out, and at least in this call, and if not other calls, that this is not the first time we're going to run into this kind of situation.
Yep, thank you very much. And with that, I will let you two, Dr. Taylor and Dr. Heath, I will let you two go ahead and take over with the final talk for the day. And then we'll have closing remarks, and that'll be the end. Thank you, everyone, for staying with us.
Thank you. Can you see my screen okay?
Yeah.
Great. I'm aware that we are definitely running a little bit over time. I think today has been just awesome to hear. And, from our perspective, I think we've heard a lot on how do we rapidly, reuse and repurpose a lot of the biotechnology platforms. I'm going to talk a little bit about how we are trying to, point one of our data platforms towards this. And one of the big takeaways I want to say is I'm going to talk about Cavatica today with Deanne. But I really view this as an example of just one platform that can really be brought to bear on this effort. I think many platforms offer a lot of resources there. People have local resources. We really just want to make sure that we're using them as effectively as possible. And this is just one example of how we're standing up a collaborative place for people to do some of especially the raw RNA-seq data processing. So just a little bit of background here.
Sorry, I don't think you're in presentation mode. I think we're still seeing your desktop slides. Maybe you'll have to share the actual presentation when it's up, the window.
I see it. Thank you.
Sure.
Okay. Are you seeing the presentation mode now? No.
No. You kind of have to call it up, then go back to Zoom and share the screen. It's kind of a pain.
That is very strange.
Yeah, we can see the small slides on the left. I think maybe if you do like a view, present or something along those lines. Potentially.
He's got present up. If you want, I can try.
I see. Okay. I got it. There we go.
Yeah.
Yes. Okay.
Yep.
How many Zoom calls do you have to be on to get this right? So Cavatica has been around for a couple years now. We originally launched it to support pediatric disease data, but it's really grown into a generalized cloud-based platform where we can share data, workflows, and results. And it's been a really great partnership with Seven Bridges Genomics to stand this up. As I was saying before, there's a lot of these initiatives on how do we interoperate, how do we make sure this data flows between the different platforms. We are just one of many emerging platforms doing this. And I think this is really a model towards the question asked earlier of how do we do this in a rapid fashion is really kind of federated interoperability. It's in production. We've run over 200,000 workflows. Most recently, we've been using it very heavily for a project you heard about, the Kids First Data Resource Center, where we are processing tens of thousands of whole genomes and thousands of RNA-seq samples. For this effort, we were really kind of brought to bear because we want to have different collaborative projects that can be public or private with different resources. We want to help support folks who want to do this on the cloud, who maybe don't have local resources or want to be able to rapidly stand up a Jupyter R environment close to the raw data or to the outputs and really be able to bring the analysis to the data. So where we're at right now is we are focused on some of the Mason Lab samples, as you heard earlier today, getting that loaded. I think we recently finished that and are testing that out right now with some of the groups. And the very first part is some of the publicly accessible ones. Cavatica is a secure platform. We process a lot of human genomic data, including clinical samples. And next is really some of the controlled access data, and solidifying the process for access there. I'll show quick snapshots of some of the data and platforms that are now available. It is on AWS, so there are charges, but they're kind of just passed through from AWS. And there's also features to use spot instances and to restart pipelines to help keep the cost down. So one of the best things about it is really being able to reuse tools and pipelines. And so towards the idea of, how do I know that I'm using the same pipeline? How do I know that the data has been processed the same way? Platforms such as Cavatica allow you to share these pipelines, be able to see exactly the logs and provenance of everything that happened. And we've been standing up some initial standard ones to do RNA-seq pipelines. The team here at CHOP has brought on some of the new tools like Kraken that we didn't have before pretty quickly, and hopefully to bear to help analyze this data uniformly. I'll just mention anyone can create an account here. You can also, with your email address, you can also use, we have an integration with NIH's ERA Commons ID. You don't have to use this, but if you do, it will actually provide access to other controlled access data sets that you might have available. I'm going to breeze through this because I really want to get to Deanne's section about some of the analysis features, but just to suffice to say, there's a lot of, kind of Dropboxy like features that allow you to have different projects, allow different members, allow people to have different files and access, request apps, copy apps, bring your own data, etc. And, we've really been working on the different tiers of this, kind of some screenshots, but, again, you can log in and check it out yourself. Being able to do members, see the different tasks that you're doing, look at different data sets that are on the platform. And then really, I'll turn it over to Deanne to talk a little bit more about the workflow and tools that are available.
Do you want to drive or?
Since it took us so long, I'll go ahead and drive. Just tell me when to go next.
You can go ahead. Yeah. So you can do the next screen if you want. There's a lot of tools in Cavatica to allow us to wrap our different workflows. Docker, you can Dockerize your tools, and we use the common workflow language, which is has all the specification necessary to really define a pipeline, and that's operating inside the Cavatica as tools inside the Cavatica platform. Next slide. So the sharing, so there's the ability to share over 400 public tools and workflows from 7bridges or CHOP or people that are inside of Cavatica. Those tools and workflows are able to be looked through the selection on the apps. When you go into Cavatica, there's an app selection, and there's a bunch of tools that you can use to build your own workflow. You can literally search for them in a search box, and there'll be an autocomplete, and you can start looking for different tools. Next slide. So there's a workflow editor, and this is not animating, but that's okay. There is a workflow editor that allows you to choose your project, find your data, and then through a drag and drop interface, which looks really cool, like one of those drag and drop graphs where you can have a node and then your connector and another node, and you can start to string your tools together and set the parameters at each tool as you go, and create a graphical model of your pipeline. And it is a physically useful pipeline because it will, in the back end, it will execute the pipeline as you've quote drawn it, or you could also define it on the command line. Next slide. Interesting that that's not changing. Do we have the next slide yet?
On my screen, I'm seeing the bring your own tools slide that not showing.
Oh no, I'm not. It's not changing here. It just says maybe we should go back to the original. It's not changing, Allison.
Yeah,
at all.
it's looking frozen on my end as well here.
Okay.
Let me see. Do you want me to try to?
Yeah, maybe if you have it up on yours.
Yeah, let's try. Let me go to. How let me share screen. You'll have to release share screens, Allison, and then I'll try mine.
Okay, I don't know if it's something going on with my Zoom. I actually stopped sharing.
Okay. I think Leo, you'll have to do something.
Yeah. Let's see here. I'm trying here. Stop video does not actually seem to be doing anything at the moment.
I think Zoom's having a problem then.
Deanne, I've enabled that a panelist can share when someone else is sharing. Can you try to share again? And if it doesn't work,
it's just telling me I cannot start screen share while the other participant is sharing, and I still see her screen. So
okay.
I think it thinks that she's still sharing.
We maybe Zoom is tired. Yeah, I'm not sure.
Yeah, I can go on and we can put these up.
Yeah.
We can put these slides up as well for anyone who's interested. We have a bring your own tools. There's a way that you can actually create a tool and you can wrap your own tool. So there's ways to do that easily. And we also have ways of integrating common workflow language. And actually for the real geeks among us, there's ways to actually edit this. You can actually look at this, the workflow language code to make sure that's what you want. And I know I'm skipping through really quick without graphics. The whole point of all this though is reproducibility. Can we wrap your tools and other people's tools, our tools? Can we create workflows? Can we recalculate things and version the things that we've done in a reproducible way? Is there a way for us to share this data among each other and to share our projects with other people and to share data with other people? That's the most important thing with this. And on top of that, all of this is once you get your workflow completed, we have interactive analyses that have been integrated on top of Cavatica so that once you do your analysis and you know where your data is, you can actually generate an instance of an R Studio or a Jupyter Notebook server and calculate on the instance. You can calculate against the files generated in Cavatica. It's all in the cloud. And then you can call up a pretty powerful, pretty powerful notebook and do your analysis on the data without ever having to move the data around. This is helpful probably mostly for the people who don't have strong compute resources or who don't want to move large amounts of data around. Probably with summary files, it's not such a big deal. But if you're doing things with IGV or you want to do some calculations on BAMs, then you'd have to start moving larger files around and maybe it's easier to do it on the cloud. So there is a data cruncher built in.
And Deanne, with that, I don't know if you want to make any maybe final concluding remarks. We're a little bit over time at this point, and then we'll conclude here.
No, we just want to thank the people involved with Cavatica, Adam Resnick, obviously, and Allison, who are leading a lot of the Cavatica work at D3B at CHOP. And then there's a whole group of D3B people at CHOP and also in DBHI who have been involved in various aspects of the data analysis, and also at Seven Bridges Genomics. It's Michele Mattioni and Jack Giovanna at Seven Bridges who've helped with this. And I think I've left a whole bunch of names out, but I can't get their slide up, so you'll have to take my word for it.
Yeah, no problem. Well, thank you very much to both of you. Thank you, and for bearing with that. And with that, this is the end of the formal symposium. The only thing left is we're going to have a few closing remarks from Todd Trangan and Afshin Beheshti. I'd also like to go ahead and thank both of them for letting me help out today. And I'd just like to say thank you for everyone who helped make this happen and for presenting, especially the presenters and the panel members. And thank you everyone for tuning in.
All right, so I guess I could start. Yeah, so thanks, Todd, or I should say thanks, Leo, for being such a great moderator and keeping us pretty much on track. So I just want to have this acknowledgement slide here, and thank everyone and give some information. So, our Twitter page and our Facebook page is there if you want to follow us. We probably put updates on there and our most current papers and research being put out, ways to contact us. In general, if you want to contact me or Todd, our emails are here. Any COVID related questions or general questions like that. Thanks to all the speakers and the symposium participants, because all you guys listening have come down. And special thanks to Krista because she's one of our founding members too. So she's leading the charge, and Deanne and Eliza for like doing some of the helping us today with the talk, and all the covert members on the side here, of course, because that's where we're heading towards. So Todd.
Thanks, Ashvin. Yeah, I just want to now just go kind of similar note here. Just an acknowledgement slide is all I have. Going back to the one, I have an update for everyone. Well, first of all, I just want to thank all the attendees for hanging out with us for most of the day. We started off with around 650 or so online. We ended up with over 1,000 attendees registering for the event today, which just blew my mind. So thank you for your interest. We started off for the first few hours. We were well over 600 attendees or participants on the meeting. And we're ending still above 200. We were at 300. So throughout the day, we had hundreds of people tuning in, checking this out, which is just amazing. So thanks everyone for your interest. Thank you for submitting questions. Sorry for those that we didn't get a chance to respond to the questions, but we're going to save all of them. We've tried to respond to as many as possible. Thanks to all the speakers, of course, and the panelists. I thought the panel discussion was great. And yeah, thanks, Ashvin. And then I just want to shout out to obviously Leo. He did an amazing job moderating the event. He's, so keeping us on time and everything. So with that, yeah, I think this was a great experience, and I hope everyone participating benefited from attending and enjoyed the material. I can't thank the speakers enough for bringing some really great content. And yeah, this was a wonderful experience. And with that, I'll conclude and we'll follow up with some material. We have the recording I'll be sharing online soon. We'll have some additional materials we'll be sharing, and that's all I'll say. Ashvin, any last?
Yeah, one thing you brought up, Todd, where can people find the recording of it once we're ready to share that? Because I know I got some questions about that.
Yeah, well, that's a good question.
Tell attendees about that. Yeah.
Yeah, so it's recording and saving on Zoom, right? And then from there, there's a URL I'll post. I'm not exactly sure what the location will be, but there'll be a URL I'll share with everyone who registered for the meeting, and I can email that out.
Can we also, Todd, can we also put up on the agenda that everyone had the links on the original agenda, maybe?
Yeah.
Or that people had that might be helpful. I don't know if people can see it on the screen yet, but
yeah, I will. You're saying do you want me to go back to the agenda now, or do you just saying post it there?
I think we're good. We could probably wrap up.
Yeah. I'll post it there though. That's a great idea. I'll post it on the agenda and I'll follow up. But that was a recurring question. I want to make sure everyone has the information where they can find the recording. And with that, Ashvin, any final comments? And if not,
No, I think it is good. Yeah, thanks for everyone being here.
Okay. All right. Thanks everyone.
Bye.